audible, visible? Hello. Hi. Can't see you, hear you. Can you see me, hear me? Hi everyone, can you see me, hear me? Please let me know in the chat section, guys. Do let me know in the chat section. Hi everyone, good evening. Welcome to Unacademy NEET English. Welcome to your favorite series, that is Game of NEET and today we are going to start with the class 11 syllabus, and we are going to start the most interesting, but yeah, you people consider it as a difficult topic as well, that is biomolecules. So this is what we are going to master today. We are going to master the biomolecules, right? It is very important chapter. It is very important for your chemistry as well, and this chapter is very important for your molecular basis of inheritance. Okay, if you understand this chapter, if you understand biochemistry, then the genetics part is also very easy for you, isn't it? So how are you people? How are you? Good evening, good evening, good evening guys, good evening. What's up? Yes, why number of students are so less? Quickly share the session link with your friends, invite more and more students, right? And hit the like button people. And if you are new to our channel, you know what you have to do. Quickly subscribe to our channel. Subscribe to this amazing channel. Look at the amazing content here, people. See, I hope you all have seen this video. Big change in NCERT. What is that big change? You'll get the clarity of everything. Okay, and in this video I specifically mentioned that what exactly is that old NCERT. Okay, trust me people, if you will check the new NCERT, the updated one, the rationalized content, and if you will check the NCERT of last year, you'll be like, "Yare yaar, ma'am, why teachers are making so many videos on that?" Trust me on that. This is what you people are going to say. Ma'am, why teachers are making so many videos on that? In my NCERT, everything is same like new NCERT. Only that morpho-structural part and that biotechnology, tissue culture part is there. Why? This is what you people are going to see. Literally, this is what you people are going to do. Okay? Okay? So do check this video. It is going to be very helpful for you. Okay? And people here, I have added in the description box of this video, I have added the official syllabus as well. So ultimately this syllabus you have to master. Okay, in the description box of the video you will get the syllabus. So this is the NEET UG approved final syllabus. This is the official document that I have added there in the description box. Fine, fine. So are you ready to start the session? Tell me, are you ready to start the session? And guys, if you are the part of our NEET Endgame 2.0, then good. If you are not, then you know what you have to do. You know what you have to do. Be the part of, right? Be the part of that batch as soon as possible. Here in the description box of this video, you will get the link above, right? You will get the link. So can we start? Can we start? Yes? Can we just start? Tell me people. So what do you know about the biomolecules? Let me check your knowledge first. Let me check your knowledge first. What do you know about the biomolecules? Tell me, what do you know about it? Molecules of life, okay. Building blocks of life, okay. Molecules present in relative abundance of carbon and hydrogen. Whatever you people know, answer it. Today I want the session to be interactive. I want your energy should be high, and quickly hit the like button. This is the chapter that we are going to master today. That is your biomolecule, people. Okay, this is the chapter that we are going to master, and trust me, after this lecture you are going to love this chapter. This is one of the good chapters, I'll say. And my actually I love biochemistry. To be very honest, I love biochemistry. right? And if you understand it once, now there is no need to cram them again and again. That's what I love about this chapter, especially for the genetics. Okay, so be no doubt, I know you people are here with your NCERT, you people are here with your notebook, but today you have to be open-minded as well. Okay, so let's talk about the chapter biomolecules. I'll tell you why I just said that you have to be open-minded. But whenever this chapter came in your mind now, according to you, that structure part is there, right? There are the structures that you have to revise. You think of that enzyme wala topic, right? You think here, yaar, I have to remember this, I have to remember that, but you do not focus on the logic here. You do not focus on the concept here. That's why you hate this chapter. Okay, now bache, what are we going to do in this chapter? If you will read this chapter directly from NCERT, you will get confused. Okay, in NCERT na, this chapter is mixed. Okay, just say they have given amino acids, then they have given lipids. Okay, and in the starting they didn't talk about the carbohydrates, the basics of carbohydrates. They didn't mention in the NCERT the basics of carbohydrates. They directly started the story from the polysaccharides. Okay, so these are the things due to which students, they find it difficult, they don't understand it. And especially if you are from CBSE board, if you in your class 11th you have studied in CBSE board, so you do not have that proper flow of this chapter. Okay, that is the problem actually. That is the problem. So today, what are we going to do in this chapter? Let me tell you. We will discuss the basics of biomolecules. Right? I'll tell you the basics of biomolecules first. Then people, we'll be talking about the chemical analysis, elemental analysis. We'll be talking about the chemical analysis, elemental analysis. Then bache, we will discuss primary and secondary metabolites also. But before primary and secondary metabolites, we will discuss the basics of carbohydrates. Okay, I'll teach you the basics of carbohydrates, then we will move towards the polysaccharide. That's how we are going to complete this chapter. Okay, so carbohydrates, their basics, then polysaccharide, then amino acids, then protein, then lipids, then nucleic acid. That will be the flow. And finally, the enzymes. Okay, finally the enzymes. That is how we have to cover this chapter. This is what you have to do. Clear? Finally, at the end, we will be talking about the enzymes. That is how we are going to cover this chapter. So let's start this chapter, the chapter number 9, of course, biomolecules. So the first thing is what is the meaning of biomolecule? Of course, what are these, right? What are these biomolecules? Of course, this is the first thing. Now bache, when you talk about the living organism, even when you talk about the non-living organism, you can take any example, living, non-living. Okay, you can take any example, living organism or the non-living organism. It's your choice. So obviously we are made up of something. We are made up of something. We are made up of elements, right? In the biology, when in general we talk about the biology, how do we start? This is what we say: cell. Cell is going to form the tissue. Tissue is going to form group, like group of cell will form tissue. Then tissues will form organ. Organs will form organ system. Right? Organs will form organ system. And finally, there will be an organism. That's how we discuss it. Yes or no? That's how we discuss it. Now, when you talk about a cell, what do we discuss in a cell? We say na, that it is the protoplasm which is the physical basis of life. Yes or no, people? This is what we discuss. It is the protoplasm which is the physical basis of life. What is there in a cell? A cell is having the cytoplasm. A cell is having the genetic material. It is having the cytoplasm. It is having the genetic material. Take it together. It is making a cell, a living, right? Something which is living. This is what we have. Yes or no? Yes or no? Trust me, we are going to start from the scratch. We are going to start from the zero. Same way, bache, we teach you in your NEET Endgame 2.0 batch as well. If you're still looking for a batch, right, where you can revise your syllabus properly, NEET Endgame 2.0 is for you. So here in the description box, you people will get the link for that video, okay? Okay, for that batch. You have to click on it and you will be the part of that batch, okay? So, listen to me, Amrutha. So what am I saying? I am talking about the protoplasm, and here I am saying that it is the physical basis of life, right? It is having that genetic material, it is having the cytoplasm, and it is something that we need for making a living structure, having a metabolism. Okay, so now when you people talk about this protoplasm, what this protoplasm is containing, right? I hope you know that. Here we also discuss carbohydrates, we discuss proteins, we discuss fats, we discuss nucleic acids, etc, etc. That is what we have here. Yes or no? That is what we have here. Now even if you will pick these terms, carbohydrates, the protein, the fats, the nucleic acid, you will be talking about carbon, hydrogen, oxygen, nitrogen, even sulfur, phosphorus. That's what we discuss. Carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. So ultimately these elements are making these compounds, these monomers, right? These polymers, they are further forming that protoplasm. That protoplasm is basically making something living. That cell, that living cell is further making tissue and all and all and all. This is how everything is related. Try to relate it with evolution. Okay people, try to relate this chapter with evolution. That is what we have studied in evolution. That is how everything is linked, right? Try to recall that. If you are the member of Games of NEET, you know that, right? If you are a regular student of Game of NEET, you know that this is what we have studied in evolution. Yes or no, guys? Show me that energy in the chat section, people. Quick. Show me that energy in the chat section. Have you seen it there? Have you studied it there? Right? Have you studied it there? Guys, energy is very low these days. What happened? What happened? Do not forget, biology is the 50% of the NEET syllabus. Physics and chemistry is important. But if biology is not there in your grip now, then nothing is possible. Nothing is possible. So be consistent, people. Be consistent. Okay, that is what we have. So that's how everything is related, and that is what we have to study in this particular chapter. Right, in this particular chapter. Okay, so biomolecule, when we talk about this word biomolecule, yes, you are right, bio means living, right? The molecules which are making living body, they are the biomolecules. If I talk about the NCERT, so NCERT says all carbon compounds are biomolecules. है ना? This is what our NCERT say. All carbon compounds are biomolecules. This is what our NCERT says. So this is what we have to study, that what are we made up of. So obviously we are not allowed to miss NCERT. So NCERT will go hand in hand. So here you can see the starting of this chapter. So what are they saying? That are all living organisms, are they made up of same chemicals? Are they made up of same elements and compounds? Right, bache? So we have to perform the chemical analysis, elemental analysis here. We have to check that thing, and this is what we have to study. So are you ready, people? Are you ready? So remember, in biomolecules, right, carbon containing compounds are biomolecules. So here we will see that living organisms are having carbon, hydrogen, oxygen and nitrogen. Bache, after that chemical and elemental analysis, now this is what we got to know. This is what we got to know. And same type of analysis was performed on non-living things also, on earth crust also. Right. So I am telling you these things in general, that what has happened, that when we analyze the living organism, we found mainly carbon, hydrogen, oxygen, nitrogen. And same way, when same type of tests, they were performed on the non-living organisms, so we also got carbon, hydrogen, oxygen, nitrogen. So whether it is living or it is non-living, they are made up of what? Everyone. Carbon, hydrogen, oxygen, nitrogen. Carbon, hydrogen, oxygen, nitrogen. So see, if we perform analysis on a plant tissue, on animal tissue, even on microbial paste, whether it's a plant, it's an animal, or even if it is a microorganism that we cannot say with the naked eyes, we will see that they have this list, right? Some other elements are also there. Okay, and if same analysis is performed on the piece of earth crust, right, bache, which is considered as non-living, it is also having the same list. So what are the differences? What are the differences? This is what we need to discuss now. Clear, bache? Clear, bache? So they are saying that no such differences could be made out. All the elements present in the sample of earth crust are also there in the living tissue. Clear? But abundance of carbon and hydrogen with respect to other element is higher in any living organism than in the earth crust. That is your MCQ. right? From this first paragraph, that is our MCQ. What is our MCQ, people? Yes, what is our MCQ here? What are they saying here? That this line is your MCQ. This will be asked in NEET 2024. Mark my words. Mark my words, Lakshmi. This question will be there in your NEET 2024. So what are they saying? That carbon and hydrogen with respect to other elements, they are higher in any living organism than the earth crust. Okay, then the earth crust. So now let's talk about the chemical analysis. So are you people ready for it? Are you people ready for it? Yes, right? So what we need to do? Chemical analysis, right? What will be done, bache? Chemical analysis. This is what we need to discuss. So for that, what do we need? Let's talk about the steps first. Okay, let's talk about the steps first. So how are we going to start, people? How are we going to start? First of all, we need a living tissue. What is required first, bache? A living tissue is required. Okay, so now when it comes to a living tissue, what can you people consider? You can consider anything. Just let's say from an animal we can take the liver, right? We can take plant material also. You can take their leaves, right? You can take their leaves. You can take the liver, right? Liver of an animal. Clear, bache? So we need the living tissue. It can be anything, but what do we need? What do we need? We need the living tissue. Now, bache, you also need mortar and pestle. Okay, we have to grind this living tissue. What we have to do? We have to grind this living tissue. Let's say here I have the living tissue. I'm going to grind it because I want the paste. So what are we going to add here, people? We will be adding one chemical, that is your trichloroacetic acid. Don't worry, everything is given in NCERT. Okay, everything is given in NCERT. So what do we need here, people? What do we need to add here, Dhanush? What do we need to add here? Yes, what do we need to add here? Tell me, what do we need to add here? Trichloroacetic acid. Trichloroacetic acid. So, bache, it's very simple. First of all, focus on acetic acid. What is your acetic acid? It's CH3COOH. What is your acetic acid? It's CH3COOH. Yes, it's CH3COOH. But here we need a trichloro. So, obviously, see. So, this hydrogen will be replaced by chlorine. How that trichloroacetic acid will be formed, right? Acetic acid is your CH3COOH. So instead of hydrogen, if we will add chloride here, right? If we will add chloride here, so this is a trichloroacetic acid. Am I right? It is trichloroacetic acid. Is it correct or not? Tell me, is it correct or not? Jeetu bache, you are saying wrong. What is wrong? Do let me know. Do let me know what is wrong here. What is wrong here? It's your trichloroacetic acid, right? It is your trichloroacetic acid. So this is something that we are going to use for the chemical analysis, and it is your MCQ. Now I have one question for all of you. That why do we need trichloroacetic acid, right? Why do we need trichloroacetic acid? That's the formula. Trichloroacetic acid. Why do we need trichloroacetic acid? Why? Why in this chemical analysis trichloroacetic acid is required? Excellent, Diya. One student is saying to make the slurry. No doubt, bache, you are taking a living tissue, you are adding a chemical, right? You are grinding it, you will get a slurry. But why are we so specific about this chemical? One student has answered it. Excellent, bacha. Excellent, bacha. Right? Because, bache, this trichloroacetic acid is very good for precipitating the living tissue. Okay? It can do what? It can precipitate living tissue. And do you know what can it precipitate in living tissue? It can precipitate proteins specifically. Okay, it can precipitate proteins specifically. That's what you need to keep in your mind. Okay, so that is why we use, because it can precipitate that biomolecules present in the living tissue. So obviously now after all that grinding and stuff, what will we get? Right, what will we get? Firstly, we need living tissue, then we need mortar and pestle. We are going to put our living tissue here, we are going to add the chemical here, then we are going to get a slurry, right? Slurry, right? A kind of paste kind of thing. Now what will you do? You will strain it, right? You will strain it with, you can take cheesecloth. Okay, let's say you have the cheesecloth, you are going to strain it. Imagine just say, let's say here you have the cloth, right? Here you have the cloth. ठीक है बच्चा, you are going to put a paste here, slurry here. You are going to do what? You are going to strain it. Something will come out, something will stay here. That is understandable, of course. That is understandable, है ना? जैसे let's say here you have the cloth, you are going to put the slurry, right? So when you will put, obviously a liquid will get strained here, that you are going to get here, and something will remain in the cloth as well. So the one which will get filtered out here, that is going to be your filtrate. So basically you are getting two composition, and the one which will remain here in the cloth, it will be retentate, right? Retentate means something which is remaining. Okay, then it will be the retentate, something which is remaining, and here what is it? It is the filtrate. So you got what, bache? What did you get here? You get two proportions. Filtrate and the retentate. Now bache, here we have one trick. What do we have? We have one trick here. Right people, what do we have? We have one trick here. The trick is fast. Just say you need to, right? You need to speed up your preparation also, na? है ना? Yes or no? So here. Thank you, bache. Shaurukh. Okay. So fast is the word. Now what's the meaning of fast, Vijay? F stands for filtrate. A stands for acid. S stands for soluble. ठीक है, T stands for trick. Sometimes students they ask me what is the meaning of trick, ma'am? T, ma'am. T means trick. So what's the trick here? The trick is FAST, that is filtrate is acid soluble. Right now you are considering it simple, but actually it is not. At the end you get confused. So we got two fraction, out of which the filtrate is acid soluble one. This question can be asked directly in your final NEET paper. And the retentate is what, people? Retentate is what? Retentate is your acid insoluble one. It is the acid insoluble fraction. Now why, ma'am? Why? Obviously something which is soluble in acid, that will come here, and something which is not acid insoluble. Now we have two fractions. Now you know that these two fractions, they vary in composition as well. Now when you talk about the acid soluble fraction, it contains micromolecules. Okay, and before even micromolecules, let me tell you, what are you going to get here in the acid soluble fraction? Acid soluble fraction is representing, or simply you can write it represents, right? This acid soluble fraction, people, it represents the cytoplasmic composition. It represents what? Cytoplasmic composition. The cell is having a cytoplasm, na? So it will represent cytoplasmic composition, but without organelles, right? Basically in acid soluble fraction, whatever you are getting, it is showing us the, roughly it is telling us that this is something which is present in cytoplasm, but not the organelles. Okay, not the organelles, not the content of organelles. And when you talk about the acid insoluble fraction, bache, it represents cytoplasmic and organelle composition. Got it? Yes, people. Is it clear? That's something you need to keep in your mind. Is it clear? And moreover, please write it down that acid soluble fraction is representing biomicromolecule. These are the two words that you got. And acid insoluble fraction is representing biomacromolecule, right? It is representing biomacromolecule. Exception is there, we will discuss about it also. Okay, you can put a star mark here, we will discuss about it. Understood? Understood? So that is something you people should know. Clear bache? That is something you people should know. Okay, so what we got? Yes, what we got? Two things. One is acid soluble fraction, and another is acid insoluble fraction. Acid insoluble fraction. Clear? Clear, people? So acid soluble, again revise it with me, same point, roughly represents the cytoplasmic composition. Okay, roughly represents the cytoplasmic composition without, yes, without organelle composition. Okay, without organelle composition. Understood? Yes, people, understood? Right? So acid insoluble, they represent macromolecules of, the word is macromolecules of cytoplasmic and organelles composition. Yeah, macromolecules of cytoplasm and organelles. That's what you can write down. Understood? That's what you can write down. Clear, bache? Clear, clear, clear? Yes? Is it clear? Acid insoluble. Understood? And moreover, acid soluble, it contains, right? This acid soluble, this is what you should mark. In NCERT, they have simply mentioned now all carbon compounds are biomolecules. This is given in NCERT. Miss Sigma, we will start human physiology as well. But as of now, whatever we have started, let's complete that first, okay? Okay, let's complete that first. Fine. So what is it? No doubt in NCERT, this is what they have mentioned. Right, this is what they have mentioned. They have mentioned it कि all carbon compounds are biomolecules. But in bache living body, these are not just the carbon compounds. We have inorganic compounds also. We have sulfates also. We have phosphates also. They are also making a living body. So that is something you should know. Okay, that is something you should know. So when we talk about the acid soluble fraction, people, it contains inorganic compounds. It contains inorganic compounds, okay, like sulfates, phosphates, etc. Understood? Like sulfates, phosphates, etc. Doubt, people? Yes? Any doubt? Any doubt? So basically inorganic compounds are there, organic compounds are also there. Okay, inorganic compounds are there and organic compounds are also there. Understood? Understood? Any doubt? Yes? Any doubt here? So acid soluble, insoluble, clear? So now the word that I have used is micro or the macromolecule. So this is something that we need to know now. Okay? So bache, what we discussed just biomolecules. Two things are there. So one is biomicromolecule and one is biomacromolecule, isn't it? That's what we have studied, right? That is what we need to know next. So the word is micro. Micro means small, macro means large. How many of you know this? Hanji, how many of you know this? Micro means small, macro means large. Small, large, but what? Obviously, it is something related to the molecular weight. So, biomicromolecules, they are the, right, they are the compounds having low compounds having low molecular weight. They are the compounds having low molecular weight, right? Low molecular weight means it is less than 1000 Dalton. Okay, Dalton is the unit, bache. So biomicromolecules are the one which are having the molecular weight less than 10,000 Dalton. And on the another hand, when it is the biomacromolecule, they are having the molecular weight, yes people, they are having the molecular weight above 1000 Dalton. Right, above 1000 Dalton. Like in some books it is given above 10,000 Dalton as well, right? So obviously here you are going to consider what? You people are going to consider, here you are going to consider polymers, right? What are we going to consider here? Polymers. And here what will be considered? We are going to consider the monomers here, right? Dimers here, like this, like this. Okay, I'll give you the example as well. Okay, I'll give you the example as well, then you will understand it in a better way, and I'll also tell you what kind of question can come from this part. Jaise bacha log, I assume that this much you know. Even if you don't know, there is nothing to worry about. Today we will clear each and every concept. Are you ready with me? Are you ready with me to clear each and every concept of yours? Yes, कि हाँ जी ma'am, today we are not going to miss anything. ठीक है, we have that open mindset, we know we have to complete it, and we will. And we will. Pakka, sure? 110? Okay, fair enough. So when you talk about, in general I'm giving you this example because question can come from this part. So when you people talk about carbohydrates, okay, so you know that, you know about the glycogen, right? You know about the glycogen. Glycogen is a stored food. Okay, in humans, in our case it's a stored food. Okay, or you know that we also call it as animal starch. What we call it, people? We also call it as animal starch. But it is a polymer. Polymer means it is made up of many subunits. Okay, okay, it is a polymer, it is made up of many subunits. Many subunit means, ma'am, what are you talking about? Guys, you know, you have heard about glycogen. Even you people know about the glucose as well. Don't you know? Don't you know? Don't you know about the glucose, the main respiratory substrate? Yes ma'am, we know about the respiratory substrate. Hana, we know about this respiratory substrate. So glucose is a monomer. It's a single unit. Understood? It's a single unit, right? Just you know that carbohydrates, especially we call them as what? Monosaccharides. We call them as what? Monosaccharide. Mono means single. Single. Saccharide means sugar. So glucose is a monosaccharide, it's a single unit. So many glucose, so many glucose, so many glucose, when they join, when they polymerize, okay, then they are going to form what? Polysaccharides. Then they are going to form what? Polysaccharides. I'm just giving you one example. Okay, they are going to make polysaccharide. So of course this polysaccharide, it is a polymer, it is made up of many subunits. I'm telling you, we will discuss it in detail. As of now, I'm trying to convey something else. Okay, so this polymer, this polysaccharide is what? It will be there in acid insoluble fraction. Okay, it will be there in acid insoluble fraction. So it means it's a biomacromolecule. Got my point? It's a biomacromolecule. Got my point? Yes. And now when you are talking about this monomer, that is your glucose, so it is a biomicromolecule. So it will be present in, it will be there in acid soluble fraction. So such type of questions can come in your paper. Okay, such type of questions can come in your paper. One another example I'll give you here. Okay, that example is proteins. Proteins are what? They are the polymers. They are the polymers. Polymers of amino acids. So that amino acids, what are they? Right, they are the monomers again. Okay, so amino acids will be in, so when you talk about the amino acids, they will be in acid soluble fraction. It will be in acid soluble fraction. And when you people talk about the proteins, where are you going to get them? In acid insoluble fraction. Where are you going to get it, people? In acid insoluble fraction. So from this part, that type of questions can come in your final NEET examination. Understood? Understood? That is what you should know. Okay, Priya? That is what you should know. Okay? Chalo, I'll ask you one more question. Let's say you have out of this, right? That's your question. Out of this, which one will be there in the acid soluble fraction and which one will be there in the acid insoluble fraction? Answer it. Which one will be there in the acid soluble fraction and which one will be there in the acid insoluble fraction? Answer it to me. Yes, answer it to me. Answer it to me. Quick. Bache, that type of questions can definitely come in your NEET examination. I am not asking Chetu which one is soluble, which one is insoluble. Acha, okay, you are telling me acid soluble or acid insoluble like this. Okay, fine, very good. So answer it to me. Answer it to me. Like in one message you can type insoluble this. In acid insoluble fraction this. In acid soluble fraction this. Answer it to me. Such questions can come in your NEET examination. Chalo, let me answer this, bache. Obviously, first of all, you should know their nature. If you are talking about the collagen is a protein. Collagen is a protein. It means it's a polymer. So it should be in acid insoluble fraction. It should be in your acid insoluble fraction. Clear, bache? Your chitin is what? It's a homopolymer. It's a homopolysaccharide. So again, it is also a homopolysaccharide. So it should be in same acid insoluble fraction. Okay. Then elastin is also a protein. It should also be in acid insoluble fraction. Am I right? Heparin is also a polysaccharide. Hai na, bache? It should also be there in there. Acid insoluble fraction. Take it, right? So now when you talk about the acid soluble fraction, the sucrose is a disaccharide. It will be in soluble purine, right? Like your adenine, guanine. So they are what? Their molecular weight is less, right? Their molecular weight is less. Adenosine, it's a nucleoside. It's a nucleoside, so it will also be in acid soluble fraction, right? N-acetyl galactosamine or glucosamine, it should also be in acid soluble fraction because their molecular weight is less. So that is what you need to know. Now, bache, here comes one question. What about cholesterol, ma'am? What about cholesterol? Cholesterol is a lipid. It's a derived lipid. Am I right? It's a derived lipid. Now, bache, here, this is something that you need to remember. As I said, acid soluble fraction. Acid soluble fraction. So, acid soluble fraction means the filtrate one, means the biomicromolecule one. Acid insoluble fraction means the biomacromolecule one. So, here I mentioned one exception. What is that exception? Now, let me tell you that exception, bache. In the acid insoluble fraction, we also get lipids. Okay. Lipids are also present here. Okay, lipids are also present here. But lipid is what? It's a biomicromolecule. It is not a biomacromolecule. It is a biomicromolecule. But still, it is the part of acid insoluble fraction. Still, it is the part of acid insoluble fraction. Is that clear, Arjun bache? And now why? Why is it so? Why is it so? Let me explain you this. Right? So, bacha, when you talk about the lipids, when you talk about the lipids, their molecular weight, okay, their molecular weight is less than what? Is less than 1000 Dalton. It is even less than 800 Dalton, imagine. Okay, but still present in Yes, still present in acid insoluble fraction. So the reason is very simple. You know that when you talk about the lipids, they are what? They are water insoluble. Yes, people, these lipids, when you people know about these lipids, what are they? They are water insoluble. Array. Yes, they are water insoluble. Right? And even you know that they are making the plasma membrane. Now we have in a living tissue, we are having the cells. So when we are trying to break it, right, when we are trying to grind that living tissue, we are getting that cells, people, we are getting that cell, right? We are getting that cells. So you know that cells membrane, they are also the lipids, they are also the phospholipids, and these lipids being insoluble in water, they are going to form some insoluble vesicles. What are they going to form? They are going to form that some insoluble vesicles, and that vesicles will not pass in that acid soluble fraction, that will be there in that acid insoluble fraction, and that's why we get lipids in acid insoluble fraction. So it is a previous year question. So tell me, how many of you are getting it? How many of you are loving this chapter? How many of you are enjoying this chapter? Trust, tell me quick. Guys, chat, let's chit chat, tell me. Chitin, because chitin is a what? Chitin is made up of N-acetyl glucosamine. Chitin is a polysaccharide, right? And NAG being the monomer. Wow, one student is asking, how do we know that they are soluble or insoluble? But I just gave you the condition. Acid insoluble fraction will get biomacromolecules, the polymers one, the one having the molecular weight above 1000 Dalton. And when you people talk about the soluble fraction, they will have the biomicromolecule. So whatever is a micromolecule, that will be in acid soluble fraction. Whatever is the macromolecule, that will be in acid insoluble fraction. That is how you are going to get it. Yes, bache, blues. So guys, up to this part all clear? Up to this part all clear? So can we just revise it from NCERT? Can we just revise it from NCERT? Can we just revise it from NCERT? Okay, but before revising it from NCERT, let's talk about the elemental analysis also, right? We have just discussed the chemical analysis. Okay, now we have to discuss the elemental analysis. What we need to discuss? Elemental analysis. Okay, so here also again you have to consider a living tissue. What we need to take? What we need to take first? Yes, people, what we need to take first? We need to take the living tissue. Yes, we need to take the living tissue. So, bache, in the living tissue, what will be there? We are going to take the living tissue. So, we will take the living tissue, we will take its weight. That will be its wet weight. Why? Why? Yes, anyone? You are going to take the living tissue, like let's say you have taken the liver of the animal, so you are going to take its weight. So that will be the wet weight. Why? Why? Because it is having the water in it. It is having the water in it. And if you relate it with the ecology, so you all know now that wet weight, when we even check the biomass, right, we do not consider the wet weight. We do not consider the wet weight. Okay, so it is the fresh living tissue that we have taken. It is having the wet, we will measure its weight. It will be its, right, we will measure its weight, and that will be its wet weight. And then what will we do? We will try to evaporate. Try to evaporate what, people? Yes, bache. Try to evaporate what? Tell me. Tell me. We have to evaporate the water. So we will take the living tissue. We will dry it. Okay, we will dry it. Why? So that water evaporates. So that water evaporates. Then after that, what are we going to get? We are going to get the dry weight. Okay, we are going to get the dry weight. This is what we need to measure. So firstly, wet weight, then we will get its dry weight. Clear, bache? We will get its dry weight. Yes or no? Yes or no? Yes or no? So how are you going to do it? How are you going to do it? Yes, obviously water will evaporate, na? So when no water is there, the remaining one will be the dry weight when you are going to measure. And when the tissue is fully burned, the carbon compounds, they will oxidize, they will form the, right, they are going to oxidize to form gases. Right? Yes or no? Yes or no? So first of all, you have taken the living tissue, it is having the water, so the weight that you are going to measure will be your wet weight. The secondly, we are going to dry it. Why are we going to dry it? So that water can evaporate. So after that you will get the dry weight. Now you have to burn that tissue. Okay? You have to burn that tissue. So when we burn that tissue, what is going to happen? Right? All carbon compounds, right, that carbon compounds, they will be released. Okay, they'll be released like in CO2 form. It will form gas, isn't it? Right, the carbon compounds, they will be oxidized now. It is going to form what? It is going to form gas. It will form CO2. The water vapor will also be removed. Yes or no? So you are burning a tissue. So burn tissue, carbon compounds, they'll be oxidized. I'm writing it in short form. They'll be oxidized to form gases like CO2. Water is also removed. So what is that remaining part? It is just the ash, right? Ash. That is just the ash. What is it? It is ash, isn't it? What is it? It is ash, and this ash is nothing but it is representing the inorganic elements like the calcium, like the magnesium. So this is the elemental analysis. The one which we performed earlier, it was the chemical analysis. So ash, it represents inorganic elements. Okay, that's how you can make your short notes as well. So it represents inorganic elements like calcium, etc. Understood? Understood? Yes. So let's revise this part from the NCERT and with the help of flowcharts as well. So it is saying, take the living tissue, grind it in trichloroacetic acid, you will obtain a thick slurry, strain the slurry through cheesecloth, you'll get two fraction. One is the filtrate, which is acid soluble, which includes biomicromolecules, and another is bache retentate, acid insoluble, which includes your biomacromolecule. Understood? Biomacromolecule. Understood? Understood? So do not forget this part, right bache? Do not forget this part. And this is the analysis of inorganic compound. For your quick revision, take a dry piece of living tissue, take a dry piece of living tissue, take a burn the tissue, but firstly before that take a fresh living tissue, measure its wet weight, then let it evaporate, measure the dry weight, then dry piece of living tissue, you have to burn the tissue, ash is left behind. So because all the carbon compounds, they are turned into the gaseous form like your CO2, water will evaporate. So this ash will have calcium, it will have magnesium, sulfate, phosphates, etc. Right? Not just calcium, basically inorganic elements are there, inorganic elements are there, right bache? So calcium, magnesium, sulfate, phosphate, that will be present here, and that is what written here in this NCERT. Okay, let me highlight few words, right? You will quickly recall it. Living tissue, okay, this trichloroacetic acid, right? We'll get the slurry after that. Okay, so strain that slurry through a cheesecloth, and then we will get the fractions. One is filtrate, right? More technically, the acid soluble pool, and the second one is your retentate, acid insoluble pool. So this is written here. Understood? Yes, Arjun Reddy, we have already covered protein biosynthesis and molecular basis of inheritance. And people, there also I mentioned the protein biosynthesis is nothing. This is just the transcription translation altogether. That's all. Okay. So now here in this slide, see, in higher classes you will learn about how to analyze a living tissue sample and identify a particular organic compound. Like further we can also study from that, right, the individual organic compounds as well, bache. Right. So we have to use different separation techniques. We can separate them as well. Okay, so we can basically isolate that organic compound, can purify, right? We can obtain that purified compound as well. Fine. And after that studies, we got to know about their molecular formula and probable structure of the compound as well, right? After further study, this is what we have studied. Okay, this is what we have studied. Clear, bache? So see, here you have that line, you should highlight it, that all the carbon compounds that we get from the living tissue can be called biomolecules. However, living organism, they have also got inorganic elements also and compounds in them. So remember this part. Fine, remember this part. Okay, Sunanda, is that clear? Yes, but bache, and see here also a slightly different but destructive experiment is also there, right, where we have to weigh a small amount of living tissue. We will get its wet weight and then we will dry it, so all the water evaporates and the remaining material gives the dry weight, and when the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form and remaining is called the ash. Right, remaining is called the ash, and this ash represents inorganic elements. So any doubt here? Any doubt here? And people already in the starting, that's why I'm saying the notes that I write, right, never miss that notes. No doubt we are going to share the PDF with you all. Okay, it's not like I will not provide you the PDF, I will provide you the PDF, but still whenever I write down something na, please take the notes. Okay, so see, in the starting itself I mentioned na, inorganic compound, sulfate, phosphate, they are also seen in acid soluble fraction. So sometimes you miss such statements, so that is a MCQ. Okay, so elemental analysis will tell you elemental composition in the form of hydrogen, oxygen, chlorine, carbon, while analysis for compound will tell you about the organic and inorganic constituents, right, bache? So that's what you have to understand. Right, that's what you have to understand. Fine. Yes, any doubt? Now come to these table. In the chapter biomolecule, you are not even allowed to miss the tables. Okay, you are not even allowed to miss the tables, bache. See, look at this table. This is a comparison. Hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, silicon. They have given a list. One question was there from this part. So all of you, please mark, right? Please mark it. Okay, please mark it. So see, this is the list of the element, their percentage in earth crust, their percentage in human body. So I told you before, look at the percentage of oxygen. It's more in human body, right? Look at the percentage of carbon. This is what we just discussed. It is more in human body. So you should know if you are targeting 360 out of 360 marks in biology, then you are not allowed to miss such tables as well. So as per that table, if I talk about the non-living organisms, right? If I talk about the non-living organisms, so they have maximum is their oxygen, they have oxygen, right? If I'm not just considering carbon, hydrogen, oxygen, and even if I have to consider carbon, hydrogen, oxygen and nitrogen, so maximum is what? Maximum is your oxygen. Then after oxygen, what do you have? The hydrogen is there, right? Then after hydrogen, what do we have? Carbon is there, and then very little nitrogen is there. That will be the sequence. Yes, that will be the sequence. Yes, right, that will be the sequence. So if I talk about the living tissue, what do they have? They have maximum oxygen. After oxygen, they have maximum carbon. After carbon, they have nitrogen, and then they have hydrogen. Am I right? So this is what I got from this table. So please do not forget this. Okay, now let's focus on the silicon. This is my favorite question. So silicon is still there in earth crust, right? But it is negligible in the case of living tissue. My important MCQ. This is my another important MCQ. Please take a screenshot of it. Okay, please take a screenshot of it. Compare the sodium and calcium also. Sodium is 2.8 in earth crust but just 0.2 in human body. Calcium is 3.6 in earth crust but just 1.5 in human body, right? Nitrogen is very little in earth crust but it is 3.3 in human body. So this point, right, this point and this silicon point is very important. Okay, it's very important. Talat bache, if you do not have any information about the NEET and about the things that you need, the documents that you need in NEET, there are many videos that you can search. Okay, please do not waste the time here. I will make a separate video later, but right now let us focus. Okay, let us focus. So guys, in this lecture it is not just the my content, right? Along with that we will cover the NCERT. So basically lecture is as per NCERT, it's just that I will follow a different sequence, right? We will not just translate each and every line of NCERT. That's what you need to keep in your mind. So this information done. So after it, in your NCERT, what is given? In your NCERT, amino acids are given. So now, bache, we will not pick up amino acids directly. What are we going to do now? We are going to start the story with the carbohydrates. What we need to discuss now? We need to discuss about the carbohydrates. So tell me, what do you know about the carbohydrates? Tell me, what do you know about the carbohydrates? This is what we need to start now. Carbohydrates. Yes, carbohydrates. Tell me quickly. Hanji, tell me quickly. Okay, whatever you know about the carbohydrates, tell me. And trust me, my lecture will be more than sufficient for this particular chapter. Okay, tell me whatever you know about carbohydrates, just type it, then I'll see this and I'll answer it. Tell me, what do you know about the carbohydrates? Monosaccharide, sugar, micromolecule, saccharide, anything else? Anything else? First of all, they are the organic compounds. Hanji, yes. First of all, they are what? They are the organic compounds, right? Carbohydrates, they are the organic compounds because they contain carbon, hydrogen and oxygen. This is what we study in the case of carbohydrates. Yes or no? Yes bache Akshita, yes bache Talat, yes bache Sudarshan. What do we have here? Carbon, hydrogen, oxygen. No doubt. And some of the carbohydrates, they are also having the nitrogen. Can you give me any example of the carbohydrate having nitrogen? I'll give you one chocolate if you will give me the correct answer. Some of them are having nitrogen and some of them are even having the sulfur. If you will give this example to me now, I'll give you a chocolate. Okay? You can type your answer later in the comment section. I don't mind. Okay? I don't mind. Although it's very simple. Okay? So carbohydrates, they are the organic compounds which are having carbon, hydrogen and oxygen, and some of the carbohydrates are having nitrogen and sulfur as well. Okay? Nitrogen and sulfur as well. Okay? Now what's my point here? What you need to know? Carbohydrate, when you break this word, what it means? They are the hydrates of carbon. This is what we say. What are they? They are the hydrates of carbon. Now what is the meaning of being the hydrate of carbon? Tell me. What is the meaning of being the hydrates of carbon? Then I will tell you its proper statement as well. Right? Let me finish the carbohydrate part. Then we will talk about primary and secondary metabolite. As of now, you answer this. Right? As of now, you answer this. And yes, before starting it, how can I forget? Sorry, students. How can I forget? Please come to this table as well. Yes. Please come to this table as well. So here in this table, they have also given sodium, potassium, calcium, magnesium, water and compound. So they are telling these are the inorganic constituents of living tissue, right? So in general, we know sodium, potassium, calcium, magnesium, even the water, but do not forget this. Phosphate, sulfates, they have also mentioned it, right, in the table itself. They are just the sodium chloride, calcium carbonate, right, the phosphate and the sulfate. So I, in the starting itself, in the notes, right, in the points, I have mentioned it, but still you please highlight this in your NCERT. MCQ can come from this part as well, okay? MCQ can come from this part as well. So now come back to this. So we are saying carbohydrates are the hydrates of carbon. What is the meaning of hydrates of carbon? Now here you see carbon is there, hydrogen is there, oxygen is there. But if you talk about the part of carbon in a carbohydrate, it is 1, hydrogen is 2, oxygen is 1. Like in a carbohydrate, carbon, hydrogen, oxygen, they are present in the ratio 1:2, 2:1, right? In which ratio they are present? They are present in the ratio 1:2, 2:1. They are present in the ratio 1:2, 2:1. Clear, bache? So when you look at this hydrogen oxygen ka ratio, hydrogen is 2, oxygen is oxygen is 1. Just in the case of water mein you have 2 hydrogen, 1 oxygen, hai na? So same way in the carbohydrates also you have 2 hydrogen, you have 1 oxygen, just like water. That's why we say it's the hydrate of carbon. That's why we say it's the hydrate of carbon. So from this general thing, this is what you people can conclude, that carbon and oxygen, it is in same ratio when you talk about the carbohydrates. Is that clear? So these basic things are important to understand this chapter. I know it is not given in NCERT, but if you don't know the basics, then there will be a problem. Understood? Right? Any doubt here, people? Any doubt here? So hydrogen and oxygen, it is in ratio 2:1. That's why we say they are the hydrates of the carbon. Hydrates of the carbon. Understood? Understood? So chemically, what are these carbohydrates? Chemically, if I have to define it, chemically these are polyhydroxy, right? Polyhydroxy aldehyde, or they are polyhydroxyaldehyde or polyhydroxy ketones. What are they? Yes, people, what are they chemically? They are polyhydroxyaldehyde or polyhydroxy ketone, or the compounds which on hydrolysis yield such compounds. Right? Right? So chemically, what are these carbohydrates? They are polyhydroxyaldehyde, polyhydroxy ketone, and moreover, bache, the compounds which on hydrolysis yield such things na, like polyhydroxy ketone and aldehyde, they are known as carbohydrate. They are known as carbohydrate. Clear, bache? So now what is the word here? The word is polyhydroxy. Polyhydroxy. Bache, it's very simple. There is nothing to cram. Poly means many, hydroxy means OH. Okay, OH. So polyhydroxyaldehyde, guys, these are the functional groups, right? In class 10th itself we have studied that these are the functional groups. Aldehyde ketones, when they are joined with one compound, they give it a special properties. I really don't think that I need to discuss that. I really don't think we need to discuss that. So polyhydroxyaldehyde, aldehyde, the functional group CHO. ठीक है, polyhydroxy ketone. What are ketones? Ketones are your C double bond. Ketones are your C double bond O. Isn't it? Isn't it? So they are polyhydroxyaldehyde or the polyhydroxy ketone. So chemically, that is how you define the carbohydrates. Okay, that is how you define the carbohydrates. Am I right, bache? Am I right, bache? So the simple carbohydrates, right? Your monosaccharides, they are very simple. They are soluble in water. They are sweet. Right, your monosaccharides, they are soluble in water. They are sweet. They are very simple carbohydrates. They are known as sugar. Okay, I'm mentioning, bache, Dipanvita, I'm mentioning something very important. All the carbohydrates are not sugar, right? You cannot say that all the carbohydrates are sugar. No, they are not. Right, these simple carbohydrates like your monosaccharides, okay, bache, which are soluble in water, they are sweet in taste. For them you use the word sugar. Right, your starch is not sweet in taste. Got it? Got it? So this is something you people should know. So any doubt here? Any doubt here? Yes. Any doubt here? So you define the so basically when we will study the carbohydrates, we will discuss the functional group aldehyde, we will also discuss the functional group ketone. So I don't think that there should be any doubt here. If still there is, please let me know. If still there is, please let me know. Okay, chalo. So we categorize these carbohydrates into three groups. We categorize these carbohydrates into three groups. Again, important. Not given in NCERT people, but it is important. So saccharide word. So one is monosaccharide, another is oligosaccharide, and then comes the polysaccharide. Okay, so that's how we categorize them: the monosaccharide, the oligosaccharide, and the polysaccharide. Okay, and the polysaccharide. Clear? Clear, clear, clear? Clear? Yes? Yes or no? Yes or no? Monosaccharide, oligosaccharide, and the polysaccharide. These are the words that we have. देखो बच्चे, when in general you discuss the carbohydrates ना, you know that it is the main respiratory substrate, है ना, especially the glucose. Okay, it is the main respiratory substrate. Now I am going to explain you something. Okay, so when right, that is your glucose. Glucose is a simple sugar. Okay, when you talk about these carbohydrates, they play different role. You will see some carbohydrate, they are helping in the formation of a structure. I'll give you one example, cellulose. I'll give you another example, chitin. They are making the cell walls. Okay, they are helping in the, they are kind of structural polymers, isn't it? Because they are helping in the formation of that structure. Some are helping in storage, your starch, glycogen. There are different ways, right? In the same way, we have the simple carbohydrates like your glucose. These are the respiratory substrate. They will get oxidized, they will release the energy. So everything we have to discuss here. So although this part is not written in NCERT, but somewhere it is from NCERT, and to understand this topic, we have to go as per that. So mono, oligo, poly. Mono, oligo, poly. Saccharide here means sugar, right? Please do not cram, please understand this topic, right? Read this chapter with an open mind. Ki yaar, today I am going to master this chapter. That's how you have to study. Okay, so who's going to stay with me till the end? Who's going to stay with me till the end? Because today we have to finish it at any cost. Who's going to stay with me till the end? And guys, please like the session, share the session link with your friends also. I know we feel bad, है ना? When we are, I know some students ना, they don't want to share good information with their friends. They don't want to share good content with their friends. It happens ना. Some students, they do things like this, है ना? कि यारे नहीं यार ma'am, we don't want to increase the competition. Isn't it? We don't want to increase the competition. We will not share your videos with others. We don't want them to know that there is a very good channel, there is a very good biology teacher, and a very lovely and sweet biology teacher as well. But that should not be your attitude, right? It's okay, it's fine. Share good knowledge, share good things with others. You will get something in return. Trust me on that. Okay, trust me on that. One student is saying session duration. Yeah, I am talking to you people, I am discussing good things with you. You are talking about session duration. Obviously it will go for 6 hours, 7 hours, it depends. It depends upon you, it depends upon me. So I know as a student that's a general mentality. Not as a student, even professionally people do such things. Yeah, I'm not going to share good information with other person. No. They'll grow more in their lives than us. Nah, that should not be your attitude. Trust me, right? You are going to get what you are working for. If you are giving your best, if you are working very hard, if you are unconditionally doing your, like putting efforts in your work, obviously, but in proper direction, you will get the result for sure. It doesn't matter. Even if they will attend my classes, na, maybe they will not solve the question. Maybe they will not come to our special class. Okay? Hai na? They will not come to our special class. They will not practice the question. So anything can happen. So people chill. Fine, chill. Do share our session link with others and ask them to subscribe to our channel. Make it 100k quickly. Make it 100k quickly. Sure. After it, we'll start human physiology. After it, we'll start structural organization. We'll finish it. We'll go for the human physiology. Why are you people getting worried? I love to teach human physiology. You are going to love that chapter. So chill. So come here. So saccharide means sugar. So mono means single. Saccharide means sugar. So these are the simple sugars. Okay, these are the simple sugars. They cannot further be hydrolyzed. They cannot give us sugars on further hydrolysis. I'll give you one example. We all know about the glycogen. We all know that this glycogen is stored in our liver. In our liver it is stored. We know that we also call it as animal starch. What we call it? We also call it as animal starch. So this is something that I'm going to tell you initially, that it is a polysaccharide. Okay, it is a polysaccharide. Poly means many. Saccharide means sugar. So poly means many, saccharide means sugar, means it is made up of many sugars. It's a polymer. It should be in acid, insoluble fraction. So if I will hydrolyze this glycogen, if I will break it, obviously I will get simplest forms. I will get glucose, because it is a polymer of glucose. It is made up of so many glucose molecules. So I'll break it, I'll hydrolyze it, I'll get glucose. Okay? So glucose is what? Glucose is my monosaccharide. Okay, glucose is what? Glucose is my monosaccharide. I cannot hydrolyze this glucose further to a sugar. I cannot break this glucose further to give me a sugar. This glucose can just give me carbon, hydrogen, but it is not going to give me another sugar, right? So it is what? It is a monosaccharide, and that's what I am discussing here. Mono means single, saccharide means sugar. They are the simplest sugars, bache. They are the simplest sugar. They cannot be hydrolyzed further into simpler carbohydrates. Okay, they cannot be further hydrolyzed into simple carbohydrates. That's what you need to keep in your mind. Okay, so it's just a monomer. It's just a monomer. We will discuss about the monosaccharides individually in detail. Now oligosaccharides, but they will contain the sugars, right? What will be the number here? Can you tell me the number? 2 to 7. What will they contain? They are formed by the condensation of 2 to 10, not 7. They are formed by the condensation of 2 to 10 monosaccharides. So basically, if 2 monosaccharides will join, if 3 monosaccharides will join, 4 monosaccharides will join, so up to 10 it will come under the oligosaccharides. Then comes polysaccharides. Okay, then comes polysaccharides. So obviously here, right, it should have the sugars more than, it should have the monosaccharides more than 10. Okay, right? So proper polymerization is there. So that is how we are going to categorize them. So firstly we need to start what? We need to start with the monosaccharide. What we need to start first, people? We need to start the monosaccharide. So mono means single, saccharide means sugar. As I said, simplest sugar which cannot be further hydrolyzed. Right? Simplest sugar, sugars that cannot be further hydrolyzed. Understood? Yes, understood. So now when you talk about these monosaccharides, when you talk about these monosaccharides, they are having a number of carbon. They are having certain number of carbons. Yes or no? Yes or no? First of all, I'll start with their general formula. When you talk about the monosaccharides, they are having a general formula. Like see, in class 10th also, we discuss about alkanes, alkynes. Right? Alkane, alkene, alkyne. Here also, right, that alkane, alkene, alkyne are also having the general formula. Here also they are having general formula, and it is CnH2nOn. Or you can also write it like this, CnH2O. This is how you can write it. This is a general formula of the monosaccharide. What is it? It is the general formula or the empirical formula of the monosaccharide. Understood? Understood, bache? So when you talk about the carbon number here, carbon number here in these monosaccharide, it can be from 3 to 7. Okay, it can be from 3 to 7. This is the confusing thing for many kids, right? Students, they confuse it ki ma'am, here you are saying ki oligosaccharides are having 2 to 10 monosaccharide, right? And here you are also saying 3 to 7. What is this? But here that 3 to 7 that I am discussing now, it is the carbon number. It is the carbon number. I'll give you one example. Just let's say this is a circle having a diameter, this is another circle having a diameter, this is another circle having a diameter. So their diameter is varying. Their diameter is varying. These are the circles. Okay? These are the circles, their diameters are varying. It is like this. Okay? So they come under the monomer. Single circles they are. It is having different diameter, it is having different, it is having different. Now let's say this is something you are discussing now. They are also made up of circle. Here you are talking about a single circle. Here you are talking about many circles. Just say 4 circles are joined together. 4 circles are joined together. Their diameter also vary. Okay? Or they can be same. So here number of circles you are talking. Here you are talking about a single circle, but you are talking about its dimension. Ki whether the diameter is more or less. It's like that. Just say for an example, in the case of monosaccharides, people, as I said, carbon number varies from 3 to 7. So simplest or the smallest monosaccharide will have C3. That's how you can check it now. N is equals to 3. This N is telling us what? This N is telling us about the number, right? This N is telling us about that number of carbon, number of oxygen. This is what this is what we can depict now. This is what we can depict. So as I said, in the monosaccharides, it is the carbon number that vary. It can be from, right, it can be from 3 to 7. 3 to 7. So I'll pick up the simplest thing that you all know. Glucose. You all know about the glucose. You all know that its formula is C6H12O6. It's C6H12O6. It's C6H12O6. So C6, N is equals to 6 here. Okay? Let's put it in this formula. This formula is saying na CN, H2N, ON. So C6, H will be 2 into 6, O6. So obviously that's how you are getting the formula. That's how you are getting the formula. So what is the meaning here? Here the meaning is that glucose is having 6 carbons. 6 carbon. Right? Ultimately glucose is a single subunit. It's a monomer. It's a monomer. It's a single subunit. But how many carbons are there? 6 carbons are there. Okay? So same way we have, right? So that is how we categorize these monosaccharides. Fine. That is how we categorize these monosaccharides. We have triose, we have tetroses, we have pentoses, we have hexoses and heptose. That's how you are going to categorize them on the basis of their number. Number of what? Number of carbon. On the basis of number of carbon. So here N is equals to 3, so it will be a triose. Here N is equals to 4, tetrose. N is equals to 5, pentose. N is equals to 6, hexose. N is equals to 7, heptose. That's how we divide. Understood? That's how we divide. Understood? Understood? Okay. So write down few properties of the monosaccharides, that they are highly soluble in water. Right? This is what we are discussing now. Yes, they are highly soluble in water. They are sweet in taste. They are highly soluble in water. They are sweet in taste, and they are of reducing nature. Okay, they are of reducing nature. Everyone just put a star mark over this point. We will discuss it. Right, let me start the oligosaccharides. We will discuss it. The reducing nature. They are having what? Reducing nature. This is something very important. Right, they are sweet in taste. This is something very important. Understood? Understood? So you have trioses, tetroses, pentoses, hexoses, heptoses. Okay. Now bache, when I was defining the carbohydrates to you, right, trust me, this carbohydrate part is lengthy. Once we will finish it, we'll immediately start amino acid, we'll immediately finish amino and protein, then lipids, then like this. Okay, then like this. Okay. So now I told you chemically they are aldehydes and they are the ketones. That's what I just said. Chemically, okay, just a minute. This is what I just said, students. Yep, this is what I just said, that they have 3 carbons, 4 carbon, 5 carbon, 6 carbon, 7 carbon. That's what I just said, right? So it can have aldehyde group, it can have ketone as, it can have aldehyde as well as ketone. Aldehyde as well as ketone. I'll give you one example. So when you talk about the simplest carbohydrate, especially aldose, it's your glyceraldehyde. What is it? It is your glyceraldehyde. Just here you talk about the phosphorylation as well. What do you discuss, people? You also talk about the phosphorylation, if you remember. Yes, if you remember. Do you remember that phosphorylation part? Phosphorylation part. Do you remember glyceraldehyde 3-phosphate? I think you have heard this word. You have heard this word, glyceraldehyde 3-phosphate. In the glycolysis we have discussed that. Okay, in the glycolysis we have discussed that. So glyceraldehyde is the example, and here it is dihydroxyacetone. In some books it is also written glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate. That is actually the first step of oxidation, phosphorylation. So don't worry about that. Right, Nito, don't worry about that. So glyceraldehyde is your this smallest this monosaccharide. So it's a triose. Triose means N is equals to 3, 1, 2 and 3, and it's polyhydroxyaldehyde and polyhydroxyketone. Polyhydroxyaldehyde and polyhydroxyketone. So ultimately we know that carbon is tetravalent. Okay? So how can you make the structure CH2CH2OH? CH2CH2OH. I think this part is clear. Just simply put the formula na. Put the formula. There is nothing to worry about. CnH2nOn. So C3H will be, yes people, H will be 6O3. That is what you are going to get. That is what you are going to get. So for sure here we have one OH. Here we have one OH. Our 3O. It's fulfilled. So 3, 4, 5. Okay. So it should be yes. What's the mistake? Tell me. What's the mistake here? Is there any mistake? Is there any mistake? Is there any mistake? Guys, is there any mistake? You study chemistry. I want you people to answer my question. We are talking about the glyceraldehyde. I told you it is going to have how many carbons? 3. Tell me what should be there. Very good, Chitra. Excellent Chitra, very good. So Chitra is telling me, ma'am, glyceraldehyde it is. So aldehyde group will be there, right? Aldehyde group will be there. Excellent, bache. So aldehyde group, you know that it's CHO. That's the group. It's CHO. That's the group. How do you write it? Like this. It is having the carbonyl bond. It is having the carbonyl bond, C double bond O. This is also a PYQ. Right, it was also a PYQ. They asked it ki which compounds are having carbonyl bond, C double bond O. Carbonyl bond is this C double bond O. Aldehyde and ketone both are having. So this is how you are going to write it. So you have 3 carbons. So here you have to, definitely you have to add the functional group. So this is your functional group and that functional group is aldehyde. Very good. Only one student was up there. So see, it's fulfilled, isn't it? It's fulfilled. So 1, 2, 3, 4, 5 and 6. 6 hydrogen, 3 oxygen, 3 carbon. So it's Cn, H2n, ON, it's C3, it's H6 and it's O3. This is what your glyceraldehyde is. So I have used 2 words. Very good, bacho. I have used two words, aldose and ketose. So aldose having functional group aldehyde, ketose having functional group ketone. So obviously your Wasim sir, your HSP sir, right, they teach you chemistry. Today you are going to feel like it's a chemistry class, don't worry. Right, you'll feel like it's a chemistry class, don't worry. So glyceraldehyde, aldehyde is the functional group, that is your CHO, and it is a terminal group, right? It will be attached to the ends. That's what you need to keep in your mind. Your ketose, right, this is the functional group, it is subterminal. It will never attach to the ends of the chain. It will never attach to the end of the chain. Okay, so for an example, you have dihydroxyacetone, C double bond O. That will be the structure. 1, 2, 3, 1, 2, 3, 4, 5, 6. That is your dihydroxyacetone. So which monosaccharides are aldoses? Having aldehyde as the functional group. Which monosaccharides are ketose? Having ketone as the functional group. Okay, so let's talk about the tetroses. Let's discuss the example of tetroses. Bache, tetroses mein you have the example of erythrose, it's aldose, and here it's very simple, ulose. Okay, I'll give you another example. Bache, this is the nomenclature, like you study in your organic chemistry, right? Nomenclature. When it is aldose, जैसे here priya it is written erythrose, that is aldose. If it is written like this, erythroulose, if this ul is added, ul is added, it's a ketose. If this ul is added, it's a ketose. If this ul is added, it's a ketose, right? Erythrose, erythriolose. Pentose mein ribose is the example, right? Xylose is the example. Here ribulose, ulose is the example, xylulose is the example. Hexose mein you have the example of glucose here. Here you have the example of fructose. Here in heptose you have the example of pseudoheptose. Here you have the example of pseudoheptulose. So I am not saying that you have to remember all the examples, but right, I'm not saying that you have to remember all the examples, but you can see, bache, right, at least pentose, hexose, this is something that you should know, right? The ul and ol part, this ul wala part clear. I'll see, you know what ribose is, you know what ribulose is. So ribose means it is aldose, it is aldehyde as functional group. Ribulose means it's ketose, it is having ketone as functional group. And people, you remember na, RUBP, ribulose bisphosphate or ribulose bisphosphate, and do you remember rubisco, ribulose bisphosphate carboxylase oxygenase, right? The most abundant, it is the, what is it? It is the most abundant protein of biosphere. What's that? It's the most abundant protein of biosphere. That is your rubisco. Ribulose is the word. So this ulose, ul is telling me the functional group is ketose. Understood? And ribose, you know that it's a pentose sugar. Means carbon is 5 here. So even if you don't know, yes students, even if you don't know the general formula, let's say in the paper they have given you one question that what is the formula of the ribose, right? They have given you the question that what is the formula of the ribose. So ribose formula mein n should be 5. N is equals to 5. Okay, n is equals to 5. So C5H 2 into 5 O5. So C5H10 O5 it will be. That's how you can calculate. Okay. So this is about the monosaccharides. Done. Done. Hexose mein along with glucose you have the example of galactose, if you remember. You have the example of mannose, if you remember. Understood? Sure? Sure? Yes, is it clear? Is it clear? Yes, is it clear? Doubt बच्चे? Doubt बच्चे? चलो, so write down one more thing. Write down one more thing. That deoxyribose, right? I hope you know that deoxyribose, right? I'll show you the image as well. Deoxyribose, deoxyribose. जैसे we have the ribose, which is pentose, right? Cn is equals to 5, so C5H2 H10O5 it is. This is your ribose, C5H10O5. Ribose is your C5H10O5. So deoxyribose, you know that it is present in your DNA. So these monosaccharides, they are so important. Ribose is present in your RNA. Deoxyribose is present in your DNA. So you know that at second position O is missing. We'll tell you about it. So C5H10O4, that's the formula. That's the formula of deoxyribose. That's the formula of deoxyribose. So let me tell you students that this deoxyribose, it doesn't follow the empirical or the general formula. Okay, it doesn't follow the general formula of carbohydrates, but still it is a carbohydrate. So that is an exception that you people should know. Okay, deoxyribose is an exception. It doesn't follow the general formula, but still a carbohydrate. Doubt? Yes, that's important MCQ. That's important MCQ. Is that clear? Sure. Chalo. So now let's discuss about the structure. I will take another 10 minutes to explain this part, then we will move to the important topic. So see this, triosis, right? You'll get the PDF students. Triosis, 3 carbon atoms, glyceraldehyde. Tetrosis, 4 carbon atom, erythrose. Pentosis, 5 carbon atom, this. Hexosis, 6 carbon atoms, glucose and fructose. Heptosis, 7 carbon atoms, pseudoheptulose. Okay? So now you know if it is ul-ose or if it is simple heptose, what's the difference? Am I right? What's the difference? So we all know about it, isn't it? We all know about it. Okay? Now bache, we have to discuss its structure. Okay, so that's your homework. Write down your homework. Hanji, write down your homework. Then we'll discuss the importance as well. But firstly, write down the homework. Bache, you have to remember the structure of glucose, fructose, ribose, galactose. It's very simple. It's very simple. These are the 4 structures that at least you should know. Glucose, fructose, ribose, galactose, and even the structure of deoxyribose. It's very simple in this class itself, right? I'll teach you this, but this is something which should be on your fingertips. Okay? This is something which should be on your fingertips. Remember this. Okay, so now when you talk about the structure, in the structure we discuss 2 things. In the structure we talk about the straight chain, we talk about the straight chain, and we also talk about the ring form. Ring form. Like bache, when you talk about the straight chain structures, na, you call it as Fisher projection. It is Fisher projection, that straight chain structure. Okay, and when you talk about the ring form, that is Haworth projection. I hope you know about it. Your chemistry students, of course. Madhumita, yes, you will get the PDF in our Telegram group. You people are going to get the PDF in our Telegram group. Yes. Clear? Any doubt? Clear, clear, clear. So what do you have here? The straight chain is your Fisher projection, ring form is Howard. Okay, Howard. So ma'am, why are you teaching this thing to us? Because we need to focus on the structure. So we'll be focusing on the glucose structure first, right? The straight chain structure of glucose first. Let's talk about it. So how many carbons are there, people? How many carbons are there? Yes, how many carbons are there? Madhumita, Shruti, how many carbons are there when we talk about the glucose structure? When we talk about the glucose structure, quickly tell me. Yes, quickly tell me. Quickly tell me. 6. So which functional group is there? It's a glucose. So it is an aldose. So aldose, which is a terminal group. So that's how I'm going to write it. That's how I'm going to write it. 1, 2, 3, 4. 1, 2, 3, 4. Okay, that is how I'm going to write it. 1, 2, 3 and 4. So basically this is what we have. Now what you have to do, this is your carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. So basically you know that, you know the basic rules of nomenclature. This is the functional group, this is the carbon number 1. I'm not going in that depth. You can ask it from your HSP sir as well. Okay, so this is what we have. So now just look at this. First carbon, this is second carbon, this is third carbon. If I am writing it incorrectly, do let me know, guys. You have to be very active. Might be I am writing it incorrectly, right? I will write things incorrectly in between and you have to point out ki nahi ma'am, it's wrong, it's right. You have to point out. Okay, you have to point out. So carbon 1, 2, 3, 4, 5, 6. Okay, I am not going into the depth of that. This is how your third OHH is present. So bache, we know that it is actually the polyhydroxyaldehyde or ketone. So it's polyhydroxyaldehyde when you talk about the glucose structure, right bache? So here is that aldehyde structure. So carbon number 3, OHH is present like this. This is the Fisher projection. This is your glucose. This is your glucose. Is that clear? What is it? It is your glucose. Clear, 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 clear. Yes, people. Is that clear? Is that clear? So please draw it with me and let's be very quick. Let's be very quick. Right? So C double bond OH, H, OH, OH, H, H, OH, H, OH, CH2, OH. Okay, so here also that's what we have. All three are glucose. All three are glucose. Yes or no? All three are glucose. This is glucose. Now see, it's very simple. This is the Fisher projection. Now bache, this is 1, 2, 3, 4, 5 and 6. Guys, look at this fourth carbon. Look at this fourth carbon, everyone. And here, look at this second carbon. Now tell me, are they glucose? Are these two structure glucose? Are they of glucose? Are they of glucose? Quickly tell me. No, no. Right, they are not Priyadarshini. This one is your galactose. Galactose. This one is your mannose. Right, this one is your mannose. Right, so now when you look at these structures, right, look at these structures, so you will notice that everything is same, but around just one carbon the position of H and OH is different. Right, this is your glucose, this is your galactose, this is your mannose. So what do we notice, Chitra? We notice here that around one carbon the position of H and OH will be interchanged. Around one carbon the position of H and OH will be interchanged. Right, in galactose this has to be specific. Right, here at carbon number 4 the position of H and OH is interchanged. Here at carbon number 2 the position of H and OH is interchanged, and such compounds are known as, that's the term epimers. They are the epimers. Okay, so galactose and mannose, they are the epimers of glucose. Galactose and mannose, they are the epimers of glucose. Is that clear? Thank you, HSP sir. HSP sir, am I right? HSP sir is there in the chat section. Thank you so much for the motivation, sir. So tell me, am I right? So these are the epimers, right? Galactose and mannose, it is the epimer of the glucose. They are the epimers because the position of H and OH is changed around only one carbon, and that's specific. Okay, that's specific. Okay, so this is the Fisher projection. Okay, so now we have to talk about the ring structure. Howard projection. Hannah sir, it's like that. Howard projection, it's like that. So now we have to talk about the pyranose and the furanose. Bache, it is important, trust me on that. Pyranose and furanose. Pyranose and furanose. Okay, pyranose and furanose. Pyranose and furanose. Hanji, this is what we need to discuss now. जैसे glucose का straight chain, now it has to form what? It has to form the ring form now. When we draw the, when we talk about the glucose, that's how we discuss it. This is carbon number 1, HOH, HOH, OH, yes, HOH, H, CH2, OH. Okay, and this is furanose. We'll come to this also. So this ring form, pyranose, furanose, pyranose, furanose, this is what we have to discuss. So for furanose, what you have to remember, 4 carbon in ring, 4 carbon in ring and 1 oxygen. Okay, and 1 oxygen. Like in the case of ribose, we have seen that such ring. We have seen such ring in the fructose as well. It is the pyranose. So in the case of pyranose also, we know 5 carbon in ring and 1 oxygen and 1 oxygen. Clear? 5 carbon in ring and 1 oxygen. Right? And 1 oxygen. Am I right? Am I right? So it is from that pyran and furanose. It is from that pyran and furan. That way I am not going to discuss. Your HSP sir is going to teach you this. Okay? So basically pyranose, it's a 6-membered ring. It's a 6-membered ring bache, having 5 carbon and 1 oxygen. It's a 5-membered ring having 4 carbon in ring and 4 carbon and 1 oxygen in the ring. Okay? So pyranose, furanose, these are the 2 rings that we see. Right? These are the 2 rings that we see. Am I right? Yes, bache, am I right? Am I right? Tell me quickly, students. Tell me quickly. Chalo, so now what's that? What's that? Hanji, what is that? Can you tell me about the structure? Yes, can you tell me about that structure? Anyone in the class? Can you just tell me about the structure? What is it? Exactly. Now it is going to be your ribose. Okay, so ribose, your fructose, they are having this furanose. I'm not going to draw fructose. You will tell me about the fructose in the next class. This is something that you have to practice. So this type of ring where 5 carbon atoms are there and 1 oxygen, this is going to be your pyranose. And that type of ring where 4 carbon atoms are there and 1 oxygen is there, it is going to be your furanose. It is going to be your furanose. Is that clear? Is that clear? So that is how they are arranged and that is how these structures will form. Clear bache? Clear bache? So this is what you should know. Understood? So you can see the structure from this slide as well. Just have a look. This is the aldotriose. Okay, that is your glyceraldehyde. This is your erythrose. Ribose. Look at the ribose. 5 carbon. 1, 2, 3, 4 and 5. Okay, so this is having the aldehyde group HOH, 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 CH2OH. Okay, this is your fructose, because fructose is having what? Its functional group is ketose, right? Ketone is the functional group which is subterminal. So that's how you are going to draw. So even in the fructose at 3rd carbon, OH is towards this side. Any doubt? Any doubt? You can just compare the structure of fructose and the glucose, right? You can just compare the structure of the fructose and the glucose. Clear, bache? So aldose ketose word clear? Aldose ketose word clear? Yes, sure, okay. So now this is the ring form as well. This is your alpha. देखो बच्चे, D and L, it is related to that, right? Left shift, right shift, right? That you remember na, dextrorotatory right one, levorotatory left one. So that is not the part of our biology syllabus. Okay, that is not at all the part of our biology syllabus. So that's why I'm not touching it. You will discuss that dextrorotatory, levorotatory part. You will discuss that in the chemistry. Okay, I'm not touching it. I will teach you just one thing, right? Whenever we discuss such structures, we talk about the alpha glucose and the beta glucose. That is something that we discuss, and most of the time students they don't understand it. Okay, they are confused about it. They don't understand it. They are just confused about it. Alpha, beta. So alpha, A above, beta, B below, A above, B below. Repeat it with me, people. Repeat it with me. Alpha, A above, B below. A above, B below. So alpha above. What is above? Your hydrogen is above. So bache, when you discuss the glucose structure, right, we say na alpha and beta. See, this is your glucose structure. I told you na, you have to remember this. This is your glucose structure. So this is your alpha glucose. Why this is your alpha? Because we have to see the carbon number 1. This is your carbon number 1. This is your carbon number 2. This is your carbon number 3. This is your carbon number 4. This is your carbon number 5. And here we have our carbon number 6. 1, 2, 3, 4, 5 and 6. So around this carbon number 1, we have to see the position of hydrogen and this hydroxyl. Take care. We have to see the position of hydrogen and hydroxyl. Clear, bache? Clear, bache? Yes, clear? So alpha A above, hydrogen is above. Now this one is your beta glucose. What's that? This one is your beta. This one is your beta glucose. Why? Because around carbon number 1 we'll see it's below, hydrogen is below. That's what you have to figure out. Any doubt here? Alpha, beta, any doubt here? Tell me quickly. Tell me quickly. ठीक है. So this is about the monosaccharides. This is about the monosaccharides. I'll tell you the importance. Look at the ribose, right? So this is a beta ribose. Why beta ribose? Because this OH is above and the hydrogen is below. So this is important for NEET. It is given in NCERT as well. This is deoxyribose. This is deoxyribose. So at second carbon, oxygen is absent, O is absent. This is another important MCQ, people, which we are not allowed to miss. Understood? Which we are not allowed to miss. So it should be straight here, like HSP sir said na, in your hippocampus. Okay? In your hippocampus it should be. This is important. Okay? So now let's discuss the few examples, right? I'll speed it up now. So now let's discuss the few examples of these monosaccharides, and then we will move towards the oligo one. So glucose, you know that it is the main respiratory substrate. Do I need to mention it? It is also known as blood sugar. Okay, it is also known as blood sugar. It is our main respiratory substrate, the glucose. It is abundant in blood, isn't it? Isn't it? Right. Then when you talk about your fructose, fructose is the fruit sugar. In the fruits, which sugar is present? It is the fructose, right? Important it is. But remember one thing, G for grapes, right? In grapes na, that fruit sugar is your glucose only. Grapes mein it's not fructose, that fruit sugar is your glucose only. Right, it is your glucose only. And if you remember fructose, it also provide energy to the sperm. When sperm moves in female vagina, they are not dependent on glucose, they need fructose. They use fructose there. Now the next is what? Next is your galactose. Next is what? Next is your galactose. What's that? Next is your galactose. So galactose is known as brain sugar. Do you know why? Is it like the brain cells feed on galactose? That's why? Or what's the reason? Your galactose is the brain sugar. Can you tell me why? Because it is most abundant in brain and nervous tissue. Okay, it is most abundant. It is most abundant in brain and nervous tissue. But it's not like that ki this galactose, the cells will feed on this galactose. No, it is actually the structural component there and not directly. Actually in the brain lactose is there. Lactose is a disaccharide that is having the galactose. So that's why we say that it is abundant in brain. Right, on that basis we say it is abundant in brain. So this is something that you people should know. Take it. So it occurs, right? So this galactose, this is the brain sugar. This is the brain sugar. It is the most abundant in brain and nervous tissue, right? It is most abundant in brain and nervous tissue, right, people? But it is never in free form. It never occurs in free form. It is always the component of the lactose. Take it? It never occurs in free form. Okay? Right, so that's what you people should know. Any doubt? So initially I use this thing now. I told you that remind me later. It is something very important that we have to read, that we need to read, the reducing sugar. So I told you that your monosaccharides, they are the reducing sugar. Which sugars are the reducing one? This is important MCQ, people pay attention. This is important MCQ, people pay attention. Guys, quick, please pay attention here. Please pay attention. Let's master this chapter. Right? Let's master this chapter. What say people? Let's master this chapter. Let's not leave anything. Okay, let's not leave anything. Reducing sugars, the sugars having free aldehyde or ketone groups. The sugars having free aldehyde, or ketone groups, that can further do what? That can convert your Cu2 positive to Cu positive, right? So what is it? Is it cupric or is it cuprous? Is it cupric or is it cuprous? I think my students can answer this. right? My students can answer this. This is cupric ion and this one is cuprous. So because in the reducing sugar they have free aldehyde or ketone group, right? Their aldehyde or ketone groups are free. They are not attached somewhere. They are free to react, right? They have free aldehyde or ketone group and can reduce Cu2 to Cu positive. So these are the reducing sugars. Got my point? These are the reducing sugars. Okay, very good. Can you tell me its use? Yes, people, can you tell me its use? Can you tell me its use? That if I'm saying that they are having the free aldehyde or ketone group, they are having the sorry, they are having the free aldehyde or ketone group, then how is it going to help? Bachi, that Benedict test in the chemistry, Benedict test in the chemistry, there used to be many tests, na, Tollens reagent, this reagent, that reagent, I still remember. Not that complete test, obviously just the name, right? So sometimes na, जैसे urine में, you know that the presence of glucose in urine is not good, है ना? If there is the presence of, Samiksha, if there is the presence of glucose in urine, then it indicates what? We call that condition as glycosuria. We call that condition as glycosuria, and that is indicating the diabetes, right? It is indicating diabetes mellitus. It is indicating diabetes mellitus, isn't it? Hanji, isn't it? So how are we going to check that? It's not like someone is going to taste the urine, ki, "Arey yaar, urine is sweet, that person is having diabetes." Of course not. Okay, that's yuck. Are you getting that yuck feeling? Imagine someone is tasting the urine and telling, "Arey yaar, urine is sweet," means that person is having diabetes. Of course not. Of course not. We have to perform some test, right? We have to perform some test. That Benedict test and this and that, they are used for detecting such things, right? It will be performed on urine to check whether glucose is there or not. Okay, okay. So this is what you have to remember. No one is going to drink the urine and to tell you, "Kyare, do you know what? Do you know what? Your urine is sweet. You might have diabetes." Of course not. Of course not. Chalo, now come back to next point. That is oligosaccharides. What are oligosaccharides? What are oligosaccharides? Tell me, what are oligosaccharides? Chee, अरे मैम, है ना? Chee मैम, cheap talk. Now tell me, what are oligosaccharides? I love this chapter. Literally, I do. So be ready today, huh? Be ready. I have seen you people, I have seen your dedication for the physics and the chemistry classes, right? HSP sir, he takes the session for 10 hours, 12 hours. Your Yava sir, that modern physics, I have seen 10 plus hour ka session. Okay? So for the biology also, there are some lengthy chapters. In molecular basis of inheritance, you people started crying. In evolution, you started crying after 9 hours. Ki, nahi ma'am, it's too much. है ना? It's too much, ma'am. Even in molecular basis of inheritance, I still remember I was asking the questions from students and they were just writing anything. They were not actually listening to me even. Or maybe you were not able to process it. Otherwise, that session can easily take 10 to 12 hours or 13 hours as well if we discuss that chapter in detail. That's a lengthy chapter. Same is the biomolecule. Okay, so if you want to run, you can. You have the choice. And trust me, if you don't know the basics of carbohydrates, you just need to cram polysaccharides. And this is not something that I want from you people, because this is also related to chemistry. Okay, I told you now, whenever you are preparing for any entrance test, just remember one thing, it is not just the subject, physics, chemistry, biology, right? You have to absorb the entire syllabus without any discrimination. Physics is important, chemistry is important, and biology is very, very, very important. We are not allowed to miss it, right? We have to give our 200 and 300% in biology, mathematically not possible, but yeah, right? And we have to give our 100%, 150% in chemistry and physics. Biology is making 50% of the syllabus, and on this channel the biology, right, is most underestimated subject. You don't like to read biology, you just want physics and chemistry, and I'm jealous of that, yaar. I'm openly telling you, I'm jealous of that. Why not biology? Biology is mandatory, we have to master it, then we'll think of physics and chemistry. Okay, okay, chalo. So jokes apart, now come back to the topic. Now, how can you take 2 hours straight, your energy level? Oh, Madhumita, I love this chapter, first of all. And second thing is, yaar, after so long, I'm meeting you people, right? Obviously, the energy should be there. And do not forget to watch this. That change in NCERT. That change in NCERT. All the changes in NCERT are explained here. And I have clarified that confusion also, ki what exactly is that old NCERT or the new NCERT wala thing. Okay, so that also you have to remember. Fine, that also you have to remember. Clear? So it is also a must watch. Do check it out. Fine, chalo. So now let's go to the oligosaccharides. So don't worry, in between I'll keep entertaining you, because these days teachers are not teachers, they are the entertainers. If they are not the entertainers, no one is going to watch their classes. Am I right? Am I right? Hai na? Ma'am, when will be microbes in human welfare? Dipanvita, I know that chapter is pending from your class 12 syllabus. Human health and disease I took on Plus platform. The session, the marathon was still not completed. The reason was I was not well on that day. So don't worry, I will take that marathon before your NEET exam. Okay, now let's complete 11 syllabus. And microbes in human welfare, I will take it. That session is just for 2.5 hours or for 3 hours. Okay, so when I'll be very, very, very free, when I will not be in the mood to teach you, that day I will take it. Okay, chalo. So in between I'll keep teaching you. As I said, teachers are entertainers these days. If they are not entertaining the kids, students are not going to watch their lectures, hai na? So in between we'll keep talking. Chalo, so oligosaccharides. So oligosaccharides, in oligosaccharides we discuss disaccharides, people. Even the tri, even the tetra, basically up to 10 monosaccharides. This is what we discuss now. When we say oligosaccharides, so you know that here the number of monomers, it's not the number of carbon, students, it's the number of monomers, number of monosaccharides. It is from 2 to 10. Number of monosaccharide is from 2 to 10. The number of monosaccharide is from 2 to 10, right? So 2 monosaccharides means 2 sugars, they are attached to each other. Oh my god, look at this lubby-dubby. One sugar is in love with another sugar, they have a bond in between. Who's going to connect them? The bond. It's a disaccharide. So if 3 monosaccharides are in love triangle, trisaccharide. If 4 monosaccharides are joined together, that is the tetrasaccharide. That is the tetrasaccharide. So what is the part of our syllabus? In our syllabus, we discuss disaccharide. So di means 2. Saccharide means sugar. So 2 sugars are attached together. 2 sugars are bonded together. So which bond do they have? Actually, this is what we need to discuss. It's your glycosidic bond. It's your glycosidic bond. So in carbohydrates, which bond do we have? The glycosidic bond. So everyone write this word in the chat section quick. The bond is glycosidic bond. Okay? Now, whenever I used to explain this topic before, I always say ki lactose, sucrose and maltose. You should know about these 3 disaccharides. Why? Because it was discussed in your digestion and absorption. In your digestion and absorption, in your old syllabus. Okay, now the digestion and absorption is deleted na. Now the digestion and absorption is deleted na. So we discussed that na, in digestion and absorption, maltose, lactose, and the sucrose. Okay, and sucrose word is also there in transport and plants. Now these chapters are deleted. So if you do not want to, okay, if you do not want to remember these disaccharides, it's purely up to you. But as a teacher, what is my suggestion? Because after syllabus deletion, your NEET 2024 examination will be the first one na. So after that paper, we can analyze, right, that what are they considering? Are they considering the deleted syllabus also? It's not properly deleted, it is somewhere in biology, in other chapters na. So that thing we will understand after your syllabus. So it is my advice that you should read these 3 disaccharides. Okay, okay. So oligosaccharides are the one, they are the carbohydrates which can yield 2 to 10 monosaccharides. Okay, which can yield 2 to 10 monosaccharides. Understood? Understood? Yes, bache. So we have to discuss the glycosidic bond formation here. But before starting that glycosidic bond formation, I will tell you, I'll mention you the 3 examples of the disaccharides: the maltose, lactose, which is also the milk sugar, and the sucrose. So maltose is made up of 2 glucose molecules. Glucose plus glucose together will form maltose. When it comes to lactose, see, in all the 3, glucose is common. Fine. In all the 3 disaccharides, glucose is common. Okay, so now it is lactose. So here it will be galactose. That's how you have to remember. And in sucrose, you have fructose. Right? You have fructose. This example should be clear in your mind. Okay, so what is this maltose? It's the malt sugar present in germinating seed. Okay, lactose is your milk sugar. Your milk is having which sugar? Lactose. Your milk is having which sugar? Lactose. And which protein is present in milk? Casein protein. Which protein is present in milk? Casein protein. Which sugar is present in milk? Lactose. Which protein is present in milk? Casein. That's what you have to remember, people. And it is your sucrose, which is also known as your invert sugar, which is also known as your table sugar, cane sugar. Right? The sugar that we eat is basically the sucrose. Okay, the sugar that we eat is basically the sucrose. Clear, bache? Clear? Doubt? Okay? Any doubt here? Sure? Sure? So remember, in lactose it is always beta glucose, beta galactose, and beta fructose. This is extra. You want to take it, you can. You want to drop it, you can. It's purely your wish. Okay, it's purely your wish. So let's start with the simplest one. Okay, let's start with the maltose. I'm teaching you what? I'm going to teach you the formation of the glycosidic bond. Which bond is present in these carbohydrates? Glycosidic bond. Bacha, this glycosidic bond is in biomolecule also. This N-glycosidic bond is in nucleic acid also. So please pay attention. Okay? So let's draw the structure of glucose, and please mark my words. Okay, mark my words. I have given you the homework now. Remember that structures only. Okay, just remember that structures only. That will be helpful for you. So this is your 1 glucose. This is your another glucose. Okay, so this is carbon number 1, this is carbon number 2, this is carbon number 3, this is carbon number 4, this is carbon number 5, this is carbon number 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Now, bache, ultimately in this glycosidic bond, there is the dehydration. This is the first carbon and this is the fourth. So the bond will form here in between the first and the fourth carbon. Right? In the NCERT itself, you might have seen things like this 1, 4. Right? Yeah, beta 1, 2. I don't know. We will discuss it in polysaccharide. I don't know whether you know it or not, but we will discuss it in polysaccharide. ठीक है, alpha 1,4, beta 1,4. So it is just telling us about the ग्लूकोज के अल्फा है या बीटा है, एंड फर्स्ट कार्बन फोर्थ कार्बन, फर्स्ट कार्बन फोर्थ कार्बन, फर्स्ट कार्बन फोर्थ कार्बन. फर्स्ट ऑफ ऑल टेल मी दिस पार्ट क्लियर, फर्स्ट ऑफ ऑल टेल मी दिस पार्ट क्लियर guys, quick, this part clear? Chalo, so now let's pay attention here. Oh no array, okay, so HOH. So ultimately what is going to happen? What is my bad students, I told you na, in between I will write incorrectly, you have to rectify it. Guys, be aware, please be aware. That's glucose, that's glucose. So basically what is going to happen, the dehydration will occur. Dehydration will occur means water will come out, means minus H2O will be there, minus H2O will be there, है ना? Right, minus H2O will be there. This is one glucose, this is another. So what is going to happen? Removal of water, dehydration. So obviously after dehydration, this is what you people are going to get. Am I right? This is what you people are going to get. So this is what we have here. Is that clear? This is what we have here. Is that clear? This is what we have. Understood? Right? So one glucose is linked to another. This is the glycosidic bond, people. This is the glycosidic bond, and you know that it is formed because of the dehydration. So here it is in between carbon number 1 and carbon number 4. Carbon number 1 and carbon number 4. So this is about the glycosidic bond. So you can see this, right? So condensation will be there, that joining will be there, and that's your glycosidic bond. This is your maltose, right? This is your maltose. So again you can see glucose means maltose, right? So now this is the lactose. When you know na, lactose means glucose plus galactose. What is the meaning of lactose? Glucose plus galactose. That's what I just said. So see bache, this is your glucose, this is your galactose. So it is a galactose, and which galactose it is? It's a beta galactose. It's a beta galactose. So beta it is. So obviously you know that OH will be above, H will be like this. So that's why here in this bond now, this is not like this. This is like this. Okay, so no need to get confused. Ultimately it is the glycosidic bond only. So lactose is having beta 1,4 glycosidic bond, like it is having alpha 1,4 glycosidic bond, right? So it is having beta 1,4. So this beta is saying that this sugar is a beta sugar, beta 1,4, and it is alpha 1,4. Understood? Right? Understood? Now then come to the sucrose, my favorite, sucrose, where your fructose is beta. Okay, so here also glycosidic linkage is there, bache. You can see this. Okay, so sucrose is basically your non-reducing sugar. It's a very important question, students. It's a very important question. After it I will give you a break, and then we will start polysaccharide. Okay, so please answer this. So sucrose is what? Sucrose is a non-reducing sugar. This is something very important. This is something very important. Yes, sucrose is a non-reducing sugar, and we also call it as invert sugar. We also call it as invert sugar, table sugar, commercial sugar. Yes, sucrose, it is non-reducing. It is not able to reduce what? Not able to reduce your cupric to cuprous ion. That's what you have to remember. Understood? That's what you have to remember. Right? Right? And in the sucrose, the bond is in between alpha 1 and This is 2, right? So this is the first carbon of glucose and this is the second carbon. This is the second carbon of fructose that forms the bond. Okay, although in that depth it will not be asked in biology, but still it is the part of chemistry, that's why I'm telling you. So sucrose is a non-reducing sugar. And students, why do you know why do we call it as invert sugar? Just there is one enzyme invertase that hydrolyzes this sucrose. Just if you want to hydrolyze the sucrose, you can use the enzyme sucrase or invertase, same. Why do we call it as invert sugar? It is in chemistry. It is in chemistry. Why do we call it as invert sugar? I don't remember it exactly, you know na, that it invert that sucrose, when it is in sucrose form, it rotate light in a different plane. When it get hydrolyzed, it rotate light in different plane, something like this. I'm not able to recall it properly. I'm telling you this thing honestly. No, that is reason is related to what I'm saying. Okay, structure changes here and the rotation of the light also changes. I really don't remember. Right, exactly. I have started it in class 12th, class 11th only, class 12th biomolecules. Okay, I didn't revise it later. It is related to plane polarized light concept, that is for sure. Now you have to answer it. Okay, and you know na, in transport in plants, though that chapter is deleted, but still I'm telling you, in the phloem na, how that glucose translocate in the form of sucrose. How that glucose translocate in the form of sucrose. So it is also a non-reducing sugar. Okay, it is also a non-reducing sugar. So these two sugars are given in NCERT, glucose and the ribose. Fine, glucose and the ribose. So ribose which is given in NCERT is the beta ribose. Okay, so the structures which are given in NCERT, we are not allowed to miss it. So after it we will be starting bache, after it we will be starting polysaccharide. Till then there will be a break for you. Okay, there will be a break for you. So break time is till, it's your dinner break. So break time is from, so this is the break time. So for how long you need the break? For how long you need the break? 10 minutes, 15 minutes? 10 minutes or 15 minutes? No, not till 9, sorry. Chalo, break till 8:45. Then again there will be a long session for 2.5 hours, then 2.5 hours. The session will be lengthy. Okay, be prepared. The session will be lengthy. Hi, hello. Welcome back, people. Welcome back. What's up? Hello. So, how many of you are charged and ready to finish this chapter? How many of you are charged enough and ready to finish the chapter? Sure, chalo. So let's start the polysaccharides, and trust me, if you know the concept, you are in love with, right? You will fall in love with this chapter. Trust me on that, right? That's very good, very nice, very lovely chapter it is. Trust me. Okay, so now the next thing that we need to start is polysaccharide. So where are you going to find the polysaccharide? In acid soluble fraction or in acid insoluble fraction? Where are you going to find it? Quickly tell me, where are you going to find it? In acid soluble fraction or in acid insoluble fraction? Yes, Samiksha, yes everyone, and guys, share the session link with your friends, yeah, invite them, right? You cannot surrender so early. We have to start the amino acid, we have to finish the amino acid, we have to start the proteins, we have to start the lipids, we need to talk about the nitrogenous bases. The session is going to be very long, right? And now I am going to increase my speed as well. It's a one shot now, so I am going to increase my speed as well. So I want you people to be the part of the session, right? Might be your session will go till 1 or 2, but today I'm not going to end it, and I want you people to support me. Okay, so the word is polysaccharide. So we know that they are going to be in acid. Which fraction? In acid insoluble fraction, right? So acid insoluble fraction means you are talking about what? You are talking about the retentate in which we are going to get it, right? So bache, for the polysaccharides, the word that we use, the suffix here is ans. Basically these are the glycans, right? When you use the word glycans, it means you are talking about the polysaccharides. Okay, when you use the word glycans, right, when you use the word glycans, right, so it simply means we are talking about the polysaccharides. Understood? We are talking about what? We are talking about the polysaccharide. This is what you need to keep in your mind. Clear, bache? Now when we talk about the polysaccharides, let's talk about their properties. So when we talk about their properties, remember they are insoluble in water. Yes, they are insoluble in water, plus they are tasteless, and they are optically inactive. What are they? They are optically inactive. What are they, people? They are optically inactive. Am I right? Yes. Am I right? Yes, optically inactive, insoluble in water, and they are what? They are tasteless. And here what are you going to see? You will see that number of monosaccharide will be more than 10. Okay, more than 10 monosaccharides are going to make your polysaccharide. So their molecular weight is going to be high. Understood, people? Their molecular weight is going to be high. So please do not miss that part. Okay, please do not miss that part. And as I said, when you talk about their suffix means the something that we are going to use at end. So suffix will be an, like glycans, right? Glycans. Clear, bache? So remember, all polysaccharides, this is the line from NCERT, everyone, so please pay attention. So all polysaccharides are non-reducing. What are they? They are not reducing, they are non-reducing. They cannot reduce the cupric into the cuprous. They are non-reducing. But here in the NCERT, along with this line, there is one more line added, right? Overall, polysaccharides are what? They are non-reducing. But there, when you talk about their ends, just say, let's say, for an example, let's say this is the polysaccharide. So it will be having two ends. It will be having the ends. So when you talk about their ends na, so there in their ends, there will be a right end, there will be a left end. There will be a right end, there will be a left end. Yes or no? Yes or no? Yes or no? Yes or no? Tell me quickly. All the polysaccharides, they are non-reducing, but bache, they are non-reducing, but in a polysaccharide chain. But in a polysaccharide chain, one end is reducing and another end is, right? But in a polysaccharide chain, one end, yes, one end, that is your right end, is reducing. One end of that chain we are discussing, and another end, that is the left end, right? And another end, that is the left end, right bache, that is going to be non-reducing. Understood? That is going to be non-reducing, means overall as a molecule, as a compound when we see, right? Overall as a compound when we see, so when we say that the polysaccharides, they are the non-reducing one. But if individually you focus on their ends, then you say one is reducing, one is non-reducing. Then how to remember that, ma'am? It's very simple. It's very simple. R for right, R for right, R for reducing. So the right end is going to be the reducing one, right? The right end is going to be the reducing one. Any doubt here, students? Any doubt here? So this is very important topic. Clear? This is very important topic. So bache, on the basis of structure, okay, so on the basis of structure, how do we divide these polysaccharides? Simple. So on the basis of structure, you have homopolysaccharide, yes, you have homopolysaccharide, and you have heteropolysaccharide. Okay, you have homopolysaccharide and you have heteropolysaccharide. What is the meaning of homo? Right, I'll tell you about the examples as well. Individually we will pick up examples. Homo means same. Like some polysaccharides, they are made up of same monosaccharides. Hetero means different. Some polysaccharides, they are made up of different types of monosaccharides. So that is the point here. Understood? That is the point here. So let me explain this part. So on the basis of structure, firstly we are discussing. What are we discussing? On the basis of structure, right? On the basis of structure. So homopolysaccharide is the word. Okay, homopolysaccharide is the word. Same, right? So same type of monosaccharides are going to make homopolysaccharide. Just say, let's say if it is a circle. So in that chain, all are going to be the circles. Okay? So that is how you have to remember. And ultimately students, which bond will be there? Which bond will be there? Yes, which bond will be there? Which bond will be there? Quick, I want to see the answer in the chat section. I want to see the answer in the chat section. Which bond is going to join these monosaccharides together? Yes, people, which bond is going to join these monosaccharides all together? Seriously? Bache, one student is saying polypeptide. Bache, I can't help you. I can't help you. It's a request of mine. Then stop learning this chapter. I am not a good teacher for you then. If you are saying polypeptide, how can this be polypeptide? You are talking about the carbohydrates. You are talking about the glucose. Even the words they resemble. Glycosidic it is going to be, people. Okay, glycosidic bond it is going to be. So what are the examples that we have? We have the example of glycogen, which is a homopolysaccharide. We have the example of starch, which is a homopolysaccharide. We have the example of chitin, which is a homopolysaccharide. We have the example of cellulose, which is a homopolysaccharide. We have the example of inulin. It's not insulin, it's inulin. It's inulin, something important. Right, something very important. It is inulin is the word. What is the word, students? It is the inulin which is the word. Is that clear? Is that clear? So homopolysaccharides all are going to be same, right? All are going to be same. Then comes the heteropolysaccharide, right? Then comes the heteropolysaccharide. Okay, so when you people talk about the heteropolysaccharide, obviously hetero means what? It means different. So it is made up of different type of monosaccharides, right? Like let's say circles, squares, right? Triangles, like this, right? So different monomers are there. So can you quote any one example here? Any one example can you quote here in heteropolysaccharides? Yes, when we discuss the heteropolysaccharide, can you just quote even a single example? Even a single example? There are many. At least quote one single example quickly, people in the chat section. Excellent, Chetu. Chetu is like, ma'am, pectin. Excellent. Your heparin. Okay, hyaluronic acid. You know na, hyaluronic acid, it is the cementing material. Hyaluronic acid, it is the cementing material. Even that hemicellulose. Cellulose is homopolysaccharide, but hemicellulose is a heteropolysaccharide. So pectin, heparin, hyaluronic acid, right bache, hemicellulose, these are the examples. Understood? These are the examples, right bache. Your proteoglycans, like your mucopolysaccharides, right, your mucopolysaccharides. So these are the heteropolysaccharides. So this is as per the structure. We will discuss their structures individually as well. So up to this point all clear? Up to this point all clear? So I'll tell you the categories also. Yes? Right, so this is what we need to discuss in detail. Okay, so bache, other than that now, on the basis of function also, do you know that on the basis of function also we divide carbohydrates? Yes, on the basis of function. These polysaccharides are of different type. Like you have storage polysaccharides. You have what? You have storage polysaccharides, structural polysaccharides, and bache, we have even the mucopolysaccharides. I will discuss the mucopolysaccharides here. Okay, I will discuss the mucopolysaccharides here. So let's talk about the structural polysaccharides. So you know that which are going to make the structure. You discuss the example of cellulose, you discuss the example of chitin, you even discuss the example of your hemicellulose, your pectin. Okay, oh sorry, not it is about the structural one. It is about the structural one. When it comes to storage, so you have the starch and you have that glycogen, right? They are meant for the storage. They are meant for the storage. Firstly, we have to talk about what? Yes, bache, Jeetu, the storage polysaccharide. So storage may, these are the reserve food. And it is not just starch and glycogen, bacha, it is inulin as well. It is inulin as well. So if you have any confusion related to inulin, let me tell you, it is the polymer of fructose, right? It's a homopolymer of fructose. Insulin is a protein. It is a question. It was a question that has been asked in NEET 2020. Insulin is a protein. Inulin is a carbohydrate. It's a polysaccharide. Insulin, protein hormone made up of different amino acid, right? Functional insulin is having 51 amino acid. Inulin, it's the polymer of fructose. The same fructose will join and they are going to form the inulin. Clear, bache? So structure may, as I said, the cellulose is the example, chitin is the example, hemicellulose is the example. There are many examples. Now what about the mucopolysaccharide? Anyone in the class who can explain me the mucopolysaccharide? Yes? Anyone in the class who can explain me the mucopolysaccharide? Anyone? What are the mucopolysaccharides? Yes, people, what are the mucopolysaccharides? What is the meaning of mucopolysaccharide? Yes, even one example, if you can give one example to me. Yes, even a single example. Bache, obviously these are the slime producing. These are the slime producing, right? Slime producing polysaccharides. Yes, your agar-agar, your heparin, your hyaluronic acid, they all are the examples of mucopolysaccharides, right? They all are the examples of mucopolysaccharides. So they are slime and mucilage, right? They are slime and mucilage producing, right? They are slime and mucilage producing polysaccharide. And how can they do that? They can obviously hold the water, right? They can all hold the water. They can make it a jelly like. So agar, even the heparin, even the hyaluronic acid, okay, which is even the cementing material. Okay, that is the example of mucopolysaccharide. Okay, so here we have mentioned the hyaluronic acid, heparin separately. So even if you want to remove this as example, you can. It's purely your choice. Okay, right? So this is what you need to remember. Understood, bache? This is what you need to remember. Now let's discuss the structure. Let's discuss the structure. So firstly, let's start with the structure of your cellulose. So cellulose is a polysaccharide. That's what I said. It's a homopolysaccharide. So what do you know about cellulose? Cellulose is present in plant cell wall. The paper that you are getting, that pulp, it is having that cellulose now. है ना, it is having that cellulose now. We will read it from NCERT. So बच्चे, when you talk about the cellulose, cellulose is a linear, it's a linear polymer of beta glucose, right? What is the meaning of बच्चे being linear? What is the meaning of linear? Straight chain. Straight chain. It is not branched. Straight chain. So it is having beta glucose. So which bond will be there? Glycosidic bond. Beta 1,4 glycosidic bond. Understood? Beta 1,4 glycosidic bond. Is it? It is present here in this. Yes, bache. It is present here in this cellulose. Understood? Yes, bache, which bond is there in cellulose? It is having the beta. Yes, bache, it is having the beta 1,4. So beta glucose is going to make the cellulose. Understood? Understood? Yes, understood. So in wood, cellulose is near about 50%, right? In wood, cellulose is near about 50%. So rest we will discuss it from the NCERT. Now let's talk about the glycogen. What do you people know about glycogen? What do you know about glycogen? I have repeated it so many times that glycogen is the animal starch. What's that, people? It is the animal starch. Am I right? What is it? It is the animal starch. Okay, it is the stored food in the case of animals. Just you know now, when there is the excess of glucose, that glucose will be taken by the hepatic cell under the action of insulin, right? Under the action of insulin, and here the glycogen will be formed. Glycogenesis will be there, the formation of glycogen, right? Glycogenesis will be there, the formation of glycogen. Clear, bache? Clear, bache? So this part clear? Yes, students, this part clear? So glycogen is also, right, it is also a storage. It is also a storage polysaccharide, not in just liver, but also in the muscle cells. In the muscle cells also we know that it is stored. Am I right? In the muscle cells also, Madhu bache, it will be stored. Yes, bache, it's a quick revision session, so please support me. Please quickly answer the questions that I am asking. Yes, so here you have the glucose. Obviously it's the glycogen, right, bache? It is the glycogen. So now you tell me, is it branched or is it unbranched? You tell me, is it branched or unbranched? Guys, it is branched, right? It is branched. Okay, even your starch is also branched. Okay, even the starch is also branched. So as of now, what are we discussing? Glycogen. So in the glycogen, what is going to be there? It is the branched polymer, okay, polysaccharide. Now let me tell you about the branches here. See, imagine, it is ultimately a homopolysaccharide. That's what I already explained you, that it's a homopolysaccharide. It is made up of same monosaccharide, and that is your glucose. What is it? That is your glucose. So glucose is going to make the glycogen. So I told you it is branched. I'm not being specific here. I'm just trying to explain you the meaning. Okay, so it is branched. Just let's say these are the glucose subunits. They all are joining. Okay, there comes a point at which the branching will be there. है ना? At which the branching will be there. Okay, I'm just giving you one example. Let's say that's how the branches are present. Just quoting one example, Krutika, Samiksha, just quoting one example. Okay, Kevin. Now what's my point? See, you know that ultimately it is made up of glucose. Let me draw this. So that is how the structure will be. That is how the structure will be. One glucose, second glucose, third glucose, fourth glucose. There are many glucose subunits. So let's say that's how you have the, right, let's say that's how you have a chain. Okay, you have a chain. I'll come to that point, n-1, n everything. Shakir bache, shukriya, right? So that is what we have to discuss. Now my point is branching is also there. Now when it is a straight chain, the bond will be 1,4. You can forget about alpha. Because it is not mandatory that always it will be alpha glucose. Just cellulose mein we have the beta glucose. But ultimately when it is a straight chain, when it is what? When it is a straight chain. Okay, so in a straight chain here 1,4 glycosidic linkage will be there. Only if it is the fructose, then that bond will change. Then it will not be, right? then it will not be 1,4. So if it is a glucose, so 1,4 glycosidic bond will be there in straight chain. Now when it comes to the branching, this is important बच्चे, this is important. जैसे let's say so because I just explained it, so I am not entering in the depth of it, I am not explaining it to you in detail, okay? But I hope you all can imagine that this is your carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6, carbon number 1, 2, 3, 4, 5, and here also there will be carbon number 6. Okay, so let's say the branching will be there. So this is one glucose. To it the next glucose will attach. Yeah, to it the next glucose will attach. So when that next glucose will attach, now at the branching point the bond will be there like this, 1, 6, right? Wherever there will be a branch, okay, 1, 6 glycosidic bond formation will be there, right? 1, 6 glycosidic bond formation will be there. What will be there, people? 1, 6 glycosidic bond formation. What will be there, people? Yes, what will be there? 1, 6 glycosidic bond formation. Understood? Understood? Understood or not? 1, 6 glycosidic bond formation will be there. Okay, okay. So how come 1, 6? Obviously the first carbon has to be there, it has to take part in the bond formation. So this is, from this point the branching will be there, no? The branching will be there, okay? So the 6th carbon, it is having that OH, the first carbon, it is having the OH. So here dehydration will occur. Here the dehydration will occur, Priya. So wherever there is the branching, right, the bond formation will be there. So in between first and the 6th carbon. Okay, and then obviously further glucose will be added here. So wherever there is branch, there you are going to get 1, 6 glycosidic bond. So anyone having any doubt here? Tell me, anyone having any doubt here? We'll show you the images as well. Just see, in the NCERT it is given the portion of glycogen. They are showing us that chain. So wherever there is the straight chain, 1, 4 glycosidic bond is there. Now let's say, look at this point. Here you have the branch. So this is the branch here. So at that branching point, it will be 1, 6. This is important. Now further again, 4th 1. Then again, because again it is also getting added in a straight chain. So here again 1, 4. So only at the point of branching, 1, 6 glycosidic bond will be there. Like first carbon of one sugar, 6 carbon of another sugar. Doubt? Yes, people. Any doubt? Quick. Any doubt? Anyone having any doubt? Sure. So this is your glycogen. So just to make sure that you are okay, right? This is about your glycogen. Let me check. Okay, so that's how they have shown the bonding. Okay, that's how they have shown the bonding. So you can see this, right? So here you can see the same subunits are joining. So at points, branches are emerging. Then again, let's say if this is a branch, further the straight chain, in straight chain it will form. So wherever there is the branching, 1, 6 glycosidic bond, MCQ. So that notes will be provided to you in Telegram group. There is no confusion. You can just take a minute and you can just observe it. If still there is any doubt, do let me know. Do let me know then. Okay, so then cellulose mein I told you it's straight, but beta 1,4 linkage is there. Which linkage is there? Beta 1,4. It's not alpha, it's beta 1,4. Why? Because here you are having beta glucose. What do you have here? You are having beta glucose. Understood? Understood? This is important. So beta glucose, you know that here you are going to have OH and H. So see, that's how the bond will be. So it's beta 1,4 linkage in the cellulose. Understood? Understood? So it is also shown like this. Oh, no, not that one, this one. This one you need to focus. So here you people can see the cellulose, the straight chain, right? Now the glycogen is also branched, and then comes the starch. So this is what we need to discuss next, the starch. Clear bache? What we need to discuss next? The next that we need to discuss is starch. So tell me, what do you know about starch? What do you know about starch? Where are you going to get starch? In filtrate or in retentate? Where are you going to get starch? Where are you going to get starch? In filtrate or in retentate? In filtrate or in retentate? Tell me, in filtrate or in retentate? Hanji, filtrate or retentate? Answer it like this. Filtrate or retentate, how are you going to get it? In retentate, of course. So now starch is also a branched polymer. Okay, it is also not unbranched. Starch is also a branched polymer. What's that? It is also a branched polymer. Okay. So in the case of starch, when you people talk about the starch, it is also made up of glucose. It is highly branched. It is also made up of glucose, right? Glucose molecules are going to join. But in the case of starch, we discuss it in two ways: amylose and amylopectin. Amylose and amylopectin. How many of you are aware of it? Amylose and amylopectin. How many of you are aware of it? Amylose and amylopectin. जैसे your amylose, okay? जैसे for an example, your amylose. Amylose is a straight. It's a straight chain. Amylose is what? It's a straight chain, okay, but helically coiled. But it is helical, simple, like this I can write down. Just let's say this is a straight chain that you have, you are coiling it. That doesn't mean it's branched. For an example, best example is our mic. It's a straight chain, it's a straight chain, it is not having branches. I am coiling it, right? I am coiling it, right? Thank you so much, bache. Thank you so much, Shahrukh bache. So I am coiling it like this. Okay? This is straight chain, Madhumita. This is straight chain, means it is unbranched, and I'm coiling it like this. This is your amylose. What's that? That is your amylose. So it is unbranched, right? It is unbranched, but it is helically coiled, yeah, helically arranged. What is it? It is helically coiled. It is helically arranged. That is what you need to keep in your mind. Clear? Amylose. What is it, bache? Amylose. Right, bache? So amylose means 250 to 300 glucose units are arranged in a, they are arranged in a unbranched chain. Right, bache? So 250 to 300 glucose. 250 to 300 glucose molecules, they are arranged in a straight chain. This is the meaning here. That is what your amylose is. So bache, if it is a straight chain, which linkage should be there? Which linkage should be there? Ultimately it's glycosidic, but what type of glycosidic linkage will be there? 1,4 or 1,6? Madhumita, 1,4 or 1,6? Which linkage should be there? Of course it should be 1,4. 1,4. Now we have amylopectin as well. Okay, we have amylopectin as well. So amylopectin is branched and approximately 30 glucose molecules are present here. So amylopectin is what? It is branched. It is branched. So here see 250 to 300, but here approximately 30 glucose molecules are present. Here glucose monosaccharides are present. Okay, so now you people tell me, so if it is branched, how many bonds should, what type of bonding should be there? Ultimately it's glycosidic, but 1,4 and 1,6 branching. 1,4, 1,6. So wherever there is a branch, 1,6. Wherever there is a branch, 1,6. Wherever there is a branch, 1,6. Everyone repeat it with me. Everyone repeat it with me. Yes, bache, everyone repeat it with me. Right? So just look at this. So in the starch, right, this is individually amylose and amylopectin. Now when you people talk about starch, so starch is made up of both, So sometimes students they get confused कि ma'am we consider starch as homopolysaccharide, then why do you say it is having amylose and amylopectin? बच्चे amylose it is just making 20% of the starch, amylopectin is making 80% of the starch. Ultimately both are made up of glucose, Madhu. Right, both are made up of what? Both are made up of glucose. Am I right? Both are made up of glucose, right? So the starch which is present in potato, that is having 80% amylopectin and the 20% amylose. Any doubt? Any doubt? Now you know that the starch gives blue color. This is important. Blue color with iodine. This is what we say. Starch give blue color with iodine. Why? Because starch is helically coiled. Okay? Sorry, the amylose is helically coiled. So this amylose, because it is helically arranged, so it can trap. Okay, so it can trap iodine molecule. Understood? It can trap iodine molecule. So ultimately this amylose gives blue color with iodine. Right, it gives blue color with iodine, and if you talk about the amylopectin, it gives red color with iodine. So ultimately it is the amylose which can hold the iodine molecule. Understood? Understood? Yes? Yes, bache? Understood? So this is another example of your homopolysaccharide. So see, amylose straight chain, amylopectin mein branches are also there, right? Branches are also there. This is also the example. Amylose straight, amylopectin mein branches are there. Understood? Now this is very important. Please pay attention. This is very important. Please pay attention, everyone. Inulin. It's not insulin. It's inulin. Jaise I still remember after NEET 2020 paper, many students were like, "Your ma'am, do you know what? There is a mistake, right? They mentioned insulin and insulin both, right? There was a question in NEET 2020 paper. You can even check out. So insulin, inulin, both the words were there. Inulin word was there. So many students they were not able to figure out it, right? So please, bache, please, inulin. It's a polymer. It's a polymer of fructose. Many fructose together they make inulin, and you know inulin is also useful. It helps in checking GFR. It helps in checking GFR, that is glomerular filtration rate. You know this inulin is neither stored nor metabolized in our body. Do you know that? It is neither stored nor metabolized in body. Not the part of metabolism. And where do we get it? In the roots of Dahlia. There is a plant, Dahlia, in its roots, from its roots inulin is obtained. Okay, from its roots inulin is obtained. So that's why it is a storage polysaccharide. That's why, what is it? It's a storage polysaccharide. Clear, bache? That is why, what is it? It's a storage polysaccharide. Is that clear? Is that clear? Yes? Understood? Sure? Sure? So you need to remember that. Now, many students, they are confused about the chitin. Bache, what is chitin? Hanji, what is chitin? What is chitin, everyone? Right, it's a favorite question of examiner. What is chitin? Tell me, what is chitin? Quick, everyone. Anyone in the class, what is chitin? Very good. Bache, chitin. It is also the homopolysaccharide. It is also unbranched. Okay, so it's a nitrogen containing polysaccharide, right? I told you na, nitrogen containing polysaccharides are also there. You know that chitin is also known as fungal cellulose. Why? Because it is present in, yes people, why is it the fungal cellulose? Because it is present in, yes, in the cell wall of fungi. Okay, it is present in cell wall of fungi, and moreover it is present in the exoskeleton. It is present in the exoskeleton of insects. You remember, like your cockroach. Okay, so it is made up of same subunit, that is your N-acetyl glucosamine. Because it is N-acetyl glucosamine, it's a kind of derived sugar, right, modified sugar. So students think that it is heteropolysaccharide. No, it's homo. So it is made up of N-acetyl glucosamine. N-acetyl glucosamine. And do you know that it is the second most abundant something? Yes, anyone in the class? Second most abundant. What is that second most abundant? I'm not telling you this thing clearly, but I think you remember. It is the second most abundant what? Second most abundant. Anyone? Just if not first most abundant, second most abundant. Anyone? Nahi. It is the second most abundant. Second most abundant. Organic is the word. Organic molecule on earth. Second most abundant. Okay, so it is the second most abundant organic molecule on earth. Understood? So this is all about the homopolysaccharide. Yes, bache, this is all about the homopolysaccharide. So if you are having any doubt, you are free to ask. Yes, you are free to ask. Yes, understood? So this is all about the polysaccharides. Bache. Now when you talk about the heterosaccharides, na, let's not miss that part also. Wait. I think there is some repetition. Okay, so see here the pectin. You know that pectin is present in the cell wall, and this pectin also fights with the infection. That's what you can keep in your mind. The heparin is what? Heparin is the natural anti-blood coagulant which is present in our body, right? It is the natural anti-blood coagulant that is present in our body. Hyaluronic acid, the cementing material, right? In extracellular matrix it is present. It joins them. In vitreous humor, you know na, eye, aqueous humor, vitreous humor. In vitreous humor this hyaluronic acid is present. Okay. Bache, Siamely, it helps in detecting, it helps in checking the glomerular filtration rate. Okay, it helps in checking the glomerular filtration rate. Understood? Yes, understood? Okay, so that is how you people can, that's it, that's all. Okay, so now let's read this part from the NCERT. Okay, let's read that particular part from the NCERT, the polysaccharide part. So are you people ready? After this we will discuss primary and secondary metabolite, then we will talk about the amino acid and then the proteins. So I think, right, in the NCERT the sequence that is given, it is not proper. I don't know why can't they update it. Obviously there should be the basics of carbohydrate, then polysaccharide, basics of amino acid, then proteins. Why I literally don't know why they have mixed it. Hanji, Sanatosh Chaudhary, we want Wasim sir. Hanji, please call him and study biology from him, please. Then I promise you are not going to get more than 20 marks. Okay. Done. So can we read this part from NCERT? So everyone please open up your NCERT. Yes, please open up your NCERT. है ना? So biomacromolecule, let's read it from NCERT. So what are they saying? There is one feature common to all those compounds found in the acid soluble pool, that their molecular weights, they are talking about acid soluble pool, their molecular weight is in between the range 18 to 800 Dalton approximately. ठीक है बच्चे? It's also a PYQ. Remember that video in which I told you, है ना? Guys, gentle reminder. This, okay. Gentle reminder. अरे यार, why this video is there? रुको. Right. So gentle reminder for what? For that NCERT video. Do check it out. Understood? Do check it out. Clear? Clear, clear, clear. And moreover, I added one more video that how to read the NCERT properly. So on this channel itself, you people can check. So I always mention, bache, whatever statements, right? Whatever statements are like whatever lines in NCERT, they were in PYQs, na? So you should highlight that. So this one was also there. It was asked as a question in the previous year paper. Okay, so acid insoluble fraction mein what do you have? Proteins, nucleic acid, polysaccharides and the lipids. Proteins, nucleic acid, polysaccharides and the lipids. So these classes of compound, with the exception of lipids, have molecular weight in the range of 10,000 Dalton. So if our NCERT is saying 10,000 Dalton, we have to pick up the 10,000 Dalton and above. Understood? We have to pick up the 10,000 Dalton and above. In some books 10,000 Dalton is given, in some book above 1,000 Dalton is considered as the biomacromolecule. That's what you people need to remember. So for this very reason, biomolecules, the chemical compounds found in living organisms, they are of two types. Okay? One, having molecular weight less than 1,000 Dalton, they are biomicromolecule, right bache, and they are in the acid. Right, see this, what are they writing? One, those which molecular weight is less than 1000 Dalton, they are usually referred to as biomicromolecules, right bache, and while those which are found in acid soluble fraction, they are the biomacromolecules, and you already know about it. So here in NCERT, in this part, they have explained that why lipid is an exception. We have already discussed it. Okay, we have already discussed it. So molecules in the insoluble fraction, with the exception of lipids, are polymeric. Why? Because lipids, their molecular weight is less than 800 Dalton. Their molecular weight cannot exceed above 800 Dalton. That's what you need to keep in your mind. Okay, then why are they in acid insoluble fraction, bache? Because lipid, they are very small molecular weight compounds, right? But they are arranged into structures like cell membrane and other membranes. So when we grind a tissue, we disrupt cell structure, cell membrane and other membranes. They are broken into pieces, but they form what? They form vesicles. Clear? They form vesicles, and these vesicles are not water soluble. Are you getting my point? These vesicles are not water soluble. So therefore these membrane fragments in the form of vesicles, they get separated, but they remain in acid insoluble pool. So lipids are not strictly biomacromolecule. I have repeated it, and even I explained it from the NCERT itself. So if you guys are having any doubt, please feel free to ask. So now from the NCERT, that's what we have to pick, that is your polysaccharide. And trust me, after all that explanation, now you are going to love this part. You are going to love this part. If there is any doubt na Madhumita, tell me. If there is any doubt na Madhumita, tell me. So acid insoluble palette, okay, acid insoluble palette, they also has polysaccharide, of course, because their molecular weight is above 10,000 Dalton. So polysaccharides are long chains of sugars. They are the threads, right, containing different monosaccharides as building block. So means the polysaccharides, they are made up of monosaccharides. Polysaccharides, they are made up of monosaccharides, right? So cellulose is a polymeric polysaccharide which is having only one type of monosaccharide, that is glucose. So cellulose example is given here. So you should remember that which linkage is there? Beta 1,4. Why? Because it is having the beta glucose. Understood? Understood. And cellulose is what? Unbranched. It's straight chain, right? So it's unbranched. Understood? So again, see, it is written now. So then starch is a variant. Starch is the storehouse of energy in plant tissue. So animals have another variant called glycogen, right? Even inulin example is given here. It's a polymer of fructose. It's a polymer of fructose. ठीक है बच्चे? So in a polysaccharide chain, जैसे कि glycogen, right end is the reducing end, I told you, and left end is the non-reducing end. Any doubt? Right end is the reducing end, left end is the non-reducing end. So this is MCQ. MCQ. Clear bache? So starch is going to form helical secondary structure, right? Cellulose do not form. See, cellulose do not form helices, so it is not going to capture the iodine, but starch will. Okay, starch will, that's all. So plant cell walls are made up of cellulose. Paper made from plant pulp and cotton fiber is also cellulose, right bache? So there are more complex polysaccharides also in nature, like your amino sugars, which are chemically modified sugar. So this glucosamine, this N-acetyl galactosamine, they are what? They are modified sugars, and that modified sugars are making your homopolysaccharides also and heteropolysaccharides also. Understood? Just say your chitin is having what? Chitin is having the NAG. Understood? So any doubt? Any doubt? So that is what we have covered. So this is all about the polysaccharide from NCERT. This is all about the carbohydrates from NCERT. So now the next topic that we are going to discuss right now is your primary and secondary metabolite, and then we will be talking about the amino acids. Right, students? So if there is any doubt, do let me know. You will get the PDF of this particular lecture, so there is nothing to worry about. So you can just mention the titles in your notebook. Okay, Priya, you can just mention the titles in your notebook. Is that clear? Is that clear? Sure? Is that clear? Okay, so what is completed? Chemical analysis done, elemental analysis done, carbohydrates done. In carbohydrates, monosaccharide done, oligosaccharide done, polysaccharide done. Now what are we going to discuss, Madhu? What are we going to discuss? Primary and secondary metabolite. That's what I told you initially. Primary and secondary. We in general know that na, primary, secondary. Primary which will be prioritized more, secondary which will be prioritized less. In general, that's how we discuss. But what's the word here? The word here is metabolite. What is the word here? The word here is metabolite. What's the meaning of metabolite? What is the meaning of metabolite? We have already discussed reducing and non-reducing part. I think it's Zelathauli. We have already discussed the reducing and non-reducing part, bacha. Okay, metabolite. Now you know that in our body chemical reactions are going on. है ना? Chemical reactions are going on. You know that here in this chapter, right, in this chapter biomolecule itself, now it is written कि living means, the synonym of living is metabolism. Okay, the synonym of being living is metabolism, the chemical reactions which are going on in our body. Right, the synonym of living is what? Metabolism means the chemical reactions which are going on in our body. ठीक है बच्चे, which are going on in our body. If chemical reactions are going on in our body, means we are living. Right, if someone is dead, there will be no metabolism, there will be no chemical reactions. These chemical reactions are keeping us alive. Okay, so metabolism and living, it is considered as synonym. Synonym means having same meaning. Understood? So in this metabolism, in these chemical reactions, in between you have the intermediates. Like for an example, Madhu, if I have to give you the example, Priti bache, you are a new student, quickly subscribe the channel. Let's make it 100K family ASAP. Okay, people? Chalo. So now what am I saying? Metabolism is going on. Chemical reactions are going on. Let's take the example of glucose. Glycolysis. Glucose 6-phosphate. Glucose 1,6, bisphosphate, fructose 1,6, bisphosphate. Is it like this or what is it? Is it glucose 6-phosphate and fructose 6-phosphate, fructose 1,6 bisphosphate, and then there will be the cleavage. Do you remember? Glucose 6-phosphate, fructose 6-phosphate, fructose 1,6 bisphosphate, then there will be the cleavage, that is breakdown. Right, bache? And we have dihydroxyacetone phosphate, we have glyceraldehyde 3-phosphate. So this is the glycolysis. So in glycolysis there are steps. Hope you remember there are 10 steps in glycolysis. So in between these chemicals they form. Right, in between these chemicals they form. What are they? They are metabolites. The chemical reactions which are going on in our body, the compounds that are taking participation, what are they? They are the metabolites there. They are the metabolites. That is also, you have to remember this. But now here, what is the meaning? See, the most exciting aspect, okay, the most exciting aspect of chemistry deals with isolating thousands of compounds, small and big, from living organism, and to check their structure and the possible synthesis, right? Synthesis. Clear बच्चे? So if one were to make a list of biomolecules, such a list would have thousands of organic compounds. Obviously, we know that. And these biomolecules are known as metabolites. These biomolecules, we can call these biomolecules as metabolites. MCQ. Mark it. MCQ. In animal tissues, one notices the presence of all such categories of compound given in figure 9.1. This is right. This is not 9. This is 9.3 now. Where is 9.1? I'll show you 9.1 also. 9.1 mein that all that carbohydrate, protein and all. This is what they are talking. So now they are saying कि they are called primary metabolites. What are primary metabolites? Like your carbohydrates, like your proteins, which are directly taking part in the metabolism, which are having direct results, direct contribution. You can directly check their role. So that is primary, main thing. Secondary will be which are having indirect involvement, not the direct one, in simple words. Not the direct one, in simple words. So the one, right, the compounds, the biomolecules which are directly taking participation in this metabolism, they are the primary metabolites, right? And the one which are indirectly involved, they are secondary. So primary metabolites will be your proteins, your carbohydrates, right, etc. And now when you are talking about the secondary, let's read it, you will understand it. Ki however one analyze plant, fungal and microbial cells, we see thousands of compound other than primary metabolite. So basically in animals, mainly we discuss about the primary metabolite. Animals are not going to give you any specific type of, compounds. Some animals can. Some animals have therapeutic properties. But see, in the case of plants, you know that from fungus, we are getting so many chemicals. We are getting the drugs from them, right? From the plants, we are also getting the oils. We are also getting the toxins. We are also getting the lectins. We are getting so many things from the plants. So when we check plants, microbes, the fungal cells, we are getting so many secondary metabolites. They are the result of metabolism. They are not directly, they don't have direct impact on metabolism. They are the result of that metabolism. They are helpful for humans, okay? They are not directly involved in the growth process of that particular organism. This is what you have to understand. But in the case of animals, from the animals, no doubt there are certain animals, there are some mollusca which are having therapeutic properties, which are having medicinal uses. We can extract that compounds from them. But in general, we see the list is more in the case of these fungus, the plants and the microbes. This is what you have to understand. Clear बच्चे? जैसे alkaloids, flavonoids, rubber, essential oil, antibiotics, okay, the colored pigment, they all are the secondary metabolites. So बच्चे, this table is very important. This table is very important. Here it is clearly mentioned. Everyone please highlight, please pay attention. Everyone please highlight that while primary metabolites have identifiable function, play known roles in normal physiological processes. What are they saying? While primary metabolites, they have identifiable function. We can identify the function of that primary metabolites. They have role, they have importance in that normal physiological processes. Right, bache? The role of secondary metabolite in host organism is not so known at this moment. Okay? So we do not at the moment understand the role or function of the secondary metabolites. Clear bache? Clear bache? So many of them are useful for human welfare. Here you have the list. So these secondary metabolites are having ecological importance. So one is having direct role, one is having indirect role. That's what you have to remember. So look at this. Alkaloids mein, example morphine. Chalo, write down your, right, morphine. You know na, morphine, it is analgesic, means pain reliever. Yes, it is an analgesic, means pain reliever. Yes or no? Morphine is what? Analgesic, means it is a pain reliever. Are you getting my point? It's a pain reliever. Clear bache? Clear bache? Yes. What is it? It's a pain reliever. So it is an alkaloid. So yes, you have to remember this. Clear bache? You have to remember this. Clear bache? Yes, then comes the codeine. Do you know what is codeine? Do you know what is codeine? Yes, this codeine is also an alkaloid. It is also bache, a pain reliever. It is also an analgesic. It's a pro-drug of morphine. Okay, pro-drug of morphine, right? So these are the examples of alkaloid. Then comes the terpenoids. Terpenoids are very easy to figure out. Mono, single terpenes, diterpenes. So another homework. These terpenes are used for the formation of your one of the plant growth regulators, and that's what you need to answer. That's what you need to answer. Okay, these terpenes, they are used for making your plant growth regulators, and that's what you have to answer. Now move to the next part, the essential oils. Lemongrass oil, easy. Toxins mein, it's a repeated MCQ, people, right? Heparin, ricin, they are the toxins. So what is your homework? In the NCERT itself, you will write down that heparin and ricin, they are obtained from. Now bache, lectins. First of all, tell me what are lectins. Just here you have one example now, concanavalin. This is also MCQ, right? This is also MCQ, concanavalin, lectins. So can you tell me what are lectins? Anyone? But lectins, they are the proteins that are attached to the carbohydrates. They are having different roles. Just in our system, they are in the stomach part, we have gastric mast cells. So these lectins, they promote the release of that mast cells. The content of that mast cell will be secreted. It is having different role when you talk about the lectins. Lectins are present in the legumes. From the legumes also, if we had that legumes like your soya bean, like your pea, they have these lectins, right? There are some pulses where lectins are present. Pulses while I have to check, but in legumes they are definitely present. So these are the proteins which are attached to the carbohydrates. Okay, so I gave you one function of these lectins. Okay, that in our system, in gastric mast cell, they help in their stimulation, right? Their content will be released. So you will tell me about their another role. That's your homework. And yes, Madhumita is right, very good bacha. Akshita is right, very good bacha. Terpenes are used for the formation of gibberellic acid. Excellent bache, very good. Okay, very good. So now drugs, vinblastine, curcumin. Curcumin is obviously obtained from turmeric. Tell me, is it obtained from turmeric? Tell me. And what about this vinblastine? Vinblastine, it is anti-cancerous, students, it is anti-cancerous. It is obtained from your Vinca rosea. You all have seen this plant, Vinca rosea. You can just Google it right now, Vinca rosea. Okay, Vinca rosea. Okay, so Catharinthus rosea. So from this it is obtained and it is anti-cancerous. Clear, bache? Clear? Then comes the polymeric substance like rubber, gums, cellulose, like this. Okay, so these examples are important. Okay, people, these examples are important. Is that clear? Yes. Bache, I have already Priyadarshini explained about the reducing sugars. Okay, so this is primary and the secondary metabolite. So if you people are having any doubt, you are free to tell me. Yes, you're free to tell me, because after it we'll be starting the amino acids. Okay, after it we will be starting the amino acids. So see, look at this, we started the general discussion. Let's have a quick revision. So we started the general discussion, bache. So we talked about the basics of the biomolecules, the chemical elemental analysis, primary secondary metabolites, and the carbohydrate. It's done. Okay, it's done. So all carbon compounds are biomolecules. However, the inorganic compounds which are also there in the cell, right, they are also there in the cell. That's what we have done, we have discussed. Okay, bache, carbon, hydrogen, oxygen, nitrogen part, this basic introduction part is done. Okay, in chemical analysis, this is the quick revision of that chemical analysis. Wait, see, I have explained each and everything to you. Okay, so when it comes to the, see this table, just read it quickly, revise it quickly. Yes, people, just read it quickly, revise it quickly. Quick, read it and revise it quickly. Yes, everyone, quick. Yes, is it clear? Sure? Are you sure? So that is how you can use these flow charts for the quick revision. Okay, then here you have the inorganic compound analysis, basically elemental analysis. That is what we have covered. So now the next point that we are going to discuss is amino acid. I know people, when you will get the PDF now, just say in between initial part, then carbohydrates all together in between, right? I am going to discuss it. Okay, so don't get confused. If, like, let's say in the PDF it will also be mixed, but still in one go I'll explain one topic. Okay, I was about to jumble the NCERT as well. I was about to I was changing it, right? Firstly I was keeping the polysaccharide part, then I was keeping that amino acid part. But the point is, if I will not keep NCERT in a flow, then you people will get confused and you will be more confused, because you do not you do not put efforts to check that thing. You get that anxiety, yeah, what is this, why this screenshot is here, why that screenshot is there, is there any new change in the NCERT, right? So you start behaving like this. Okay, you start behaving like this. Take it. Okay, so Arjun Bache, I think I have answered all the questions that I am getting. And Priyadarshini, as I said, I have already explained reducing sugar, the one which can reduce cupric to cuprous. The one reducing sugars are the one which are having free aldehyde or ketone groups. They can reduce cupric to cuprous. ठीक है जी. Heroin, what do you know, what do you want to know about this heroin? It is formed by the acetylation of morphine, right? It is formed by the acetylation of morphine. So now you tell me, where are you going to put it? Is it something natural or artificially you are making it? On that basis you can get the answer of your question. Okay, you can get the answer of your question. Okay, so the next point that we need to discuss is amino acid. What's that? Amino acid. Why amino acid, bacha? Because amino acids are going to make the proteins. Okay, so amino acids, where are they going to be, where are they present? They are present in the acid soluble fraction. Where are they present? They are present in the acid soluble fraction. Is that clear? Is that clear? Yes, they are present in the acid soluble fraction. What are they? They are the amino acid. So tell me people, what do you know about the amino acid? Yes, it's a very interesting topic. It's a very interesting topic. So tell me, what do you know about these lovely amino acids? Yes, and don't start with this line, that they are the building blocks of protein. Right? They are the building blocks of proteins. So other than this line, now please tell me anything else that you know about the proteins other than this line, that they are the building blocks of protein. Anything else? Anything else? Yep. Anything else? Amino acid topic is a very important topic for your NEET syllabus, bacha. It's a very important topic for your NEET syllabus. Yes, so they are the, yes, it is for the grade 11th, it is for the grade 12th, it is for the grade droppers as well. Building blocks of amino acid. Other than that, dekho bacha, see, no doubt in your syllabus you talk about the 20 amino acid, right? This is what we discuss, 20 amino acid, hai na? Right? Yes or no? Tell me, this is what we discuss na, 20 amino acid. Tell me. Hanji. But bache, it is not just the 20 amino acids that are present. Okay, let me tell you, these 20 amino acids are considered as the magic numbers. What are these 20 amino acids are considered? These 20 amino acids are considered as the magic amino acids because they are used for the protein formation. Okay, but that doesn't mean we just have the 20 amino acid. Right now, even in that 20 amino acid wala list, to other amino acids are also added. Let's not go in the depth of that, but still people, I'm just telling you this thing. So now what am I saying? Yeah, so now what am I saying? That nearly 200 to 300, right? Nearly 200 to 300 amino acids are present, but out of that 20 amino acids are the one that will make protein in our body. Okay, bache? And now when you talk about their structure, amino acids, they are having amino group as well. they are having that amino group as well, they are having the carboxylic group as well. So now we have to discuss their structure. So any doubt here? Any doubt here? So they are the building blocks, right? They are the building blocks. So nearly 200 to 300 amino acids are present, but 20 amino acids are, for amino acid I will use short form AA, 20 amino acids are used for protein formation. Okay, so guys, if you are a dropper, then I know you have completed molecular basis of inheritance, so you clear bache. So when you talk about the amino acids, mainly they are made up of what? Amino acids are having carbon, they are having hydrogen, they are having oxygen, yes, they are having nitrogen, but some are also containing, some are also having sulfur. Do you know that? Some are also having sulfur. So I am writing the example here. I am writing the example here. It is very, 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 very important example. That is your cysteine and methionine. Bache, that's a previous year question. That is cysteine and methionine. What is it? Cysteine and methionine. They are, cysteine methionine is a sulfur containing amino acid, right? Cysteine methionine is a sulfur containing amino acid. Is that clear? Cysteine methionine is a sulfur containing amino acid. That is the trick here. Are you getting my point? That is the trick here. Understood? So sulfur containing amino acids are there. So now let's discuss about their basic structure. Okay, let's discuss about their basic structure. Okay bache, and then we will continue further. Okay. Okay, let's discuss about the basic structure of amino acid. So do you know about the basic structure of amino acid? Do you know about the basic structure of amino acid, people? Please tell me. Do you know about the basic structure of amino acid? Anyone? Anyone? It's very simple, bache. Obviously there will be a carbon. Carbon is tetravalent. Okay, carbon is tetravalent. Okay, so here it is having NH2 group, it is having C double bond OH group, means acidic group is also there, basic group is also there. To one carbon bache, hydrogen is attached. To another carbon, the hydrogen, or R means alkyl group. Anything can get example. Okay, everyone in the class, listen to me carefully. One very simple example of amino acid. Okay, so C, one side is having NH2, one side is having COH group, one side is having hydrogen. If another side is also having hydrogen, if another side is also having hydrogen, guys, am I audible, visible? Yes, am I audible, visible? Am I audible, visible? Please let me know. Students, am I audible, visible? Now okay na? Now okay na? So let's continue. But I think there is no lag. Internet is working fine. I don't know what's the issue. Is it still lagging? I don't think so. It's okay na? Chalo, let's continue. So look at this part. So this is the basic structure of amino acid, right? And here, when you see this part, when you see this amino acid, it is glycine. It is glycine. It is the simplest amino acid. It is the simplest amino acid. Guys, just look at the structure. Definitely question will come from this part. So from biomolecules, it's sure that one question will come from, right? That one question will come from the amino acid. For sure, you are going to get one question from amino acid. So this is the basic structure of amino acid. And see, look at this basic structure. So amino group is as such, carboxylic group is as such, hydrogen is as such, and here, right, instead of alkyl group, hydrogen is present. This is the glycine. This is the simplest type of amino acid. One more example I'll give you. One more example I'm going to give you, and I'm pretty sure then you can compare the structures as well. So now at the place of R, what do we have? Now at the place of R, what do we have? At the place of R, what do we have? Please tell me quickly. Now at the place of R, what do we have? Bache, CH3 na? CH3, methyl group is present. Here R means CH3 methyl group. So this amino acid is the alanine. This amino acid is the alanine. Are you getting it? This amino acid is the alanine. Clear? One more example. So here the basic structure, here the other structure. So one more example, people, one more example. So NH2, COH, H, and here if it is CH2OH. So what's the alkyl group here? What's the alkyl group here? What is the alkyl group here? It's the CH2OH. What is it? It's CH2OH. Yes, this is serine. This amino acid is serine. So it is given in NCERT. So they can ask you the question from these structures. Okay, they can ask you the question from these structures. This is important. Clear, bache? This is important. Okay. Sure? Sure? Right? So that's the basic structure. That's the basic structure. Now, see, bache, when you talk about the amino acid, na, we say that this carbon, first of all, understand it, then we have to read everything from NCERT. So when you people talk about this carbon, na, this carbon is known as alpha carbon. We call it as alpha carbon. And these amino acids are known as alpha amino acids. Do you know why? Do you know why? Yes? Do you know why? Yes or no? Yes or no? These amino acids are known as alpha amino acids, bache, and this carbon is the alpha carbon. Which carbon it is? It is the alpha carbon. Do you know why? Do you know why? Let me tell you. See, when you talk about the structure of amino acid, I hope you all have seen that amino acids are considered as substituted methanes. What do we consider them? We consider them as substituted methanes. What is the meaning of substituted methane? Right, methane means CH4. That is methane. And all the sides are having 4 hydrogen, CH4. So from that methane, now see, the 4 sides, what that 4 sides are containing. One side is having amino group, one side is having COH, one side is having hydrogen, one side is having alkyl group or the hydrogen. So obviously methane is substituted. Methane is substituted, and that's what we get. That's what we get. Okay? So on what basis you are going to say that this carbon is the alpha carbon? If bache, it's all the 4 sides, right? If this carbon is asymmetrical, right? If this carbon is asymmetrical means if all the 4 sides are having different group, right? If all the 4 sides are having different group, you can see here, if all the 4 sides are having different group, then this will be your alpha carbon, because it is the asymmetrical carbon, right? It doesn't have the same functional group, right? So it is the asymmetrical carbon. Understood? Understood? It is the asymmetrical carbon. Is that clear? Is that clear? Yes. So alpha carbon is the asymmetric. There is only one exception. There is only one exception, and that exception is your glycine, right? Glycine is not having alpha carbon. So this can be asked as a question, people. This can be asked as a question, okay? This can be asked as a question. Understood? Understood? So glycine, it is because why is it so? I have already explained you the structure, right? I have already explained you the structure. Look at the glycine. So this side is having hydrogen, this side is also having the hydrogen, right? So all the 4 sides are not having the same functional group. That is why. Understood? Understood? So glycine is not having the asymmetrical carbon, all others are having. So amino acids are also considered as the substituted methane. So ultimately, bache, there will be the change of this R. Okay, there will be the change of this R, and that is going to change the property of the amino acid. Now everyone, right, very good. Obviously it's not chiral. So everyone here, right, now I am going to write down the amino acids. So you are going to note it down. Okay, you are going to note it down. Clear, bache? Clear? Okay, and do you know most commonly occurring amino acids? This is what you can write down. Or you can say that naturally the amino acids that exist, natural amino acid, they have L configuration. Do you know the Hemant, bache? Do you know the meaning of L configuration. जैसे naturally occurring amino acids ना, they have L configuration. Do you know that? Naturally occurring amino acids have L configuration. Anyone in the class who can explain this line to me? Anyone in the class who can explain this line to me? It means NH2 is written. It means NH2 is written on left hand side. Okay, it is written on left hand side. Understood? It is written on left hand side. This is the meaning of the L configuration. Okay, we have to write down this NH group on the left side. Understood? On the left side. So this is the R group, which is going to decide that which is going to make the 20 different types of amino acids. So everyone, are you going to repeat that amino acids with me? Okay, I challenged many students many times, so I think this time it's your turn. Yes people, this time it's your turn. So let's start. Okay, let's start. So are you ready to remember the name of 20 amino acids? Are you ready to remember the name of 20 amino acids? Tell me, are you ready to remember the name of 20 amino acids? Everyone please write down. Yes everyone. Is anything missing? Is anything missing? Yes? Tell me, is anything missing? Quickly tell me. And note down, please take the screenshot as well. It is little bit related to your, yes, genetic code as well. It is related to your genetic code as well, isn't it? So alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine. Oh, glycine is missing. Glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, PRSTUV, right? So let me check. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Now it's 20. Glycine was missed, right? So bache, I remember them in the alphabetical order. Okay? In the alphabetical order, right? So you can repeat it with me. Alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, serine, threonine, tyrosine, tryptophan, and the vinine. So these are the 20 amino acids. So they are used for the formation of the protein, right bache? So 21st and 22nd amino acid is there, but that is not the part of your syllabus. So it is selenocysteine, and pyrrolysine. Okay, it is selenocysteine and pyrrolysine. So these are that 21st and 22 amino acid. You should know the names. Okay, right? So I challenge you, if you can remember. Wait here, let me array. Okay, so just come to our lovely channel. Okay, so here you can see the important update about the NCERT change. You people can see, and here we have the trick for the genetic code. This video. You can check. Here there will be the trick for the genetic code. In less than 10 minutes, you can remember the genetic code. But before that, you have to remember this part. Okay, so alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, serine, tyrosine, tryptophan, threonine, and then we have the valine. These are the 20 amino acids, right? These are the 20 amino acids, right, bache? Right, bache? So 21st and 22nd is also there. So these are the 20. So see, dekho, in your syllabus, what do you have? You should know about the name of acidic amino acid. You should know about the name of basic amino acids. You should know about the name of aromatic amino acids. You should know about the names of neutral amino acid. So if you have to remember these examples, why can't you remember all the 20 amino acids? Why can't you remember all the 20 amino acids? What say people? है ना? Why can't you remember all the 20 amino acids? Right? So out of it, I will tell you that, you know, what you have to retain and what not. And now see, they are also having the symbols. Just we also write now this alanine, ALA, right? Arginine, ARG, right? This is how we write it. Yes or no? This is how we write it. Alanine, arginine, asparagine, aspartic acid. So I'm not going to write it. It is in your NCERT genetic code wala part. You can revise. So they are also having one letter code. Just alanine A, arginine R, asparagine N, aspartic acid D, cysteamine C, glutamine it is. One is glutamic acid. Then it is okay, aspartic acid. Okay. So in between even I forgot. I'm not going to lie. I have the tricks here. Okay. In between even these alphabetical codes, na, even I forgot. Okay. I get confused here. Just say glutamic acid is D, I guess. Glutamic acid is D or E. Okay. So that's how we have to remember. So it is having one letter, right? It is having one letter codes as well. One letter codes as well. Okay. Okay. So that's what you need to remember. Understood? That's what you need to remember. Understood? Okay, so alanine, arginine, aspartic acid, right? Aspartic acid, and then we have the cysteine, bache. Then we have the glutamine and the glutamic acid, right? Glutamic acid. So this is what you need to remember. जैसे ये देखो tryptophan के लिए it's simple W, right? Proline के लिए also simple P, M. Glutamine, G, glutamic acid, E. Okay, so here even I got confused. I'm not going to lie. When it comes to the one letter symbols, right? One letter words, even I got confused. So this is something if you want to check, you can. Otherwise, question will not come. But yeah, you should know about this, that ALA is alanine, ARG is arginine, right bache? Right bache? So question like this should be clear in your mind. Just ASN. Many times students, they make the mistake here. It is there in genetic code. It is linked to genetic code. That's why I'm teaching you. Asparagine, ARG, arginine, asparagine, arginine, ARG, asparagine is ASN, aspartic acid means ASP. Okay, ASP. So that's what you need to do. Fine. That's what you need to do. Take care. Take care. Thank you, HSP sir. HSP sir, tell me the symbols here. I'm pretty sure that you remember all of them. I'm pretty sure you remember all of them. I'm quite sure about it. Hannah, HSP sir? No, I remember the name. I'm talking about the symbol, sir. And I'm pretty sure you remember all of them. And this one, bache, I challenge your Wasim sir. Even your Wasim sir, he doesn't remember the name of all the 20 amino acids. I'm pretty sure about it. I'm pretty sure about it. Even he doesn't remember the name of all the 20 amino acids. But yes, right? It's sir, HSP sir, I'm sure. Yeah, Hannah, you have the tricks. Even I do have. But sometimes I remember the name of the amino acid. I don't remember the symbol. So SSP sir, dekho, alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, serine, tyrosine, threonine, tryptophan, and the valine. Okay, this much I remember. But sometimes even I forget that symbols. Okay, I'm not going to lie here. Okay, bache. So whatever is the part of your syllabus, I already told you. This is something that you should know because it is linked to your genetic code. Fine, it is linked to your genetic code. Now, as per our syllabus, what we need to remember, let me tell you. Right. First of all, sulfur containing amino acid. So we know sulfur containing amino acid. We have the trick for that, CMS, Chief Minister sir. So C means cysteine, M means methionine, right? C means cysteine, M means methionine, or S means sulfur, right? S means sulfur. So cysteine, methionine, sulfur containing amino acid, right, Chief Minister sir. Now bache, that is again a repeated question. Acidic amino acids, right? Acidic amino acids. So bache, if it is acidic means it is negatively charged. Of course it is negatively charged, yes or no? Yes or no? So in that list if you remember, aspartic acid, glutamic acid, simple. Okay, aspartic acid, glutamic acid, aspartic acid, glutamic acid. These are the symbols. That is what we have, right? So they are the acidic amino acid. And do you know why do we call them as acidic amino acid? Why do we call them as acidic amino acid? Wait, we will discuss the structure as well. But before that, let me tell you basic amino acids as well. Because that is, bache, all of them are MCQs. Basic amino acids mein we have the trick. The trick is Hall. We have the trick is Hall. What is the trick, Chetu? Hall, H-A-L. So H means histidine. H means histidine. A means arginine. A means arginine. L means lysine. So these three are basic amino acids, right? So if it is acidic means it is having two COH group. If it is basic means it is positively charged. Yes, bache, what type of amino acids are they? They are positively charged. And here what do you have, people? Here what do you have? You know that there will be two amino groups, right? I will draw the structures, don't worry. There will be two amino groups, right? So it's not just lysine and arginine that are the basic amino acid. Even your histidine is also the basic amino acid. Understood? Even your histidine is also the basic amino acid. That is what you people should know. Okay? That is what you people should know. Clear, bache? Even your histidine is what? Even your histidine is what? Histidine is also your, yes, bache, basic amino acid. So these examples are important. And what are there in the histone proteins? Lysine, arginine. So it is a previous year question. It has been asked in 2020 and even it was in 2022. It was a repeat. It was repeated in 2018. I don't remember the year, but yeah, this lysine wala question was there in 2020, and even after a year or two, it was again repeated. It was again repeated. It is the most repeated question, the basic amino acids. It's the most repeated question, the basic amino acids. Clear, bache? Clear, bache? So one amino group, right? So what are the acidic amino acids? One amino group, two carboxylic groups, and here two amino groups and one carboxylic group. Okay, because it is basic. So obviously NH2 group is the basic here. NH2 group is the basic here. So that's what we need to remember. So any doubt? Yes, people, any doubt? Just I'll give you the example of. Yes, I will give you the example of your, what we call it, glutamic acid. Okay, I'll give you the example of your glutamic acid. See, that's the basic structure. H on one side, NH2, COH. So in glutamic acid, you will have extra CH2. This is that alkyl group. CH2, and what should be there? Yes, people, what should be there? Which group should be there? Tell me, CH2, which group should be there? Of course, your carboxylic acid, right? So how many carboxylic acids are there? 1, 2. How many amino groups are there? 1. So this is the glutamic acid denoted by D. Okay, denoted by D. So 2 carboxylic groups are present. Understood? 2 carboxylic groups are present. Understood? Same way I'll give you the example of, I will give you the example of basic amino acid, right? So in the basic amino acid, what I just said, people, what are you going to get in basic amino acid? Tell me, what are you going to get in basic amino acid? You are going to get 2 NH2 group. Take it? You are going to get 2 NH2 group. Is that clear? Is that clear? Is that clear? Sure? Sure? So your homework is at least try to remember it, okay? At least try to remember it. Fine. At least try to remember it. Uh-oh. Chalo, so one structure is of lysine. Lysine is also examiner's favorite question. H, NH2, COH. So it was CH2, 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 C. So that's your lysine. That's your lysine. Okay. That's your lysine. Basic amino acid. Positively charged. Understood? Positively charged. Clear, bache? So any doubt here? Any doubt here? Yes. So now PTT, right? Your PTT. Okay. PTT means, I'm writing it here. Your phenylalanine, your tryptophan, your phenylalanine, your tryptophan. One more is there. Can you tell me about that? Phenylalanine, tryptophan, and one more is there. I'm pretty sure you all are aware of it. Phenylalanine, tryptophan, one more is there. Of course, it is your tyrosine. It is your tyrosine. These are the aromatic amino acid. But everything is given in NCERT, right? What is the meaning of being aromatic amino acid? They are having a ring in their structure. They are having a ring in their structure. So I hope you remember that tryptophan is used for the formation of which plant growth regulator? Tryptophan is used for the formation of which plant growth regulator? Tryptophan is used for the formation of which plant growth regulator? I know you know that. I'm talking about the tryptophan. Yes. I'm talking about the tryptophan. It is also a previous year question, people. It is also a previous year question. The tryptophan. Tryptophan. It is denoted by W. Okay. Right. TRP is the short form. So it is important for the synthesis of IAA. Right. That is your indole acetic acid. Indole 3-acetic acid. That is your oxin. How can you forget? That is your oxin. Right, and tryptophan is the most complex type of amino acid. What amino acid it is? It's most complex type of, what type of amino acid it is? It is the most complex type of amino acid. Understood? Most complex type of amino acid. Okay. So tyrosine, can you tell me about the tyrosine? Do you remember tyrosine is used for the formation of skin pigment melanin? It is needed for the formation of skin pigment melanin, right? Do you remember it from genetics, bache? Do you remember it from genetics? If there is no phenylalanine hydroxylase, phenylalanine will not be converted to tyrosine and the melanin will not form, right? So it is very important for the formation of, skin pigment melanin. Clear, bache? Skin pigment melanin. Understood? Understood? Sure? Sure. So tryptophan is the most complex type of amino acid, and which one is the simplest? You already know, that is your glycine. So all the amino acids, they are levorotatory except your glycine, right? All the amino acids are levorotatory except your glycine. Understood? Understood? So leaving this part, okay? Just here you have acidic amino acid 2, basic amino acid 3, total 5. So leaving these 5, all the 15 amino acids are the neutral amino acids. What type of amino acids they are, bache? What type of amino acids they are? They are the neutral amino acids. Yes, bache. What type of amino acids they are? Yes. What type of amino acids they are? Anyone in the class? They are neutral amino acids. They are neutral amino acids. They are neutral amino acids. Clear, bache? Clear? So 2 extra, I told you already, right? Not the part of your NCERT, but yes, this is should be, you should know about it. It is for your general knowledge, the selenocysteine and the pyrrolysine. Chalo. So can I ask you one question? If you are in class 11th, nothing to worry about. You will learn it in class 12th. So can I ask you one question, students? So how many of you are droppers? Or I can say that dreamers, right? That will be a better word, dreamers. How many of you are dreamers? So tell me which one is the initiator codon. Tell me which one is the initiator codon. Yes, people. Tell me which one is the initiator codon. Very simple question. Which one is the initiator codon? Obviously methionine. Obviously methionine. Okay. So which amino acids are having only one codon? Which amino acids are having only one codon that is going to code them? Which amino acids are having only one codon which is going to code them? Yep. Tell me. Tell me quick. Which amino acids are having only one codon that is going to code it? But obviously methionine and the tryptophan. So I'll say, do check this video, bache, do check this video, right? So genetic code in 10 minutes. Trust me, you'll learn a lot from this video, okay? Okay, I have included many points there. Fine. So this table clear? Okay, done. So can we move to the next part, that is amphoteric nature of the amino acid? Okay, so the next part is saying what? The amphoteric nature of the amino acid. But yeah, before starting the amphoteric nature of the amino acid, let's talk about the essential and the non-essential amino acid. So these examples clear? Tryptophan, the role of tyrosine, okay, bache, the sulfur containing amino acid, the acidic one, the basic one, okay, the basic one, and the structures here. Okay, so next is essential and non-essential amino acids. Next is what? Essential amino acids and non-essential amino acids. Essential amino acids and non-essential amino acids. Okay, bache, essential means something which is very important for our body. We need that. Okay, what do we have? Essential amino acids and the non-essential amino acids. Essential, which is important for the body, okay, which we need in the body. So it is not synthesized by the body, so we have to take it externally. Clear? We have to take it externally. So essential amino acids, I'm mentioning it again, they are not synthesized by the body. Not synthesized by the body, but so they should be the part of our diet. In simple words I'm writing, so they should be the part of our diet because body needs it. They are the essential. So now do I need to mention the non-essential amino acid? Right? Do I need to mention the non-essential amino acid? Of course not. So what should be the trick here? Private Tim Hall. Right? This is the most repeated trick. It's private Tim Hall. It's private Tim Hall. I'm telling you, bache, there are, see, so all the things that we have discussed, they are important. Okay? So that is the reason now. I remember I remembered all the names of 20 amino acid because I know. Okay, right, because I know ultimately in examples itself I have to revise it. That's why. So what is the trick here? The trick here is private Tim Hall. So obviously, जैसे from P we have proline, from P we have phenylalanine. So of course we can get confused, है ना? From this T, tyrosine is there, threonine is there, tryptophan is there. So we have 2 T here, 1 is missing. So obviously we can get confused. So let's start it in this way. L for lysine, L for leucine. That's what we have. Then comes bache your H. H means histidine. H means histidine. 1A here is telling us arginine. This A is for arginine. Clear? Clear? 1M is here, that is telling us about methionine. What is it? It is methionine. 1I is telling us about the isoleucine. Are you getting my point? right? V here means valine. V here means valine. So what do you think, proline or phenylalanine? Which should be the essential amino acid? Which should be the essential amino acid? Is it the phenylalanine? Is it the phenylalanine, or is it the proline? Very simple it is. What should be essential? What should be required in our, what is required in our body but not synthesized by our body? Something which is actually important. Obviously बच्चे phenylalanine. Phenylalanine, ठीक है? And when you talk about the T's here, so T stands for tryptophan. T stands for tryptophan, and the another T stands for the threonine. Another T stands for the threonine. Clear बच्चे? So tryptophan and threonine. Tryptophan and threonine. Clear बच्चे? Tryptophan and threonine. ठीक है बच्चे? Tryptophan and threonine. So they are the essential amino acid. They all are the essential amino acid. So 9 there. 1, 2, 3, 4, 5, 6, 7, 8. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 they are now. So in some books, right? In some books, right, bache, they didn't mention the histidine. In some books, histidine and arginine, they are not mentioned, because histidine and arginine, you can add a note here. Histidine and arginine, they are considered as the semi-essential amino acid. Right, these two are considered as the semi-essential amino acid. Clear bache? Clear bache? They are considered as the semi-essential amino acid. Understood? And why are they considered as semi-essential? Right, because they can be synthesized in adult. Okay? Okay? Right, because they can be synthesized in adult, but in children they are not synthesized in sufficient amount. They can be synthesized in, right bache, they can be synthesized in, right, where are they synthesized? They are synthesized in the adults, right, but in children they are not synthesized in the sufficient amount. So examples are important. Can we quickly revise it? Can we quickly revise it? So we started the story with the help of this 20 amino acids. So we know that aspartic acid, glutamic acid, they have acid in their name. So obviously they are acidic. For basic we have trig. What is the trig? Hall is the trig. What is the trig? Hall is the trig. Okay? What is the trig? Hall is the trig. Okay? So histidine, arginine and the lysine. Histidine, arginine and the lysine. That's what we have. Then bache, see, phenylalanine, tyrosine, tryptophan, aromatic. They have the ring in their structure. Cysteine, methionine, they are the sulfur containing amino acid. Right? So acidic basic amino acid, you know, remaining 15 are the neutral amino acid. Remaining 15 are the neutral amino acid. That is what you have to remember. Okay? So glutamine to glutamic acid. You remember from now transport in plant chapter is deleted, but there a topic of reductive amination, transamination was given. Hanji, I hope you know that reductive amination, transamination was there in transport in plants. So there they pick up the example कि glutamine will form glutamic acid, asparagine will form aspartic acid. Yes? Yes, do you know that? So I hope you remember, okay? Right, I hope you remember. If you know it, then good, otherwise you can just leave it. But yeah, this trick is important. The private Tim Hall. What's that? That is the private Tim Hall, right? Private Tim Hall. Phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine, lysine. Understood? So remaining are what? Remaining are non-essential, right? Remaining amino acids are what? They are non-essential. Got it? They are non-essential. Clear, bache? Clear, clear, clear. Doubt? Doubt. So which one is the simplest amino acid? Which one is the simplest amino acid? Which one is the simplest amino acid? Tell me, which one is the simplest amino acid? Yes, glycine. Which one is the most complex type of amino acid? Which one is the most complex type of amino acid? So another important topic, amphoteric nature of amino acid. Okay, amphoteric nature of amino acid. This is our another important topic. Amphoteric nature of amino acid. Okay? So, देखो. So when you look at the basic structure, let's not even consider the example of any of the amino acid. So when you look at the basic structure, okay, when you look at the basic structure, what? The basic structure of amino acid. So you know that it is the amino group, which is obviously making it basic, right? It is having the acidic group, which is making it acidic. Like basically what is going to happen, this NH2 can become NH3 positive, this COOH can become COO negative, right bache? So if it is NH3 positive, if let's say there is a positive charge on NH3, like in the basic amino acids, right, like in the basic amino acid, NH3 positive, so it will be cation, right? It will be cation. If the charge is on COO negative, it is the anion. So actually in different pH solutions, these amino acids, they behave in a different way. Okay? In different pH, right, pH solutions, these amino acid, they behave in a different way. Actually what you have to remember, that amino acid, they are ionizable nature. Basically from this side what I want to explain, that they can form the anions, cation anions. So can I say that amino acids, they have ionizable, nature? Can I say that they have ionizable nature? Means they can form the ions, right? They can form the ions. They are having the ionizable nature. Any doubt here? Yes. Do you have any doubt here? Yes or no? They can form the ions. They have the ionizable nature. And why? Why, bache? Why is it so? Due to the presence of NH2 and COH. So in different pH solutions, they'll form the ions. Now what is the meaning of being amphoteric? Just see, here you have one amino acid. Right? Here you have one amino acid. So see, so what can you conclude from this? Yes, people, what can you conclude from this? Yes, this is positively charged, of course, cation. This is negatively charged, of course, anion. Now the one which is present in between, right? So if it is NH3 positive, and here if it is having COO negative, so see, the plus and the minus charge is present on this amino acid. Both the charges are present simultaneously. Both the charges are present simultaneously. So this is the Zwitter ion, and that is what we see in the case of amino acid, amphoteric in nature, something that can behave like acid as well as the basic one, right? Something that is having both the natures, the acidic one as well as the basic one, right bache? The acidic one as well as the basic one. So when you look at this part, this is the Zwitter ion, right? This Zwitter ion is having one positive charge, one negative charge. This is how you can take it, that COOH. So H from this COOH, it will be transferred to this ammonia, right? Sorry, it will be transferred to this amino group, right? This amino group will pick up that H. So it is, right, it is like this. This is how you can take it. This is how you can understand that. This is how you can understand that. So this is a Zwitter ion. So ultimately it is what? It is electrically neutral. It is electrically neutral. Why do we say it is electrically neutral, bache? Because plus 1 minus charge, plus minus 0. Okay, Homo sapiens G plus, tell me bache, what is your doubt? Okay, so so basically what has happened here bache? So Zwitter ion, it is electrically neutral. So amphoteric in nature. What is the meaning of that amphoteric nature of amino acid? That it can behave like as an acid or as well as in basic, right? It depends upon the pH of the solution, and when both the charges are present simultaneously, right, it's a dipolar, isn't it? Two charges are there, plus minus, so it is electrically neutral. Okay, it is electrically neutral. Clear bache? Clear. It is electrically neutral. Understood? Right? So this is all about the amino acid. So can we just revise it quickly from the NCERT? Can we just revise it quickly from the NCERT? Then let's move to the to the polymeric form of amino acid, that is protein. Okay, then we will move to the polymeric form of amino acid, that is protein. So any doubt here, Samiksha, Chetu, Madhu? Yes, बच्चे, any doubt here? बच्चे ज्वेटर आयन में ना पॉजिटिव एंड होमोसेपियंस जी, योर केमिस्ट्री टीचर्स कैन आंसर इट इन अ बेटर वे, बट स्टिल आई कैन से एज पर माई नॉलेज. ठीक है, ज्वेटर आयन is having plus charge and minus charge simultaneously, both the charges are present, so basically charge is there on it, but same, right? It is dipolar, it is having plus minus, so it has become electrically neutral because of the presence of same charges, opposite charges na, but one is plus one is minus one, so it has become electrically neutral. When you are generally saying neutral amino acid, they are not even getting charged na, they are totally neutral. Zwitter ion mein it's the total charge that will, right, in totality the charge will become zero. ठीक है, so this is what I can explain as of now. Baki you can ask it from your chemistry teachers, right? So amino acid, obviously they are also organic, right, having amino group and the acidic group as substituents on the same carbon. So basically they are saying the amino acid and the COH, it is present on the same carbon. Clear bache? So we call them as alpha amino acid, they are the substituted methanes. So this is what I have already explained you, that why do we call them as substituted methane, right bache? Bache, homo sapiens, I told you na that from the biology perspective, this is what I know. Okay, like so many years back I have studied the biochemistry in detail na, and in the chemistry in detail, so that's why, right, that's why I don't remember that. So you should ask it from your chemistry teachers. What say? So there are 4 substituent groups occupying the 4 valency positions. So these are hydrogen, carboxylic group, amino group, and a variable group which is designated as R, that alkyl group. So R group, there are many, because of on the nature of these R groups there are many amino acids bache, right bache? So in proteins we only talk about the 20 types. Okay, so that R can be hydrogen. If that R is hydrogen, amino acid is glycine. If it is a methyl group, it is a alanine. If it is hydroxymethyl, it is serine. So these diagrams are also given in this figure 9.1. Here you have that figure 9.1. I think it is on the next slide. Okay, I'll show you that figure 9.9 as well, don't worry. So now the chemical and physical properties of amino acids are essentially of the amino due to the amino, carboxylic, and the R functional group. These 3 functional groups now, these 3 functional groups are going to decide the properties of the amino acid. So based on the number of amino and carboxylic group, these are acidic and basic. Just say you know that if there are two COH group and 1 NH2 group, so acidic it will be. If 2 amino group, 1 COH group, it will be basic. So basic is not just lysine, but right, basic is not just lysine, no doubt lysine is basic, but along with that it's histidine also, it's arginine also. Okay, it's histidine also, it's arginine also. Understood? They are basic. So it is a previous year question. It's a previous year question. It has been repeated so many times. Even your histones are having arginine and lysine. Okay, histones are having arginine and lysine. Clear, bache? So there are aromatic amino acid. We have already discussed the examples, right? So amino acid can form ions. So in solution of different pH, the structure of amino acid change. So here see, this is the Zwitter ion, but it is electrically neutral. Okay, it is dipolar, electrically neutral it is. Done bache? Electrically neutral it is. So this is all about the amino acid. So now after amino acid we need to start the proteins bache, right? We need to start the proteins. So tell me up to this part if there is any confusion. Hanji, up to this part if there is any confusion, do let me know. Do let me know, students. Do let me know. We need to start the proteins now. We need to start the proteins now. Yes, proteins are polypeptide. Very good. What else do you know about proteins? What else do you know about proteins? Hanji, what else do you know about proteins? Peptide linkage, very good, Hemant. Anything else? Anything else? Chalo, first of all, I want you people to send some emo for you, for your dedication. Right? I know you people consider this chapter as a difficult one, but trust me, it is a very interesting chapter. It's a very literally, it's a very nice chapter. Right? You just need to understand it once. Very good, Samiksha. Excellent. Excellent. So first of all, send some emo for you to motivate you people. Right? Whatever you want to, like, obviously nice emojis, of course, nice emoticons you people can send for yourself. Very good. Primary structure, like, you know a lot about the proteins. So that carbohydrate part, na, because that is not in NCERT, and for you people that is a phobia ki, yaar, why are we discussing so many extra things? But somewhere they are important. Somewhere they are important. Very good, very good. Keep it up. Right? So next is what? Next, the next one that we need to start is what? It is the protein. So guys, there will be a break for 15 minutes and then we will continue. Done? Then we will continue. Okay? So you can enjoy that break. So now, so break time. Right? So after every 2 hours you will get a break. So break time till 11:05, and this time I will be consistent. I'll be punctual. I will join sharp at 11:05. Okay, I will join sharp at 11:05. Okay, it's okay, it's fine. Till then you can just revise. After it, proteins, lipids, nitrogenous bases we will discuss, or that living part. Okay, then we will move to the enzymes. So 2 to 3 hours more and the chapter will be over. Right, now there is no need to go beyond NCERT. Now we will discuss everything in the limits of NCERT only. Fine, fine, but yes, we have to master this chapter. Today if you are thinking ki ma'am will end the class early, okay, and she'll set us free, no, enzyme topic is very important. Today we have to do it, we have to finish it. And moreover, bache, see, protein part is still easy. Nitrogenous bases, lipid part is still easy. I'm telling you, it is still easy, okay? But the main problem is that metabolism wala part. You people miss that part so many times. Just in nature of the bond, okay, dynamic state of body, metabolites wala part, living state. So that things are very important. Okay? No. Today we are going to finish it. Right? Tomorrow there is no marathon. You know, na, one day, one chapter, and you are quite free these days. Chalo, so we'll meet you soon. Hello. Now am I audible? Yes. Now am I audible? Chalo, let's continue, people. So, are you feeling confident? Yes, are you feeling confident? Trust me, after this lecture now, after this one shot, you'll be very happy because all the basics of this chapter will be clear. You're going to love this chapter, and you will answer all the questions from this particular chapter. And these days 3 to 4 questions are coming from this chapter. Okay, so one question for sure is from amino acid, one question for sure is from enzymes, one question will be there from your nucleic acid part, which can be in collaboration with molecular basis of inheritance. From metabolism also question will be there. Okay, metabolism also question will be there. So what we have covered so far, people, what we have covered so far, we have covered the amino acids. So now these amino acids, as I said, they are the building block of the proteins, right? They are the building block of what? They are the building block of the proteins. So proteins are present in acid insoluble fraction, right? Where are these proteins present? They are present in acid insoluble fractions. Am I clear? Where these proteins are present, people? These proteins are present in acid insoluble fraction. Clear, bache? Clear, bache means in retentate. So they are the, yes, proteins are the heteropolymer of amino acid. So they are made up of what? They are made up of amino acid, and they are the heteropolymer of amino acid. What are they, bache? They are the heteropolymer of amino acid. So the point here is that what is the meaning of heteropolymer? What is the meaning of heteropolymer? Hetero means different. Hetero means different. What is the meaning of heteropolymer? Different. So basically in a protein chain you cannot say that all the amino acids are going to be same. Just as cysteine is making the entire protein. No, not possible. Not possible. Different type of proteins they will join with different type of amino acids, they will join and they are going to form the proteins. Clear, bache? They are going to form the proteins. So now how that proteins will form? Obviously peptide bond is going to link amino acids, right? It is the peptide bond which is going to link amino acid. So that is why in the case of proteins, because there will be so many amino acids, which are present in a protein, so obviously they are linked with the peptide bond, right? They are linked with the peptide bond. So that's why for proteins we also use the word polypeptide, right? We also use the word polypeptide. Is that clear? We also use the word polypeptide. Is that clear? Yes, bache, that so many proteins, so many amino acids, they are joined together and they are forming the, right? They are forming a chain. That's polypeptide. So sometimes one polypeptide chain is present in a protein, sometimes many polypeptide chains they are making a protein, and that's what we need to understand. Okay, that's what we need to understand. So in general, in a protein, in a pure protein, people, in a pure protein, 2 ends are there. How many ends are there? 2 ends. Obviously one will be the start, another will be the end. And in general now, the start end is having the NH2, the group. This is what, right? This is what our literature says, right? That in a pure protein, what will you see? You will see 2 ends. What are you going to see in a pure protein? You are going to see the 2 ends, and that one end, right, bache? And that one end will have the amino group, right? This is what you have to remember, right? This is what you have to remember, right, bache? So how that proteins are going to join? Obviously, as I said, it is the peptide bond which will link it, right? Of course, it is the peptide bond which is going to link it. So now let's talk about this peptide bond. Clear, bache? Let's talk about this peptide bond. Okay, so peptide bond is also formed by the dehydration. How will that form? With the dehydration. Just let's see, you have one amino acid. I told you now that majorly naturally occurring amino acid, right? I told you, students, that naturally occurring amino acid, in their case we write the amino group on the left side, right? That is the L amino acid, right? So we write the amino group on the left side. That is what I just said. Okay, that is what I just said. So here this is one amino acid, the basic structure of one amino acid, and let's say here you have another amino acid. Okay, what do you have here? Here you have another amino acid. Clear bache? Clear bache? Now we have to link them. How can we link them? You can see this. How can we link them? You can see this. See, this is one amino acid, this is another amino acid. I have to link these amino acids together, right? I have to link these amino acids together. So students, it's very simple, right? The first amino acids, it's carboxylic group. Second amino acid, it's amide group. So what is going to happen, bache? What is going to happen? The water will be released. What is going to happen? The water will be released. So basically the dehydration will be there. Yes or no? Everyone in the chat section, do let me know, people. Quick, quick, quick. Your energy should be high. Yes, everyone. Your energy should be high. So what is going to be there? Yes, people, what is going to be there? So you guys can see the water will be released. The water will be released. Yes, people, the water will be released, and these two peptides, they will join with each other. So this C double bond ONH bond is actually your peptide bond. Okay, the C double bond ONH is your peptide bond. Is that clear? Right, this C double bond ONH is your peptide bond. So that is what you need to remember. That's how the bond will form. Okay, that's how the bond will form. So now, bache, when two amino acids are joined together, they have one peptide bond in between. So you call it as dipeptide. What do you call it? You call it as dipeptide. What is it, bache? What is it, bache? It is the dipeptide. Is that clear? Is that clear? Yes. Is that clear? Yes or no? Yes or no? Any doubt here, students? Desiti? Anyone having any doubt here? I hope it is clear to you, people. Sure. So now on the configuration of the protein molecule, that's what we have to discuss. So first of all, just keep that bond formation in your mind. This is important. Dipeptide means having two amino acids when they are linked with one peptide. This is known as dipeptide. This structure is a dipeptide. Two amino acid having one peptide bond. This structure is a dipeptide. Got it? This structure is a dipeptide. So now let's talk about the structure of protein, or you can simply say configuration of protein. Okay, this is interesting because, bache, when you talk about the enzymes, you know, enzymes are also, mostly enzymes are also proteins. Okay, so please listen to me carefully. Even you will cover a lot from the enzyme. Okay, you are going to cover a lot from the enzyme. So very first thing is the primary structure. Okay, so basically when we see a protein, we take it as a line. We imagine it as a line, as a straight line, and in that straight line amino acids are linked together by a peptide bond. So when it is the primary structure, it is the simplest form of that, right? It is the simplest form of that protein. It's a straight chain. What is it, bache? It is a straight chain. It's a straight chain of amino acids. What is it, bache? It is the straight chain of that amino acid. That is what the primary structure is. Are you getting my point? That is what the primary structure is. A straight chain of amino acid linked together by peptide bond. And bache, this form is the most unstable one. I'm not going to write these basic points. I want you people to write down that point. So primary structure, what are these primary structures? They are the straight chains of amino acids, right? It is the most unstable structure, and here peptide bonds are going to link these amino acids. Is that clear? Is that clear? Now you know that in our body, who's going to, right? The cells, you know that the ribosomes, they join. Do you remember this? The ribosomes, they join. right? In the presence of magnesium ion, the larger and the smaller subunit will join, and obviously they will form the protein. So that protein, that newly synthesized protein, is also the primary structure, bache, right? The ribosome that forms that protein, that is also the primary structure. So as per the requirement, further it will get folded and will go to the place where it has to. Okay, so this is about the primary structure, right? This is about the primary structure. Now come to the next part, that is the secondary structure. Now bache, it's like it level up will be there. Okay, what will be there? The level up. Now let's say you have a straight chain, okay, and someday I will definitely destroy this mic. Okay, so I don't know why, but always for the illustration I got this mic only. So this is a straight chain, this is the primary structure. Now further coiling is there, that will become secondary. Now further coiling will there, it will become tertiary. Now further coiling and other polypeptide chains, they will join, that will become quaternary, right? That will become quaternary. So when you talk about the secondary structure, here we are going to talk about the alpha helical structure, here we are going to talk about the alpha helical structure, and we will be discussing the beta pleated structure. Okay, so when you are talking about the secondary structure, what are you going to discuss? We are going to discuss the alpha helical and the beta pleated structure. What are we going to discuss, people? We are going to discuss the alpha helical and the beta pleated structure. Understood? Right? Right? So basically here in secondary structure, what has been done? The the coiling, right? The folding and the coiling of amino acids. What is there? The folding and coiling of amino acid will be there, right? And obviously it's not just the peptide bond now that we are going to discuss, we will talk about other proteins also. So first of all, let's discuss about the alpha helical structure given by Linus Pauling, right? So in alpha helical structure, we generally talk about the right-handed alpha helical structures, right? What do we discuss, bache? We discuss the right-handed alpha helical structure, right? Right-handed alpha helical structure, something like this. And how that alpha structure is going to form? How that alpha structure is going to form? Obviously intramolecular bonding. Just let's say this, that's your alpha helical structure. Clear, bache? That is your alpha helical structure. Okay, alpha helical structure, something like this. Fine. So what is going to happen in this alpha helical structure? Obviously bache, when it is a straight chain, amino acids are there, that amino acids are going to coil. And how that amino acids are going to coil, right? And because if we are making one structure, that has to be stable as well, right? The structure needs to be stable as well. You cannot say that the structure is not stable and it is existing. Of course not, right? You cannot say that it is something unstable. No, it has to be stable as well. So you are right. Here what are we going to see? We are going to see the intramolecular hydrogen bonding. That is a direct PYQ, students. Okay, that is a direct PYQ. So intramolecular is the word. Intra means within. So within that polypeptide chain, the first amino acid, the amide group of that first amino acid, the amide group, basically the NH2 of that amino acid, is going to form the bond with the fourth amino acid. Okay, so one amino acid will contribute oxygen, another amino acid will contribute hydrogen. So intramolecular, samajh bache, intramolecular hydrogen bonding will be there. What will be there? Intramolecular hydrogen bonding. Yes, people, there will be intramolecular hydrogen bonding. Understood? Yes, Samad, understood? Everyone here in the class, understood? There will be the intramolecular hydrogen bonding. Done? So that's what you have to remember. So what I just said, the bond will form. The bond will form in between O and H. The bond will form in between O and H, and obviously O and H of same amino acid. The bond is going to form in between the O and H of same amino acid. So basically oxygen of the amino acid, one amino acid residue, and NH2 group of the fourth amino acid residue. Right, bache? In this same chain na, oxygen of first amino acid residue and hydrogen of fourth amino acid residue. So in the same chain, so in between them the intramolecular hydrogen bonding will be there, right? The intramolecular hydrogen bonding will be there. So we have many examples. We have the example of keratin, you know na, keratin protein. The dead part is having the keratin protein. Our nails are having keratin protein. Here are having the keratin protein. Keratin therapy is going on, right? There are shampoos in which you know, the companies, they promise that they are having what? They are having the keratin protein, which is good for the hair growth, right? Which is good for hair shining and all. Okay? So keratin is there. Myosin, tropomyosin, so that are the points. Okay, keratin, myosin, tropomyosin. So these are few examples. So what do you know about the myosin and tropomyosin? What do you know about the myosin and tropomyosin? Yes, students, tell me, what do you know about the myosin and tropomyosin? Anyone in the class? Anyone in the class? Anyone in the class? Exactly, locomotion, myosin, sliding filament theory. Bache, our NEET exam is there in the May month. Okay, even if you are in class 11th, right? I know that you people are having exam in the month of February. Okay, so even if I'm asking a question now, so you immediately need to answer that question. Okay, immediately you need to answer that question. Fine. So myosin associated with that sliding filament theory part. Fine, done. So one is alpha helical, another is beta pleated structure. One is alpha helical and another is what? Another is beta pleated structure. What is this? It is beta pleated structure. So when you people talk about the beta pleated structure, so here you will see a kind of zigzag pattern. Actually, bache, here we see that one protein chain, one polypeptide chain joins with another. So here it is not the intramolecular hydrogen bond, but here it will be the intermolecular hydrogen bond. Which bond will be there, people? There will be the intermolecular hydrogen bonding. So basically two different polypeptide chains will be there, and that two different polypeptide chains, they will join together with the help of intermolecular hydrogen bonding, right? Two different chains. Inter means in between, right? So here you will see the zigzag pattern as well. Let's say there is one polypeptide chain, right? So sometimes it gets folded also. Just say you have seen the girls in sari, right? Or the girls in skirts. So that skirts are also having the pleats now, one after another, one after another, one after another, that folding, that pleats. Right now I don't have any cloth, otherwise I would have shown you that pleats are there, that foldings are there. So beta pleated structure is somewhere like this, right? One pleat, another, another, another. So how are they linked? They are linked with the intermolecular hydrogen bonding. Is that clear? Intermolecular hydrogen bonding. Is that clear? Is that clear? Yes or no? Yes or no? Yes or no? Please tell me yes or no. So two or more polypeptide may join together and they can bend into parallel folds to form the pleated structures. Okay? So just say there are two polypeptide chains. Sometimes that single polypeptide chain can also fold. It can also form the folds, right? It can form the pleats. So this is what you need to remember. Clear, bache? Clear, bache? Yes or no? Yes or no? And it's not mandatory that these adjacent chains, they should be in a same direction. They can be in same direction, they can be in different direction. But what is your main concern here? This thing, intermolecular hydrogen bonding. And when you talk about the example, so it is fibrosin protein which is present in the silk. Okay, which is present in the silk. Done. So the secondary structure, one student was mentioning globular, I guess. So when you talk about the secondary structure now, they will give fibrous appearance. They are the fibrous protein, right? Just allow me 5 more minutes and then I will explain what is the meaning of fibrous protein. Okay? What is the meaning of fibrous protein? Just allow me few minutes, then we will discuss. So this beta pleated part clear? This beta pleated part clear? Yes, bache? This beta pleated part clear? Sure? Sure? The examples clear? Just say fibrosin is here, keratin, myosin, tropomyosin. So myosin and tropomyosin, we discussed that in the sliding filament theory, right? Where do we discuss? We discuss that in the sliding filament theory. Clear, bache? Clear, bache? And then what do we have here? The keratin. Okay? So keratin, you know that dead part is having that keratin. So this protein is rich in cysteine. Okay, it is cysteine rich. It is cysteine rich. And cysteine is sulfur containing amino acid. Understood? Chaliye. So next is the tertiary structure. Very important one. Tertiary structure. Tertiary. Right? Here you will see more folding. Just let's say now there will be more folding, more coiling will be there. Okay? More folding, more coiling will be there. The tertiary structure. Right, here you will see it will be in globular form. It will be in globular. Right, tertiary structure is very important, bache, because biologically that's how the proteins they are active. Just if you talk about the protoplasm of a cell, na, let's say you have a cell, the cell protoplasm, right? You know protoplasm, cytoplasm, right? So that protoplasm is majorly having the tertiary protein. Which proteins are there in that protoplasm? Majorly tertiary. So in tertiary protein, the further infoldings are there, bache, right, bache? Just your myoglobin. Myoglobin is the example of tertiary protein. Do you know enzymes? Enzymes you know now? Yes, bache, biocatalysts. Enzymes you know na? Biocatalyst. Biocatalysts. Yes, Hemant. Do you know biocatalyst? Enzymes. So mostly enzymes are also proteins. So enzymes are also in their tertiary structure. Clear, bache? They are in their tertiary structure. So they are highly folded. They give a globular appearance. Understood? They are highly folded. Tertiary structure. They are highly folded. And they give a globular, like this, globular appearance. Clear, bache? So maximum proteins of this protoplasm, they are having tertiary structure. Even the enzymes, they are having tertiary structure. So obviously it is not just the hydrogen bond which is present here. You will talk about disulfide bond, you will talk about ionic bond, you will talk about hydrophobic interaction. So everything, right, this is what we have to discuss. Understood, bache? This is what we have to discuss. Right, clear? So come to this part. Just here they have given now the protein part. I'll come back to that. I'll go back to that slide also. You please focus here. Proteins are polypeptides. This line clear? They are linear chains of amino acid linked by peptide bond. I hope this line is also clear. So each protein is a polymer of amino acid, and there are 20 types of amino acid, bache. So protein is a heteropolymer because all the amino acids, they are going to be different. Okay, they are going to be different. Done, bache? Right. Okay, let's see. So now that dietary protein, the proteins that we take in our diet, they are the source of essential amino acid. See, dietary proteins are the source of essential amino acid. So this is the underrated line, right? I know while reading you are not going to pay attention, okay, and then in the paper you are going to cry. Okay, you are going to cry. So this is important. Dietary proteins are the source of essential amino acid. Therefore, amino acid can be essential or non-essential. So do you remember essential amino acid, people? No, bache, Rabiul, it's not same. We will discuss about it as well. Do you remember essential amino acid? Yes. Private Tim Hall. Private Tim Hall. Private Tim Hall. Clear, bache? Clear, bache? So protein, they carried out many functions in living organisms. Some transport nutrients, we will discuss that, across cell membrane. Some fight with infection like your immunoglobulins. Some are hormones like your insulin. Some are enzymes. There are many examples of that, right? There are many examples of that. So that is how they have explained the proteins, but we need to know more about the proteins. So in NCERT it is given in a different heading, in a different subheading. Okay? It is given in a different subheading. So we will discuss it here. Understood? We will discuss it here. I will mark your bonds here. So any doubt here? Don't worry, I will explain the tertiary structure, the myoglobin, hemoglobin difference as well. Don't you people worry about it. As of now, please focus here, okay? Please focus here. Okay? So any doubt? Hanji, tell me, any doubt? Any doubt? Sure. So proteins, as mentioned earlier, they are the heteropolymers. Please highlight this line, right? They are made up of different amino acid. So when it comes to the structure, bache, see, in inorganic chemistry, the structure invariably refers to the molecular formula. But in organic chemists, it always write a two-dimensional view of the molecule while representing the structure of the molecule. Right, so in inorganic chemistry, how are you going to explain it? With the help of molecular formula. But in organic chemistry, we are going to represent the structure of the molecule, right? Jaise benzene hai, naphthalene hai, like this. So physicists, they are using three-dimensional views of molecular structure, while biologists, they describe the protein at 4 levels. Imagine, at 4 levels we are going to, right? We are going to discuss that. Clear, bache? So when there is the, when there is a sequence of amino acid where you can see the position of amino acid as well, where there is a linear chain, then it will be the primary protein. Everyone just write it in the chat section quick. Then it will be the primary protein. What will it be? It will be the primary protein. Is that clear? So the sequence of amino acid, that is positional information in a protein, which is the first one, which is the second one, which is the last one. So such information we are going to get from the primary structure. So that's why after explaining a topic, I'm reading the things from NCERT, because we have that, right? We should have that coordination, right? We should have that coordination in our brain ki, okay, fine, that is what our teacher has explained, right? She has cleared the topic. Now let's relate it with the NCERT. Okay, let's relate it with the NCERT. So that's something that we all need to do. Okay, that is something that we all need to do. Understood? Understood? So protein is imagined as a line, bache. Left end is represented by first amino acid, I told you. Right end by last amino acid. So first amino acid means the end terminal. Why? Because amino group is there. So anyone having any doubt? Anyone having any doubt? So first amino acid means the left side, the left terminal, and it will be having NH2 group. So obviously this side will be the end terminal of that protein. This side will be the end terminal of that protein. Any doubt here? Yes, bache. Hemant, Chetu, Sneha, Leharika, Diya, Soumyadeep. Next is the last amino acid is the C terminal amino acid. Right, it is the C terminal amino acid. So a protein thread does not exist throughout as an extended rigid rod, right? So thread is folded. Now here they are talking about secondary structure, right? So you know that helical form. See, it is given in NCERT. Helically folded. Okay. Okay, so some portion of the protein thread are arranged in the form of helix. So only right-handed helices are observed in the protein. So this is MCQ, right? This is MCQ. So what is written in the NCERT? Only right-handed. Okay, only right-handed helices are observed in the protein. So this is important. Please mark it. This is important, right? So other regions of the protein thread are folded into other forms in what is called the secondary structure. So after that, so here they just talked about the helical structures. They didn't mention the beta pleated, but you know beta pleated, right? So beta pleated is having intermolecular hydrogen bonding. Sahil, that is having intermolecular hydrogen bonding. Is that clear? Is that clear? Chalo. So the next part, other regions of the protein thread, they are folded into other forms. Other regions of the protein thread, they are folded into other forms. We have already discussed that. Now they are saying that long protein chain is also folded upon itself like a hollow woolen ball. Long protein chain is also folded on itself like a hollow woolen ball. That is your tertiary structure. That is your tertiary structure. Understood? Understood. So it gives the three-dimensional view. 3D view. So this question can be asked in paper. I am repeating this point again, bache. In the tertiary structure, this protein is biologically active. What is the meaning of it? Right? Your enzymes are also in this tertiary structure. No, biologically active, doing their job. Right? Biologically active. I told you, I have given you so many examples here. So tertiary structure is absolutely necessary for many. Yes, for many. Yes, bache, biological activities of the protein. The most important line. Please highlight that. Okay. So the tertiary structure is absolutely necessary for many biological activities of the protein. Please mark it. Yes, everyone, please mark it. Quick, please mark it. Understood? So tertiary, right? So tertiary structure is absolutely necessary for the many biological activities of the protein. Is that clear? Is that clear? Sure, is that clear? So which bonds are present? Let's discuss about that. So look at this diagram. Primary, the straight one, the secondary. See, in the text they didn't mention the beta pleated structure, but here it is. Okay, so that's why I'm telling you, you should know about the inter and intramolecular part. So here intramolecular hydrogen bonding is there, bache, and here you have the intramolecular hydrogen bonding. Okay, so can you tell me about the bonds here? No doubt here they have mentioned hydrogen and disulfide, but any other bond? Any other bond? So obviously, first of all, there will be the peptide bond. Yes, in tertiary protein, first of all, there will be the peptide bond, and of course it is going to be the strongest one. It is going to be the strongest one, yes or no? It is the peptide bond and it is going to be the strongest one. Yes, it is going to be the strongest one. And then here you will be having the hydrogen bond also. Hydrogen bond also. So after the hydrogen bond, there will be the disulfide bond also. Right, there will be the disulfide bond also, and bache, that disulfide bond is the second strongest one. Okay, it is the second strongest one. Okay, so first peptide, then comes your disulfide. First peptide, then comes your disulfide. So hydrophobic, ionic, these bonds are also there. ठीक है बच्चे, hydrophobic and the ionic bond is also there. Understood? And ionic bond is also there. Clear bache? Clear? So this is what you need to keep in your mind. And when it is the tertiary structure, students, when it is the tertiary structure, student, remember one more thing. When you are talking about the disulfide bond, disulfide bond, but that bond will form in between the cysteine residues. You are not going to talk about the methionine here. Okay, you are not going to talk about the methionine here. This bond is in between the SH group of, right, the sulfur group of cysteine amino acid, not the methionine one. And why? There is a very interesting reason behind it. Why is it so? There is a very interesting reason behind it. Yes, bache, there is a very interesting reason behind it that you will search and you will let me know about it in the, right, you will let me know about it in the chat section. Clear bache? Clear bache? Right, so disulfide bond is the one which is going to stabilize the structure of, this disulfide bond in the tertiary structure is the one which is going to stabilize the, structure of tertiary protein. Okay, which is going to stabilize the structure of tertiary protein. Please note down, kindly note down, kindly note down. Please be quick. Clear? But I am talking about the disulfide bond. Why is it is in cysteine residue? Why not the methionine one? That's my question. Why not the methionine one? That's my question. Yes, so this is, next is the quaternary protein. Okay, so this diagram clear bache? This diagram clear? This NCERT text clear? This NCERT text clear? Right, look at this diagram once again. So they are showing the cartoon A secondary structure. So this is the alpha helical one. This is the alpha helical one. So this is the tertiary structure. So here in this tertiary structure also N terminal, C terminal. N terminal means the first amino acid, it should be towards the left side, although it is folded here, and it should be towards the right side. So that's what you have to keep in your mind. Understood? That's what you have to keep in your mind. Understood? Understood? Yes, bache. Understood? So any doubt here? Please tell me any doubt here. This information is very important. Literally, this information is very important. Now let's go back to that slide. So this tertiary structure, the bonds clear. So I was telling you about the myoglobin. Ki myoglobin is the example of, this is what I just said now, myoglobin is the example of your tertiary protein. So myoglobin, what is that myoglobin? The pigment of muscles, the pigment which is present in muscle. Yes bache, the pigment which is present in muscle, that is myoglobin, that's a tertiary protein. Bache, myoglobin and hemoglobin, what is the difference? That's what you have to tell me. I told you the major difference, that myoglobin is the tertiary structure and your hemoglobin is the quaternary structure. But what other differences are there? What other differences are there related to their saturation and that's what you need to tell me. Clear bache? That's what you need to tell me. Done bache? Okay. Rabiul bache, you need to check it again, okay? So this is what you have to tell me. And remember, you're biologically active, your proteins are biologically active when they are in this tertiary structure, when they are in this three-dimensional structure. Do not forget this point, right? Do not forget this point at all. Now the word is quaternary protein. Now the next is quaternary protein. Next is what, students? Next is what, students? Next is the quaternary protein. So quat means 4. So here what has happened bache, the polypeptide, just let's say many tertiary chains, okay, they came together, they joined together, then they formed the quaternary structure, right? Then they formed the quaternary structure. So you have the best example of hemoglobin. Remember, hemoglobin. So here dissimilar, right? Here you do not have the similar type of protein chains. Here we have the dissimilar type of peptide chains. है ना? This dissimilar type of peptide chains. Do you remember bache, hemoglobin, the respiratory pigment? So it is having 2 alpha chains and 2 beta chains. It is having 2 alpha chains, 2 beta chains. Yes, it is having 2 alpha chains and 2 beta chains. Do you remember? Hemoglobin, globin is the protein part, right? Heme word is also there. So quaternary protein, this is the quaternary protein. Clear bache? 2 or more polypeptide chains of tertiary structure, right? 2 or more than 2 polypeptide chains of tertiary structure means this polypeptide chain is also having a tertiary structure, this polypeptide chain is also having a tertiary structure. So they will unite, right bache? Rabil, they will unite. Okay, what is going to happen? They will unite. Okay, so they will form bonds. That is how we will get it. So when dissimilar types of peptide chains are there, just 2 alpha 2 beta, example is hemoglobin. So we have another protein, you might know about this enzyme, lactate dehydrogenase. So that is also a quaternary protein, but here the chains are similar kind of. Okay, the chains are similar kind of. Clear? The chains are similar kind of. So this is your quaternary structure. So linear strings of sphere, yes, sphere arranged one upon the another, so they will form the quaternary proteins. Clear bache? They will form the quaternary protein. So I am going to mention one line. Everyone write down. Everyone write down. Everyone write down. The specificity of a protein, okay, specificity of a protein is due to its primary structure. The specificity of a protein, that how specific it is, right? You know about the specificity, we'll come back to this. So specificity of the protein is due to primary structure, and biological activity, kindly highlight this point, biological activity is due to tertiary structure. Okay, biological activity is due to tertiary structure. Clear bache? It is due to tertiary structure. Is that clear? Is that clear? So this is all about the configuration of protein molecule, primary, secondary one, right? Then I will tell you about the simple, compound and the derived also. But tell me, up to this point all clear? Yes, up to this point all clear? Up to this point all clear? Please let me know. Yes bache, please let me know. Okay, right? So this is about the quaternary protein. So let's revise this. Let's read the structure from the NCERT itself. Okay, let's read that structure from the NCERT itself. So here you can see that some proteins are an assembly of more than one polypeptide subunits, right? So these individually folded polypeptide or subunits are arranged with respect to each other. Just here you can see linear string of spheres are arranged upon one another upon each other in the form of a cube or a plate. So this is the quaternary structure. Okay, so here they have given the example of the hemoglobin. Can you see that? Right, so it is having 4 subunits, right? 2 subunits of alpha, 2 subunits of beta are present. Okay, 2 subunits of alpha, 2 subunits of beta are present. Doubt? Yes, people, do you have any doubt? Just allow me a minute. Yes? Clear? Sure, so read this paragraph again. Okay, read this paragraph again. Done? Okay, sure. And here you can see I was telling you about this image, no? So this is that amino acid wala image, glycine, alanine, serine. Not just important, very important, right? It is also a PYQ. It is not important, it is very, very, very important. Okay, it's very, very, very important. Done? Done? Sure? Okay, so let's go. So do you know, bache, that on the basis of shape also you have different types of protein? Do you know on the basis of shape also you have different type of proteins? You know, again and again I'm moving the slides because I don't want you people to get confused, right, in that PDF. So in the PDF also things are in sequence, don't worry about it. Fine. So now on the basis of shape, 2 types of proteins are there. One is fibrous and another is globular. One is fibrous, another is globular. So globular means tertiary kind of. So if I have to give you the example of fibrous, you have the example of your keratin, that is fibrous. Globulin, albumin, you know, albumin present in the blood, right? So fibrous one, they are quite stable, right? And globular one, it is unstable. It is unstable. It is having that globular shape. Okay, it is having that globular shape. So fibrous may, it's thread like, long, parallel, right? This is rounded one, right? Fibrous, thread like, long, parallel. It will be kind of rounded one, globular in shape, right? Globular in shape. So bache, one is, this fibrous is structurally very stable, right? In comparison to that, right? They are stable. And here these globular one relatively means in comparison to fibrous. I'm not saying they are totally unstable, no, but in comparison to this, it is unstable. Is that clear? In comparison to this, it is unstable. Is that clear? Is that clear? Right, bache? Right, bache? So these fibrous proteins, they are insoluble in water. They are quite tough as well. Their structure is quite tough and they are soluble in water. Okay, so even they are contractile. Do you remember the example of your myosin, contractile protein? So yes, fibrous, they are contractile, but they all will be, right, they all will be non-contractile. Any doubt? Any doubt? So can you quote any other example other than keratin? Other than keratin, can you quote any other example? Obviously collagen, right? Do you know collagen? Do you know collagen? Most abundant protein of animal world. It is the most abundant protein. How many of you remember this? Of animal world. Or you can also call it as most abundant protein of, this is a PYQ, students. It's a NEET 2020 question. It's a NEET 2020 question. It's a 2020 ka PYQ. Okay, so even it is the most abundant protein of extracellular cellular matrix. Even in extracellular matrix, it is the most abundant protein. Understood? It is the most abundant protein. Right, bache? So it is having triple helical structure, okay, which was discovered by Ramachandran. I hope you have heard Ramachandran plot. Ramachandran plot is not the part of your syllabus. I was solving the PYQs, obviously. They were of year, I don't remember, 2005 or 6, I don't remember. So Ramachandran plot was mentioned. So it is related to proteins. That's what you need to keep in your mind, nothing else. So Ramachandran has discovered the structure of the collagen. So it is triple helical structure, right? It's triple helical structure. Fine. So it is the most abundant protein of the animal world, even the extracellular matrix. If there is any doubt, do let me know. If there is any doubt, do let me know. And elastin protein. Elastin, right, bache? Even in chapter structural organization it is given. Collagen protein form white fibers, elastin forms yellow. So that is also the example of fibrous. That is also the example of fibrous protein. Bache, spherical shape is for the globular protein. Right, Hemant? Spherical shape is for the globular protein. So globular protein mein, jaise one example is albumin, your ribisco. Do you remember ribisco? Most abundant protein of the biosphere, ribisco, globular. Most abundant protein of the animal world, collagen, it is fibrous. And your globulins, immunoglobulins. Okay, these are the examples. Any doubt? Any doubt here? Yes, any doubt here? Okay, sure. And even bache, your hemoglobin, globin protein na, histone protein is also globulin. Okay, so that is it, that's all. You can take the screenshot of this part, okay? You can take the screenshot of this part, okay? Doubt? Yes, any doubt? So, bache, these are simple amino acids. Do you know that these are simple amino acids? What are the simple amino acids? Enzymes are what? What are enzymes? They are also the proteins, mostly. I'm not saying all the enzymes are protein. Mostly enzymes are protein. Am I right? Mostly enzymes are protein. Hai na, bache? So, the point is ki when you talk about this now, this enzymes. So I just said that mostly enzymes are what? They are the proteins. So even in enzymes we discuss simple enzyme or the compound enzyme. Simple, which is just having protein, compound, which is having other groups also. So in general also in the proteins, that's what we discuss. Just sometimes protein and nucleic acid, they are joined. Nucleoprotein, chromoprotein, that color producing pigments are also there attached to it, right? Which is lipoprotein. So they are the kind of compound proteins. Okay? They are the kind of compound protein, right? They are the kind of compound protein. Clear, bache? Nucleoprotein, chromoprotein, lipoprotein. Okay? So that is what you need to keep in your mind. Okay, right, bache? It is the collagen triple helical structure. That's all. So now this table, okay, this table we have to discuss yet, but yes, this table is the another most important table. Okay? This table is the another most important table. So here in this table, right, one question was there from this GLUT part, GLUT 4, and many students, they were not able to answer it, right, because they didn't pay attention on these tables. So once this question, this GLUT 4 was asked in the paper. So collagen, you know that it is making the white fibers. Okay, it is the most abundant protein of the animal world. Okay, it is written here, ribisco, that is ribulose bisphosphate carboxylase oxygenase. It is the most abundant protein in the whole of the biosphere. Important part, PYQ it is. And now you know na, ribulose means it is pentose and it is ketose. Ribulose means it is pentose and it is ketose. Clear, bache? Pentose and it is ketose. So now trypsin, it is an enzyme where that trypsin is present. Now see, digestion and absorption is deleted from your syllabus. Yes, bache, Salavadi, digestion and absorption is deleted from your syllabus. Digestion and absorption, you know that. So trypsin word, this is trypsin is what? Trypsin is an enzyme. Hai na? Trypsin is an enzyme. What is trypsin, people? Trypsin is an enzyme, and that enzyme, the enzyme that we have studied. Okay, it is the enzyme that we have studied, yes or no? Yes or no? Tell me, it is the enzyme that we have studied. Do you remember that? Anyone in the class? Trypsin, chymotrypsin. If you remember, it is in pancreatic juice. So if the question on the basis of right, if in the question in the on the basis of this will come, na, you'll be like, arey, they asked the question from digestion and absorption. Many teachers will start making the videos, but actually this trypsin word is given in this table, and this is what you need to know. Then insulin, which is a hormone. So here they are telling us about the different right, different functions of the protein. So insulin, the protein hormone. How many amino acids are there in functional insulin? 51 amino acids. Okay, 2 chains are there. They are also linked with the disulfide bond. Okay, there also the cysteine is there. Then antibody, which fights with the infection. Proteins also behave like receptors. Okay, they sense, smell, taste, and moreover, one cell is there. Cells are also having receptor, right? The molecule which is complementary to it will bind, and the process will also start. Okay, the process will also start. Done, bache. And the GLUT4. GLUT4 is a transporter. It helps the glucose to enter within the cell. Glucose cannot directly come. You remember co-transport, sim transport wala point. So GLUT4 is that transporter which is present. Now what's your homework? You will tell me where is that GLUT4 present. Okay, where is that GLUT4 present? Clear? Where is that GLUT4 present? So this is important, bacho. Okay, right? It is very important. Not just important, it is very important. Clear? Clear? So now after it, what do we need to discuss? Let's talk about the denaturation of protein. Hanji, according to you, what is denaturation of protein? And give me the examples also. Right? What is the denaturation of protein? And give me the example also. Tell me, students, what is the denaturation of protein? Anyone? What is the meaning of denaturation of protein? Yes? It's very simple, bache. See, biologically, right? Biologically, how that protein is present. Biologically, you know that proteins, they are present in their 3-dimensional form, in their tertiary structure. This is how they are biologically active. Now, bache, if there will be any change, let's say there is any physical change in temperature, right? There is any change can be there. That can be physical change, that can be chemical change, right? That can be physical change, that can be chemical change. Means if this protein, right, if this protein, if it subjects to any kind of temperature change, okay, any kind of temperature change, any kind of pH change, so obviously hydrogen bonding will get disturbed. Hydrogen bonding will get disturbed. Yes or no, people? Why your energy is so low, everyone? Why your energy is so low? We need to finish this chapter today. Okay, let's finish it today. Then let's start structural organization in animal. Then let's finish that structural organization also. And then, bache, then let's move to the human physiology part. So, do you want to start human physiology? Do you want to start and finish human physiology in the month of February? If it is a yes, then let me know in the chat section, please. If it is a yes, then let me know in the chat section, please. Quick. Yes? Done? Quick. Yes, bache. Understood? So any type of temperature change and the pH change will result in disturbing the hydrogen bonding, and this tertiary structure will become secondary, then become primary. So obviously what is it going to do? It will lose its activity, it will not be able to do its job because its structure, right? Because its structure is disturbed. Am I right? So during denaturation, the tertiary will become secondary will become primary, right bache? But primary structure will remain intact. Okay, so obviously because changes are there, so tertiary will become secondary will become primary, but primary structure will remain intact. Okay, primary structure remain intact, it will not further be destroyed due to denaturation. So this is the denaturation. So yes, you are giving me the examples, right? That the coagulation of egg white. Just when you talk about the egg, in the egg, what do we have in the egg? In the egg there is a protein, that is albumin, right? So on boiling this albumin, okay, on boiling this albumin, what will happen to this albumin protein? It will also get coagulated. So this is also the example of denaturation, because of the change in the temperature it is going to occur. Yes bache, right? So because of the boiling. Now curdling of milk. In the curdling of milk, what is going to happen in milk curdling? You know that in the milk there is a protein called casein. So bache, casein protein will be coagulated there. How that casein protein will be coagulated, right? Can you just type the milk sugar in the chat section? Can you just type the name of the milk sugar in the chat section? Remember, lactose. So due to the bacteria, due to the presence of lactic acid bacteria, this lactose will form lactic acid. This lactose will form lactic acid. That will act on milk protein casein, and that will get coagulated. So that's how we get the curd. So that curdling of milk is also example of the denaturation. Understood? It is also example of the denaturation. Understood? Yes bache. Now do you know that the organisms that are poikilotherms, what are poikilotherms? Anyone here in the chat section? What are poikilotherms? Anyone here in the chat section? What are poikilotherms? Tell me, what are poikilotherms? Yes, what are poikilotherms? Obviously they are cold-blooded animals, right? Cold-blooded animals. So in their case, you know that they go for the summer sleep and the winter sleep. They go for the estivation and the hibernation. Why is it so? Why they need to go for the hibernation and the estivation? Why they need to go for the hibernation and the estivation? Anyone in the class? Yes, anyone in the class? Why hibernation and estivation? Of course bache, because they cannot maintain the constant internal condition, right? No constant conditions. So because of that, what is going to happen? Like let's say if their body will face high temperature, if they have to experience high temperature, what is going to happen? Their enzymes, they will get denatured. They will not be able to do their job. So if enzymes will not be functional, the metabolism will not be carried out. Now let's say if the temperature is low, then what is going to happen? The protein will be deactivated. Then again, right, in high temperature case it will be denatured, in the low temperature case it will be deactivated, right? It will be deactivated. So both the things are going to affect what? Both the things are not favoring that organism, right? It can result in its death also. So they have to escape that conditions. Okay, so that is how everything in the science is related. So we were talking about the proteins, we were talking about the denaturation, we were talking about the tertiary structure. Now we got to know that tertiary structure is, enzymes are actually the tertiary structure. When we are saying that proteins, tertiary proteins are biologically active, we are talking about the enzymes, because in the enzymes also the folded structures are there. Right? Don't you know that enzymes are also having the folded structures, and due to that, in the enzymes, if you remember, there are services. These are that depressions, the structures, and you know that to these structures the specific substrate will bind. How are we relating the things? We were talking about the denaturation of protein and we related it with the hibernation and estivation. We related it with the hibernation and estivation. So see how everything is related to each other. Right, this is biology, this is science. So read it with open mind today. Right, be in that receptive mode. Right, be in that receptive mode. Forget everything. Be in that receptive mode, people. Okay. Okay. Right. And one more thing, bache. One more thing. Dekho, in one shots, usually teachers, especially in the biology, they go for the NCERT reading. They explain the NCERT in detail, which is good, which is no doubt very good, because ultimately it is the NCERT from where the paper is going to come. But we know that still till March you have the time, right? You have the time for the complete revision, for the proper revision of these chapters. Okay, just see, after this we will go for structural organization, we will go for the 6 days for human physiology, 1 day for structural organization, 1 day for your animal kingdom. Okay, and the syllabus will be over. Syllabus will be over from our side. So basically in your in this series I need 10 more classes, and from 10 more classes, 1 for microbes and 1 for human health, let's say 12 more classes I need, and the chapter will be over. So I can finish it before March. I can literally finish it till mid of the March. Right, so if I can finish it till mid of the March, you still have the half March and the April where you can revise the syllabus, you can read the NCERT, then we will start the NCERT highlights only. Okay, so that's why we are teaching you the things in detail, so that you have that concept, especially in the biology. Right, and you are not paying attention on the biology, you are just ignoring the biology. And trust me, this year, this is what I have, this gut feeling, and I really want that, don't mind me saying that, I want that NTA should give you a surprise related to the biology. Okay, the paper will be from NCERT, but at least the level of questions should be good. They are asking very easy question from last 3 to 4 years. Right, or I can say that 2 to 3 years, they are asking very simple questions. You are going to be the future doctors. The paper should be challenging for you, right? You are going to take care of the lives of many people. So paper should be challenging. I'm not saying very difficult. No, it should be from NCERT, but direct questions should not be there. It should be indirect as well, right? So be ready for such type of paper. You never know when the things will change. Okay. So be ready for that competition also, and start relating everything all together. So this is all about the proteins, right? This is all about the proteins. So now let's talk about the lipids, right? Let's talk about the lipids. It's not a very lengthy topic. So tell me if there is any doubt. If there is any doubt, so we have discussed everything. We just missed this particular table. Okay, so here in this table, what is given? Composition of cell. So we already know water is maximum, followed by protein, then nucleic acid, then carbohydrate, lipid and ions. So from this part, if I will be the examiner, I will confuse you in between the nucleic acid and the carbohydrate. So remember, after protein it is the nucleic acid. Okay, it is the nucleic acid, right? Understood? Chaliye. So let's start the lipids. So what do you know about the lipids? What do you know about the lipids? Where are you going to get the lipids? Yes, people, where are you going to get the lipids? See the diagrams for that fats, oils and everything. So tell me, what do you know about the lipids? Tell me, students, quick. I am going to start it from the NCERT only. Lipids. It's a very chotu sa topic. It's a very short topic. Let's complete it. Okay. Lipids. What do you know? You don't know anything? No, ma'am, we don't know anything. Hai na? Right now we just want to sleep. Ma'am, we cannot give time for the biology. We do not have time for the biology, ma'am. We are so busy. Am I right? We are so busy, ma'am. We do not have time for biology. Am I right, bache? Chalo, everyone say Ola. Everyone say Ola. O-L-L-A. Everyone say Ola. O-L-L-A. Quick. Everyone say Ola, O-L-L-A. Quick. That's a trick, of course. That's a trick, of course. Chaliye, let's start the lipids. So fat and its derivatives, they are commonly known as lipids. Fats and their derivatives, they are commonly known as lipids. So when you talk about the lipids, are they organic? Yes. Are they organic? Of course, ma'am, they are. So when we talk about the lipids, they are biomicromolecules. Why are they biomicromolecules, bache? Because their molecular weight, okay, because their molecular weight is less than, it is even less than 800 daltons. Okay, it is even less than 800 daltons. What is it? Lipids. Understood? So now, so obviously as per the molecular weight, it should be in acid soluble fraction, but we know this point that it is present in acid insoluble fraction, right? It is present in acid insoluble fraction, so we know about it. So lipids are also the organic compounds. What are they, people? They are also the, yes, they are also the organic compounds. Am I right? So they are the organic compounds having carbon, hydrogen and oxygen, but hydrogen and oxygen are not in the ratio, right? They are not in the ratio of 2 and 1. They are not in the ratio of 2 and 1. Okay? Like in the case of carbohydrate, we had that, na? They are not in the ratio of 2:1. Okay? Okay? Understood? Understood? This is about the lipids. What are we discussing? We are talking about the lipids, right? So see, you know, carbohydrates are the primary respiratory substrate. Do you know that carbohydrates are the primary respiratory substrate? But bache, if you will read it, you know that lipids are also going to generate a very good amount of energy. So why can't we consider lipids as our, right? Why can't we consider lipids as our main respiratory substrate? The reason is, bache, in the case of lipids, more oxygen is required. Okay, more O2 is required for oxidation, right? You know about the, yes, you people know about the lipid absorption. You know that it is going to take a lot of time and it requires more and more oxygen. So when it comes to the carbohydrates, the glucose, it will immediately get the oxygen, it will easily oxidize, and our brain cells, they will get the energy. But in the case of lipids, it is going to take a lot of time. It is having more demand for oxygen. So that's why it is not our primary respiratory substrate, right? It is not our primary respiratory substrate. But let's say if our body is in the condition of starvation, okay, then what is going to happen? Firstly, it will feed on the carbohydrate, on the stored carbohydrate, then lipids, bache, then finally, in the worst case scenario, the protein. Okay? In the worst case scenario, it will be the protein. Okay? So what do we have in lipids? We have carbon, hydrogen, oxygen, but hydrogen and oxygen is not in the ratio 2:1 like in the carbohydrate. And some lipids, they also have what? They also have phosphorus. Phospholipids are there. Some are also having the sulfur, right? Some are also having the sulfur, and some are also having the, how can I forget, nitrogen. Okay, this is what we need to discuss. Okay, lipids, right? Lipids. So we are going to divide it into 3 categories: simple lipids, compound lipids, derived lipids. How are we going to read it? We are going to divide it into 3 categories. One is simple lipids, then compound lipids, and then comes the derived lipids. That's what we have. Then comes the derived lipids. That's what we have. So bacha, when it is the simple lipid, okay, so simple lipid mein further you will talk about fats, you will talk about the waxes, okay, fat, right? You will talk about the fats and you are going to talk about the waxes. So these fats are nothing, they are the true fats, they are the neutral fats. Okay, they are the neutral fats. So this is the categorization, this is the categorization that we have to discuss. So can you tell me what is the difference in between the simple and the compound lipids? Can you tell me what is the difference in between the simple and the compound lipid? Yes, What is the difference in between the simple and the compound lipids? I'm repeating this again. Simple lipids is having true fats, right? It is having true fats or it is having waxes. It is having true fats, it is having waxes. So what is the meaning of simple lipid? Okay, what is the meaning of simple lipid? Anyone in the class? Bache, very simple. They are the esters. They are the esters of fatty acid and alcohol, right? In general, first of all, we are discussing the simple lipids, right? So they are the esters of, yes, they are the esters of fatty acids and alcohol. No, alcohol is the word. Alcohol is the word, not the glycerol here. Alcohol is the word, not the glycerol here. So fatty acid and the alcohol. Right, bache Chetu? Fatty acid and the alcohol. Right, bache? What should be there? Fatty acids and the alcohol. Clear, bache? Clear, clear, clear. So esterification will occur when we talk about the lipid. Ester bond will form when we talk about the lipid. We will discuss that. Now, ma'am, what about the true fats? True fats are actually the esters of fatty acids. For fatty acids, I will use short form FA and glycerol. So when it is a true fat, you can also call it as a neutral fat. So it is the glycerol. You are being very specific. Right, you are being very specific that it will be the glycerol, right? That it will be the glycerol. Now, when it is the wax, in the wax, they are also the esters. There will be the fatty acid. But here, bache, monohydric alcohol will be there. Single OH group, monohydric alcohol will be there. Understood? What will be there? Monohydric alcohol will be there. Right, bache? So waxes. Can you quote one example of waxes? Right? Can you just quote one example of waxes? Anyone in the class? Can you just quote one example of waxes? Any one example of waxes? Alright, it's very simple. The earbud that we use, that we put in our ear, that yellow colored wax, what is it? That is the ear wax. That is cerumen. That is cerumen, ear wax. Ceruminous glands, they produce it. Ceruminous glands, they produce it. Clear, bache? Clear, bache? Right? So what about the compound lipids? Anyone? According to you, what about the compound lipids? Yes, what about the compound lipids? Bache, that's important now. What about the compound lipids? Are they also the esters? Of course, they are also the esters. So when we talk about the compound lipids, they are also the esters of fatty acids with alcohol, right? I'll show you the diagram as well. They are also the esters of fatty acid with alcohol containing other groups, right? Here alcohol is containing other groups. Jaise we have the example of phospholipids. The best example, phospholipid. Remember lecithin, phospholipid. So that is the example here. Glycolipid. That is the example here. So these are the examples of the compound lipids. Right, bache? Srinath? These are the examples of the compound lipids. Is that clear? What about the derived lipids like your steroids? Right? What about the derived lipids like your steroids? Anyone in the class who can give me the example? Anyone in the class who can give me the example of derived lipids? जैसे कि your steroids. I'm giving you direct example. Steroids. Anyone? Yes? Derived lipids. Anyone? Then, bache, I will explain the bond formation as well. Next is the derived. Next is the derived lipid that we are discussing. Derived lipid. It is derived, right? Means they are formed from the simple or compound lipids. They are the derived lipids. They are formed from the simple or the compound lipids. How are they formed? These derived lipids, they are formed from simple or you can say that they are derived from simple and compound lipids. Like in simple and compound lipids, when we make their structure complex, we get them. ठीक है, derived from simple and compound lipids having complex structure. Rings are associated here, right? In their structure, you talk about the rings. They have complex structure. They are insoluble in water and soluble in organic solvents, right? They are insoluble in water and they are soluble in organic solvents, right? They are insoluble in water and they are soluble in organic solvent. What is it? It is the derived lipid. What is it? These are the derived lipids. So here in these derived lipids, बच्चे, hydrocarbon rings are there. ठीक है? What will be there? Hydrocarbon rings are there. So basically derived lipids में you have the examples. Okay, जैसे you have the example of steroids, you have the examples of chromolipids, and in steroids जैसे you have the example of sterol and you have the example of sterone. The word is explaining everything. The word is explaining everything. So anyone who can explain this? Anyone who can explain this? Yes? Anyone who can explain the steroids? Na, they are sterols and sterones. They are sterols and sterones. The word is explaining it. बच्चे, sterol, all alcoholic steroids. Alcoholic steroids. They have alcohol as functional group. जैसे cholesterol. Your sterones, they are ketonic steroids. ON. So functional group is going to be ketone. And these chromolipids, they are also the complex one. They are also present in protoplasm. They are also present in protoplasm. They are also the complex one. So in that detail, it is not the part of our syllabus, right, bache? So they are also present in the terpenes are basically making your chromolipids. That's all, right? The terpenes are making your chromolipids. That's all. Okay? So cholesterol, you know, the most decorated molecule by micromolecule in biology. That's the example of your sterols. Okay? So I'm not going to discuss the structure there in detail. I will just show you the picture. So I hope this part is clear, right? I hope this part is clear. Okay? So steroids, so cholesterol, the most decorative molecule of the biology. Okay? So that is about the derived lipid. So in general, we categorize. What we discuss lipid. What are they? Esters. Esters of what? Fatty acids with the alcohol. Fatty acids with the alcohol. That's how we started the journey, right? That's how we started the journey. So here fats, waxes, they are there. Compound lipids may ultimately they are also the esters of fatty acid with alcohol, but that alcohol is having another groups. Okay, that alcohol is having another groups. Then comes the derived lipids, bache. So they are having hydrocarbon rings. That's like your cholesterol, Clear? So this is what the in general, right, this is about the compound and derived lipids. Now let's discuss these simple fatty acids in detail, but ultimately because this is the part of our syllabus now, so let's discuss that simple, right, simple lipids in detail or simple fats in detail. Let's talk about it. Clear, bache? Let's talk about it. Clear? Yes? Okay, so fats, are they the product of polymerization? Are they the product of polymerization? Are they the product of polymerization? No, because they are micromolecules. They are micromolecules. Right, bache? What are they? They are micromolecules. Is that clear? Is that clear? Yes, is that clear? So when you are talking about the simple lipids, they are the esters of long chain and fatty acids, right? Right, long chain and fatty acids. This is what they are. Yes or no? Yes or no? When you are talking about the simple lipids, they are the esters. They are the esters of fatty acid with alcohol. And bache, in majority of neutral fats, right, in majority of neutral fat, it is going to be glycerol. Okay? In majority of neutral fats, it will be glycerol. Okay? It will be glycerol in the simple fats. That is what you need to remember. And glycerol is, right, glycerol, it is also considered as glycerol, though it is an alcohol, but still bache, glycerol is trihydroxypropane. It is also PYQ, please pay attention. It is trihydroxypropane. It is also PYQ, people. So it is also considered as simple lipid. That is given in NCERT. I will discuss it with you. Okay? So glycerol itself is considered as simple lipid. Let me tell you what. Dekho, one thing is fatty acid. One word is fatty acid. Bache, see, fatty acids, they are the long chain, right, long carbon hydrogen chains. The word is fatty acid. One thing that you should focus here is कि it is acid, so means it should have COOH group. It should have COOH group. If it is an acid, it should have COOH group. That is for sure. That is for sure. This is something that we cannot replace, right? This is something that we cannot replace. Yes or no? Yes or no? Fatty acids are basically the esters and glycerols, na? So basically I am saying that this is for sure that fats are going to have, right, that fats are going to have the, right, the fats are going to have the COH group, and then there will be long chain of carbon hydrogen. Am I right? Then there will be the long chain of carbon hydrogen. That's what we have. Yes, ma'am. So let's say I'm giving you one simple example to explain this topic. So when it is a fatty acid, can I write it like this? RCOOH. This R is telling me about that long chain. This R is telling me about that long chain of fatty acid. Can I say that it is fatty acid? Now the another word is alcohol, right? And majorly I told you it is going to be glycerol. It is going to be glycerol. That is trihydroxypropane. That is trihydroxypropane. Bache, propane means how many carbons? Propane means how many carbons? Propane means 3 carbons. 1, 2, 3. 3 carbons. And trihydroxy means 3OH also. Means 3OH also. So trihydroxy. That's it. This is your glycerol. This is your glycerol. This is your trihydroxypropane. A very simple structure it is, right? Tri means 3 hydroxy, 3OH. Trihydroxypropane it is. Trihydroxypropane it is. What's that? It is trihydroxypropane. Is that clear? Is that clear? Is that clear? Is that clear? Trihydroxypropane. Trihydroxypropane. Right? So now we have to show the esterification. Okay? So let's write the glycerol first. Okay, so let's pick up the acid, C double bond O, OH. So again here, the water will go out. What is going to happen here, bache? What is going to happen here? Yes. What is going to happen here? It's simple. The water will be eliminated. Okay, the water will be eliminated, Krutika. It's simple. So imagine, like this is a glycerol, right? This is a glycerol. So now here in this glycerol, students, right, here in this glycerol, what has happened? Right, the bond formation has occurred, and what are we going to get? We are going to get the, right, neutral fat. What it will be? Let me show you. H2 and 2HO. So it will be O. It will be okay, right? That's what we are going to get. That's what we are going to get. Or is there any mistake? Is there? It is H2O, na? So that is what you are going to get. That is what you are going to get. So dehydration will be there. Tell me, dehydration will be there. So basically OH from this, H from this. Okay, OH from this, H from this will be out. You are left with this. You are left with this. So what do we have here? right? Rahul, what do we have here? We have one glycerol, we have one glycerol, we have one fatty acid. So what are you going to get? The ester bond has formed. This is the ester bond. This is the monoglyceride, students. What is it? It is the monoglyceride. Yes, what is it? It is the monoglyceride. Yes, bache, what is it? It is the monoglyceride. It is the monoglyceride. Clear? Clear, clear, clear. It is the monoglyceride. Is that clear? Is that clear? Yes. Yes. So one more diagram, and then with the help of that diagram only I will explain it. So now what is the next point here, bache? See, now obviously water will go out. Now here you do not have just one fatty acid, you have more than one, you have two fatty acids. These can be same fatty acids, they can be different fatty acids. Krutika, they can be same fatty acids, they can be different fatty acids. Okay, so here what will be done? The water will be eliminated. The water will be eliminated. So obviously now, and let's say here also, so ultimately what are you going to get? Now three fatty acids are there, and of course only one. Yes, bache, three fatty acids are there, but only one glycerol. Pay attention, this is important. Okay, please pay attention, this is important. Okay, so now what will you get? Yes, people, what will you get? Tell me, what will you get? This is the triglyceride, the most common type of fat. It is the triglyceride, which is the most common type of fat. So if there is only one fatty acid, right, if it is esterified with the glycerol, monoglyceride. If two fatty acids are esterified with the glycerol, diglyceride. If three fatty acids are esterified with the glycerol, then it will be the triglyceride. Is that clear? It will be the triglyceride. Is that clear? Is that clear? So these fats, right, bache, these fats, they can be oils, right? They can be oils, right? It depends upon their melting point. Okay, these lipids, they can be fats, they can be oil. It depends upon their melting point. Is that clear? It depends upon their melting point. Okay, okay, done. So now bache, when you talk about these fatty acids, what do we have in these fatty acids? This is important, please pay attention. Right, now when you are talking about the fatty acids, देखो, one thing that you need to keep in your mind, it is the acid, so it will have COH group. Definitely it will have COH group. Then this fatty acid can have, right, the R group will be there. As I said, R group, this R group can be CH3, this R group can be C2H5, so this can be anything, right? So this R group means this methyl group will have carbon number from 1 to 19. Okay? The carbon number can be from 1 to 19 in the R group. What am I saying? I'm repeating this again. In the R group, the carbon number can be 1 to 19. The carbon number can be 1 to 19. And now add one carbon from this COH, so it will become 20. So maximum it will be 20. Okay? So I'm repeating this again. I'm saying R group will have maximum, right? The carbon number here can be from 1 to 19. The carbon number here can be from, yes people, the carbon number here can be from 1 to 19. Is it clear? Is it clear? Is it clear? Sure? Sure? Are you sure? Right? Right, so this R group waste, right? So overall that is how you are going to get the fatty acid. Okay? So before, okay, done. So now bache, do you know that in this carbon hydrogen chain there can be the saturation and there can be the unsaturation. What is that saturation and unsaturation? Anji, chemistry students, now tell me. Right, when you are talking about these fatty acid, overall I told you the total number of carbon maximum will be till 20. Okay, maximum will be till 20. So what am I saying here? I'm talking about I'm asking this thing from you, that what is the meaning of saturation and unsaturation? Saturated fatty acid and unsaturated fatty acid. Can you tell me what are saturated fatty acids and what are unsaturated fatty acids? Can you tell me? Hanji, can you tell me? Single bond is saturation, presence of double bond is unsaturation. Very good, right? So yes, you are right, only single bonds are present, right? Only single bonds are present. They are present in between carbon carbon, in between carbon carbon, only single bond is there, right? But here, right, double bonds are also present. Double bonds are also present. Yes people, they are also present in carbon carbon, carbon carbon chain, है ना? Double bonds are also there in carbon carbon chain, right बच्चे? So unsaturated fatty acids are your essential fatty acid and these saturated fatty acids are your non-essential. This point is very important. Right, now again I am not going to explain you, right? I'm not going to explain you the meaning of essential and non-essential. Literally I'm not going to explain you should know about it. So these are the non-essential fatty acids, these are the essential. Can you tell me the meaning of essential and non-essential in the chat section? Can you tell me the meaning of essential and non-essential in the chat section, please, quick? Can you tell me the meaning of essential and non-essential in the chat section? Obviously essential, which are very important for the body but are not synthesized in the body. They should be the part of our diet. Okay, they should be the part of our diet. They cannot be synthesized in the animal body. Okay, okay. And now here, because they are saturated, right? So the fats having the more saturated fatty acids, they will be in solid. They will be in solid form at their room temperature. जैसे कि butter, right, like ghee. They are the examples of what? Saturated fatty acids. But now when you talk about the unsaturation, right, when you are talking about the unsaturation, so they are lipids with more unsaturation. They are mostly present in liquid form. They are mostly present in liquid form. Even in but you might have seen the ads where they talk about MUFA, PUFA, right, where they talk about MUFA, PUFA, MUFA, PUFA, monounsaturated fatty acid, polyunsaturated fatty acid. If in that fatty acid only at one point only double bond is there, monounsaturated fatty acid. If in that chain many unsaturations are there, polyunsaturated fatty acids, right, then it will be polyunsaturated fatty acids. Are you getting it? Are you getting it? So they are liquid at room temperature. Just your oils, right, your mustard oil is the example. Mustard oil is the example. Now if I talk about the metabolically, but metabolically these saturated fatty acids, they are not very reactive. They will get deposited here in our body. They are the one which result in the formation of, in the blockage of the blood vessels, right? Because they are not so reactive, they do not participate in the metabolism, right? They are the one which cause the obesity. Clear bache? They are the one that causes the obesity. Clear bache? Right? So metabolically, yes, so when you talk about the saturated fatty acid, so metabolically are less reactive. Metabolically, yes, metabolically they are what? Metabolically they are less reactive, isn't it? They are less reactive, right bache? So do not participate in metabolism. So obviously they are the one which are responsible for obesity, right? So this is what I am writing here. I hope that hint will be sufficient for you. So in unsaturated fatty acid it will be opposite, isn't it? It will be opposite. Clear bache? Clear बच्चे? So mostly when you talk about the animal fat, okay, when you talk about the animal fat mostly, so it is saturated, it is having the single bond. And here when you talk about the plant fat, right, mostly it is having the unsaturation. So this is something that you need to know. Clear बच्चे? So can you just give me the examples here? Can you just give me the example here? Palmitic, stearic, palmitic, stearic. ठीक है, examples are the most important one. Palmitic acid, stearic acid, right? Palmitic acid, stearic acid. And I gave you the trick in the starting na, ola. I gave you the trick in the starting na, we will come back to this. जैसे oleic acid, linoleic acid, linolenic acid, all of these things are important. And arachidonic acid. Arachidonic acid. So these are the examples of the unsaturated fatty acid. Okay, these are the examples of the unsaturated fatty acid. Clear बच्चे? So it is ola, it is ola, it is ola. Clear बच्चे? Done? It is ola. So saturated fatty acid and unsaturated fatty acid, this is what we have here. Clear? Clear, clear, clear. So palmitic acid, here the carbon number, this carbon number will decide everything. 16, stearic is having 18, stearic is having 18. And this is ola, 1 2 3 4, about the unsaturations, right? About the unsaturation. Clear bache? Now I'll give you the examples here. So this trick is important, ola. Okay, this trick is important, that is ola. Okay, okay, chalo. So I'm writing it again. The examples are important, they are very important, students. Okay, they are very, very, very important. Saturated fatty acid and unsaturated fatty acids, saturated fatty acids and the unsaturated fatty acids. Okay, so when it is the saturated fatty acid, so C is 16 and 18. So it is PS, right? Palmitic acid and it is stearic acid. It is palmitic acid and it is stearic acid. So palmitic acid, CH3, CH2, basically COOH, 1 carbon, 2 carbon, and we are left with 14. So what should be here, right? This. So that's how you can write it. Okay, that's how you can write it. This is palmitic acid. This is a formula for palmitic acid, because right now I don't want to draw that, no, that complete carbon hydrogen chain. So this is the formula. Yes, it is important. Yes, it is important. Now stearic acid, how many? Yes bache, how many carbon number? What is the carbon number? 18. You need to know about the carbon number, right? You really need to know about the carbon number. So simply you can write it like this: COH, CH3, then CH2, then 16, and then 17, 18. So this is your stearic acid formula. Important they are. Okay, they are important. Understood? They are important. Done bache? Okay, so the trick here is OLA, O-L-L-A. So oleic, linoleic, linolenic, arachidonic. So remember one thing, when it is arachidonic, your carbon number is 20 and here carbon number is 18, carbon number is 18, carbon number is 18. Okay, so ma'am, what's the difference? If carbon number is same, then what is the difference? Difference is of the saturation बच्चे, that how many double bonds are there. जैसे oleic में only 1, linoleic में 2, linolenic में 3, and here you are having 4. Right? So one double bond means you have to say bye bye to the hydrogen. 2 double bond means, right, 2 double bonds means you have to say bye bye to the hydrogen, isn't it? Isn't it? So how are we going to start? So carbon number is 18 overall, right? Carbon number is 18 overall, so H will be 34, right? Carbon 18, because 2 saturations are there, H will be 32. Carbon 18, H will be 30. Okay, H will be 30. Clear bache? And when it comes to the O, because you know that COH is having the O only, so O2, O2, O2. And here C is 20, right? So at 4 places there is the saturation. So tell me what will be the hydrogen number here. You can, yes bache, what should be the hydrogen number? Of course, it's very simple. You can just look at the formula and you can answer it now. So at 4 places there is the saturation, right? But here the number of carbon is more, right? There the number of carbon is more, so do not get confused. So it should be H32, that's all. So this is what you need to remember, Ola. Okay, this is what you need to remember, Ola. Okay, so out of that bache, mainly, right, mainly these 3 are your essential fatty acid. Okay, because essential fatty acid, and they are PUFA. What is PUFA? PUFA, polyunsaturated fatty acid. They have more than one bond. Okay, so polyunsaturated fatty acid it is, and this one here only one. Okay, these are the polyunsaturated fatty acid. Okay, okay, clear bache? Clear, clear, clear? Any doubt? Clear? Sure? Done? So this is important, please take the screenshot. Okay, this is important, please take the screenshot. We are about to finish, right bache, we are about to finish the lipids, and then we will start the nucleic acid, of course from the nitrogenous base, and then we will move to the metabolism and the enzyme part. Okay, and I know you people can do it. What's it? I know you people can do it. It's a lengthy chapter, it's worth giving time, and it's an important chapter. I cannot just read NCERT, I cannot just translate NCERT, right? I am not going to justify my job in that way, because I know, right, the NCERT translation is not helpful for this chapter. For this chapter, the concept needs to be very clear. Fine, the concept needs to be very clear. So please try to understand that, okay? Right now next is the compound lipid. So you know that in compound lipid what do we have? So from examination point of view, I'm telling you the important thing, and here we will be talking about the phospholipids. I already told you what exactly are the compound lipids. Okay, I already told you what exactly are the compound lipids. So we are going to talk about the phospholipids. So phospholipids mein kya hoga? There will be 2 molecules of fatty acids. There will be 1 glycerol. Glycerol will always be 1. Okay, because it's a trihydroxypropane. 3 OH are there. Then there will be the phosphate group, H3PO4 will be there, and then there will be the nitrogen containing compound, right? Nitrogen containing compound. So what am I talking here bache? I am talking about the lecithin, the most common, the most abundant phospholipid, right? Lecithin, the most abundant phospholipid. Do you remember this? Lecithin, the most abundant phospholipid. Clear bache? So phospholipids are the most common type. You know that they are making the cell membrane, and lecithin is the most common type there. Clear bache? Clear bache? Yes? Yes, yes, yes. Okay, so let me tell you, if you remember, even in the, this is how we draw the phospholipids in the plasma membrane. This is how we draw the phospholipid. But we know that this is the hydrophilic head, right? If this is a cell, here it is having the plasma membrane, so you know that it is phospholipid bilayer. It is phospholipid bilayer. So here it is having the head, so it is hydrophilic head. It is hydrophilic head. This tail is hydrophobic, right? So you have hydrophilic head, you have hydrophobic tail. So hydrophilic is water loving. Okay, hydrophilic is water loving, and this is polar as well. Okay bache, and the tail is non-polar as well. Understood? The tail is non-polar as well. And moreover, my dear students, here in this head region, you will have that H3PO4, you will have that nitrogen containing compound. Okay, you will have that nitrogen containing compound. Understood? That is what we are going to get here. Okay, okay, and when it is the hydrophobic tail, obviously the fatty acid is present there. The fatty acid is present there. So most common type of phospholipid is lecithin. This is what you have to remember. Okay, Okay, so lecithin is known as the phosphatidylcholine. Fine, that is phosphatidylcholine. Lecithin, it is having that name now, phosphatidylcholine. Most common type. Fine, it is the most common type of the phospholipids. Phosphatidylcholine. Any doubt? Any doubt? So its structure is given in NCERT. Okay, so we have another examples also. As of now, you just focus on the lecithin, the most common type of phospholipids. Clear, bache? Clear, bache? That's it. That's all. Okay, that's it. That's all. So look at the NCERT. So here in the NCERT also, see, it is given a, they are water insoluble. They could be simple fatty acid. They are having a carboxylic group attached to R group. That could be methyl, ethyl, or higher number. I told you. So that R group can have carbon 1 to 19. जैसे palmitic acid is having 16 carbon. Important it is. Arachidonic acid is having the 20 carbon atoms, including the carboxylic carbon. So don't make the mistake here. Including the carboxyl carbon. Clear, bache? Clear? So fatty acid could be saturated, that is without double bond. It can be unsaturated with double bond. So another simple lipid is your glycerol. Do not forget that. So glycerol is a trihydroxypropane. It is also a PYQ. Fine, it is also a PYQ. Clear, bache? Clear, clear, clear. So lecithin is the, see, lecithin is the one example of the phospholipid. So remember this point. So we have covered everything from this part. Now move to this part that is related to the nucleic acid. Now for nucleic acid, let me tell you one thing very clearly. Let me tell you one thing very clearly. Hanji bache, aesthetic, I don't know how to teach, right? I am a very bad teacher. Please do not waste your time by watching my lecture. Okay, bye bye aesthetic. Tada. Okay, tada. So guys, lipid wala part clear. Esters they are. What are they, bache? They are the esters. What are they, bache? They are the esters. Esters of fatty acid and glycerol. The structure of glycerol clear? The structure of glycerol clear? So the very common mistake is the glycerol number. Sometimes students, they make it 1, they make it 2, they make it 3. Glycerol is 1, people. When you are talking about the simple fats, not true fats, glycerol is just 1. It is trihydroxypropane. Means it is having 3 carbon, it is having 3 OH. It is trihydroxypropane. It is trihydroxypropane. Clear? Right? Right? Okay. So how many glycerol? 1. And even in phospholipid, how many glycerol? 1. How many fatty acids? 2. Important it is. How many phosphate group? That H3PO4 will be there. And then nitrogen containing compound. So nitrogen containing compound means you are talking about lecithin. You are talking about lecithin. Right? You are talking about lecithin. Is that clear? Is that clear? You are talking about lecithin. Okay, okay. So now see what are they saying, that living organisms, right? That living organisms, they have a carbon compound in which heterocyclic different rings can be found. Some of these are nitrogenous bases. See students, in the chapter molecular basis of inheritance, right, in the chapter molecular basis of inheritance, in detail we have discussed that. So if you are following this Game of NEET series, I believe that you have attended that chapter, so you know about the nucleic acid. So I will just give you the overview. We are not going to discuss the structure here. I will just give you the overview. Done, bache? Clear, bache? Clear, clear, clear? Clear, clear, clear? Okay, so first of all, what you need to know, that we are going to talk about the nitrogenous bases. So bacha, nitrogenous bases, the nucleoside, nucleotide, where are they present? They are present in the acid soluble pool. Where are they present? Where are they present, Chetu? They are present in the acid soluble pool, right? Where are they present? They are present in the acid soluble pool. Is that clear? Is that clear? So nitrogenous base. So nitrogenous bases may, obviously they are nitrogen containing compound. You talk about the purines and the pyrimidines. Okay, you talk about the purines and the pyrimidines. So purines may it will be puga, that's the trick. Pyrimidines may it will be cut, that's the trick. So puga, pu means purine, G and A means guanine and adenine. Guanine and adenine. And what is the meaning? What is the meaning of this cut? Before that, see this. Guanine, adenine, 9. So purine is a 9-membered ring. It's a 9-membered ring. So in this 9-membered ring, at position 1, 3, 7, 9, what do you have? Nitrogen is present. What do you have? Nitrogen is present. Is that clear? What do you have? Nitrogen is present. Understood? Now when you are talking about the pyrimidine, we have cytosine, we have uracil, and we have thymine. We have cytosine, uracil, and thymine. And these pyrimidines, bache, they are the 6-membered ring. What's that? It's a 6-membered ring. It's a 6-membered ring. Okay. Okay. So uracil, thymine, cytosine. That's what I have. So bache, see, cytosine and thymine, it is present in DNA. Okay, cytosine and uracil, it is present in RNA. So that is important MCQ. We have covered that in molecular basis of inheritance. Still I am explaining you, this is important MCQ. Is that clear? This is important MCQ. Is that clear? C and T in DNA, C and U in RNA. Clear bache? C and T in DNA, C and U in RNA. Is that clear? Any doubt? Any doubt? Yes? Any doubt? This is about the purines and pyrimidine, 6-membered. So here at position 1, at 3, nitrogen is present. Nitrogen is present. This is nitrogenous base. Next, nucleoside. Okay. Next, nucleoside. So nucleoside, N for nitrogenous base, S for sugar. N for nitrogenous base, S for sugar. Okay? N for nitrogenous base, S for sugar. So they are going to join. See, nucleoside it is. So when they will join, they will form nucleoside. So sugar mein bache, you know that the ribose sugar, you know the structure as well. Today itself we discuss it. And we have the deoxyribose. What do we have? We have the deoxyribose sugar, DNA. Okay? DNA is there, and nitrogenous bases. See, it's very simple. If it is adenine plus ribose, how are they going to join? Yes, people, how are they going to join? Which bond do they have? Right? Which bond do they have? Adenine plus ribose. They are joined with the help of N-glycosidic bond. So nitrogenous base and the sugar, they will join with N-glycosidic bond. But we have already completed this part in the series. In which series? In molecular basis of inheritance series. Okay? In molecular basis of inheritance series. So we have already completed that in that particular chapter. So I am not discussing the structure. I am not explaining the bonding because it is already done there. In detail we have discussed it. So if you want to revise, you can check the molecular basis of inheritance part 1. Okay? So this is just for quick revision. If you have problem, please go and revise there. Okay? So adenyl plus ribose, it is N-glycosidic bond. It is N-glycosidic bond. So it will form adenosine. Right? So if it is simple adenine, that's a PYQ. If it is simple adenine, that is nitrogenous base. If it is adenosine, it is the nucleoside. It is the nucleoside. Right? Same way, I'll give you one more example. Let's say if it is cytosine plus ribose. Let's say if it is cytosine plus ribose, it will be cytidine. It will be cytidine. Clear? It will be cytidine. Understood? Understood? Is that clear? So here also you will have N-glycosidic bond. Here also you will have N-glycosidic bond. Clear? So if sugar will be the deoxyribose instead of ribose, if sugar will be the deoxyribose, so it will be deoxyadenosine. It will be deoxyadenosine. If sugar will be deoxyribose, it will be deoxycytidine. So that's the nomenclature, bache. That's the nomenclature. Just say it will be guanosine and uridine. These are the nucleosides. Okay, deoxyguanosine. If deoxyribose is the sugar, deoxythymidine. Deoxythymidine or simple thymidine, it is also the nucleoside. Understood? It is also the nucleoside. Clear, bache? So next is the nucleotide. Next is the nucleotide. Next is what? Next is the nucleotide. So T means 3. So nitrogenous base, sugar, phosphate. Okay, so nitrogenous base plus sugar means nucleoside. So in nucleoside you have added 1 phosphate, so it will be nucleoside 1 phosphate. Can I say so, people? Yes, can I say so? It's the nucleic acid. So which acid it will be? Phosphoric acid. Phosphoric acid is phosphoric acid. Which acid it is? Phosphoric acid. Which acid is it, Ansari bache? Phosphoric acid. So nucleoside monophosphate. So simple it is. In nucleotide, let's say if it is adenine, if it is ribose plus, there will be the phosphate. There will be the phosphate. So you will get adenosine. You will get adenosine monophosphate. You will get adenosine monophosphate. Are you getting my point? Adenosine monophosphate. So it is also known as adenylic acid. Okay, adenylic acid. So that's how you are going to read it. So deoxyadenosine monophosphate, deoxyadenylic acid. So that's the nomenclature. So question will definitely come from this nomenclature part. Okay, the question will definitely come from this nomenclature part. So this is about the nucleic acid in detail. I am not going to discuss it, but because already I think I have given 2 to 3 hours to explain this. Okay, already I have given 2 to 3 hours to explain it in one to 1.5 hours to explain it in molecular basis of inheritance. So do not forget the nomenclature, do not forget the structure of the, right, do not forget the structure of the nucleic acid. Okay, bache, right? So after the class now you can read NCERT, you can read NCERT in detail. You can read bache NCERT in detail. You will get the answer of all the questions here. Okay, you will get the answer of all the questions. So see here also in journal they have explained. See, sugar, they are called nucleoside, we have discussed. Nucleotide, we have discussed. So the nomenclature needs to be clear. Okay, chalo. So let's move to the another part. The diagram. Yup, this is the diagram. So bache, this is the cholesterol. So cholesterol diagram is given. See, try to remember this. So it is the example of steroid. What type of steroid? Sterol. It is the example of derived lipid. This is important. This is phospholipid lecithin. It is also the MCQ. See, so here they have confused, but R1, R2, these are 2 fatty acids attached to this glycerol. This part is the glycerol, students. This part is the glycerol. So to this carbon now, if you will notice, students, to this OH, O group of this glycerol, phosphate is attached, and this phosphate is further, right? This H3PO4 is further attached to this nitrogen containing compound. So this is actually, because this is lecithin, so this is actually choline. This is actually choline. Clear, bache? This is actually choline. Is that clear? Is that clear? Understood? Is that clear? So this part clear? This part clear, everyone here in the class? This part clear? Tell me. This part here clear? So the diagram is important. Diagram is important. Please do not miss that. See, palmitic acid. Take it, triglyceride. Diagrams are important. So if this N wala part is given here, you are talking about the phospholipid. Don't make the mistake here. Fine, do not make the mistake here. And see these structures. So adenine is a nitrogenous base, adenosine is a nucleic acid, adenylic acid is a nucleotide. It is a nucleotide. So this diagram is also a previous year question. Okay, Grutika, this diagram is also a previous year question. So see, nitrogenous base is attached to the sugar with the help of N-glycosidic bond, hai na bache? With the help of N-glycosidic bond, and this sugar is attached to this phosphate with the help of phosphoester bond. Clear? It is attached here with the help of phosphoester bond. Clear? With the help of phosphoester bond. Understood? So uracil, pyrimidine, uridine, so you should know about it. Please do not miss that. Okay, please do not miss that. Chalo. So now let's talk about the metabolism and then the enzymes, bache. Okay, so see, the nucleic acid part is given here, right? As I said, we have already discussed it in molecular basis of inheritance. ठीक है. So adenine, guanine, which is substituted by what, everything is discussed there in detail. I'm repeating this again, you can check that lecture. Okay, you can check that lecture of molecular basis of inheritance. So that biomolecules done. Chalo, so let's move to the metabolism. So for the next part now, it is only the NCERT reading that we have to do. Okay, there is no need to go beyond NCERT. It is only the NCERT reading that we have to do. Fine. Fine. So here bache, in this particular table they have explained the nature and bond. What have they done? They have explained the nature of bond linking monomers in a polymer. Okay, so they are explaining the nucleic acid. So guys, do revise it from NCERT just to refresh your concepts. Adenine will form 2 hydrogen bonds with thymine. Cytosine will form 3 hydrogen bond with guanine. When it is a simple nucleotide, okay, then it is the phosphoester bond and glycosidic bond. So if it is a polynucleotide chain, then you will have phosphodiester bond. Which bond will be there? Phosphodiester bond will be there. Okay, so see, they have explained amino acid are linked by peptide bond, monosaccharides are linked by glycosidic bonds. ठीक है बच्चे. And in nucleic acid, one phosphate molecule links 3' carbon of one sugar of one nucleotide to the 5' carbon of the sugar of the succeeding nucleotide. That's how you have to revise it. Okay, so Watson and Crick has explained that DNA structure. So again, it is in class 12th, it is discussed there. So it is showing that sugar phosphate backbone. Understood? So this is the next part that we have to start. So are you ready, people? So here in this portion, there are few things that students, they don't understand. Let's read it directly from the NCERT and then let's talk about the enzymes. So I think till 2 AM we can finish this. We can finish this. Acha karu na? Okay. This metabolism wala part is important karu na. We have to discuss it. Okay. We have to discuss it. Okay, chalo. So now what are they explaining? We are talking about the dynamic state of body constituents, the concept of metabolism. So I told you, if it is a living body, means you are talking about the metabolism, means chemical reactions are going on. If it is a living body, what is going on? Karuna, you please focus here, bacha. So metabolism means chemical reactions are going on. If your body is living, definitely chemical reactions are there, right? Definitely chemical reactions are there. So metabolism means 2 words. Okay, one is catabolism, that is breakdown, and another is anabolism, that is build up. Okay, one is catabolism, one is anabolism. So cata means cut, ana, right? Cata means cut, means to break. That's how I remember. Cata means cut, means to break. So here in catabolism, you are talking about the breakdown. There is a compound, you are breaking it, catabolism, right? And when it is the anabolism, of course, anabolism means the build up. So there are 2 words, cata, ana, cata, ana. So metabolism is made up of these 2 words, catabolism and the anabolism. Okay, okay. Done. So just read it carefully, read it carefully. So we have learned that that even in simple organism, even in a bacterial cell, we know that there are thousands of organic compounds, right? These biomolecules are present there. And how do we express it? How do we express its concentration? Everyone in the chat section, please be active, revise it, guys, quickly revise it. Just a minute, bache, just a minute. Just a minute. If there is any confusion regarding the syllabus, na, let me tell you. Okay, so yes, the enzyme type is there, properties, enzyme actions, classification and nomenclature. Okay, fine. Then let's directly focus there. Okay, let's directly focus there. Let's not read it. Okay, let's leave it because it is not mentioned therein. It is not mentioned there in new syllabus. So let's not talk about the dynamic state part. So let's directly talk about the enzymes. So obviously that we will finish very quickly, है ना? Then we can finish this chapter till 2 a.m. What say? Let's finish it. So what are enzymes? Yes, for this also we are going to use the NCERT. So what are enzymes? Tell me people, what are enzymes? Tell me. What are enzymes, guys? But still I will advise you that about the metabolism, do read it. Metabolism is given in your living word chapter also. Okay, I'm not saying that you should know extra things about that, but at least read that particular paragraph of NCERT. Okay, so almost all enzymes are proteins. Yes ma'am, we know it very well, है ना? We know it very well that almost all enzymes are protein, but we have the exceptions also. And what is that exception? The nucleic acid that behaves like an enzyme, that is your ribozyme. When nucleic acid start behaving like an enzyme, that is your ribozyme, right bache? So just for your quick revision bache, do you remember in protein synthesis the peptidal transferase? Do you remember in protein synthesis the peptidal transferase? So that is an example of ribozyme. Understood? That is an example of ribozyme. Clear? That is an example of what? That is an example of ribozyme. So one can depict an enzyme by the line diagram. So an enzyme, like any protein, it is also having a primary structure, of course. What exactly is the enzyme? Enzyme is a tertiary structure. It is biologically active structure. That's what I told you. It is a biologically active structure. That's what I told you. So it is a primary structure of amino acid. Right bache? It is having secondary tertiary structure. So it is actually the tertiary structure. It is actually the tertiary structure, right bache, which is making it biologically active. Why? Because in tertiary structures there is the foldings, right? If you are following this lecture from the starting, you know that in the tertiary structure, right, we see that backbone of protein chain fold upon itself. So the chain crisscrosses itself, right? So there are many services or the pockets. There are many services and the pockets means the place where the specific substrate will bind. I'm repeating this point again. The primary structure of the protein is going to decide its specificity. It's specific na, even the enzymes, and the tertiary structure is going to decide its biological activity. Now when it is the enzyme primary structure, then it will convert to tertiary. So specificity is there, but actually that role, that biological activity will be done by that tertiary structure, right? It will be done by that tertiary structure. So the pockets are made there, and that services, that pockets are nothing but the active sites, right? So actually it is the crevices. I always call it as services just to remember this. Okay? I don't know, jaise hota na, sometimes you start reading a word incorrectly and lifelong you pronounce it incorrectly. That's my case. So it is crevices basically. I always call it as services. Okay? That's my fault. So it is crevices basically. Okay? Crevices basically. I rectified it many times, but you know, you start reading one word incorrectly, then you continue that. It happens with you or not? Or I am the abnormal one? It happens with you or I am the abnormal one only? Yes, tell me. Happened now? So it is crevices actually. The pockets are made. So once that pocket is the active site to which the substrate will join bache. So enzyme through their active site they catalyze the reaction at a higher rate, so they will transform that substrate into the product. But it is not a direct transformation. In between there will be the transition states, there will be the unstable stages as well, right bache? So enzymes, they are very different from inorganic catalysts. Now I will speed it up. I know it's getting too late, so let's speed it up as well. So enzymes bache, what I just said, that when you are talking about the enzymes, they are differ from inorganic catalysts. Why? First of all, here enzymes are biocatalysts. They are going to catalyze the living reactions, and they are highly specific bache. They are highly specific. They are very specific to the pH, very specific to the temperature, right? They need optimum pH, they need optimum temperature for their activity, isn't it? Isn't it? They are very sensitive to the changes in the pH and activity. But on the another hand, inorganic catalysts, they are not, right? So see, inorganic catalysts, they work more efficiently at higher temperature and at higher pressure, while enzymes get damaged at high temperature. Remember, I gave you the example of poikilotherms as well. I gave you the example of poikilotherms as well, right? Right. I gave you the example of poikilotherms as well, right bache? So enzymes are isolated from organisms who normally live under extremely high temperature, right? Jaise hot vents, sulfur springs, they are stable and retain their catalytic power. But here they are talking about the thermal stability of the enzyme. That is something very important when you talk about the enzyme, right? Thermal stability, that it can withstand that temperature or not. Normally, you know that if temperature goes above 40 degrees Celsius, enzymes, they get denatured. But we have the examples of archaebacteria, the thermophilic bacteria, the bacteria which lives under very high temperature conditions. So in that case, do you remember the example of Thermus aquaticus? Yes. Do you remember the example of Thermus aquaticus? From which we used to give, from this bacteria we get the Taq polymerase. From this bacteria we get the Taq polymerase, right? So it can withstand high temperature. So it shows the property of thermal stability, right bache? It is very important quality that these enzymes, they do not lose their catalytic power even at very high temperature. Clear bache? Even at very high temperature. Is that clear? Is that clear? Right? Right. So enzymes, they can increase the rate of the reaction. I know you must be like, yeah ma'am, this is also not given. This is also not given in the in that part. In our syllabus, ma'am. Directly let's finish the enzyme part. That's what we are going to do. Okay? So here in simple words, they have given one inorganic reaction. They have shown the transformation there. Nothing else. Nothing else. So they are saying that hydrolysis of starch into glucose is an organic chemical reaction. So rate of physical or chemical process refers to the amount of product formed. That's how we check the rate of a reaction, right? That what product will be formed at a given time. ठीक है बच्चे, at a given time. Is that clear? Is that clear? ठीक है. So now before it, let's talk about this part. Let's talk about this thing. Okay. Before starting that part, let's talk about the thing that how these enzymes, they can speed up the chemical reaction. I'll give you one example. I'll give you the example of carbonic anhydrase. You know na carbonic anhydrase? We discuss it in breathing and exchange of gases. Bache, it is the fastest enzyme. Do you know that? It is the fastest enzyme. Do you know that? Yes, bache, do you know that? It is the fastest enzyme. Do you know that? Yes. So it increases the rate of the reaction. Right, it increases the rate of the reaction, and you can say that it accelerated the rate of the reaction by about 10 million times. Do you know that? This carbonic anhydrase, which is the fastest enzyme, it accelerates the rate of the reaction by 10 million times. Do you know that? By 10 million times, imagine. And if you are not able to recall the carbonic anhydrase, let me tell you, we discussed the RBC, the plasma. When we talk about the carbon dioxide transport in breathing and exchange of gases, that it is CO2 plus H2O. So in the presence of carbonic anhydrase, it forms carbonic acid. It forms carbonic acid, and then that will further form bicarbonate and H positive. If you people remember, carbonic anhydrase, right? Carbonic anhydrase. Clear bache? Clear bache? right? Clear. So it accelerates the rate of reaction by 10 million times. So how that enzymes are going to do that? How are they going to achieve it? How are they going to increase the rate of reaction? Bache, it's very simple. They decreases the, right? They decreases the, yes, bache, everyone in the class, right? I know your energy is very low, very lame. This is the last topic, right? We can finish it till 2. So please show some energy. Please show some energy, right? Otherwise I will sleep. Okay, so decreases the enzymes, they decreases the activation energy, right? How are they going to increase the rate of reaction? They decreases the activation energy, right? They decreases the activation energy. Okay, so what is that activation energy? It is that, right? What is it? Bache, we have to convert the substrate into the product. We have to transform the substrate into the product. So how can we do that? By breaking and making of bonds, right? By breaking and making of bonds. Some bonds will break, some bonds will make. Some bonds will break, some bonds will make. That's how we can convert substrate into the product. That is simple thing. So if we have to achieve it, right? So there is some that minimum energy, that a barrier that we have to cross. Just say, let's say if you have to cross this wall, okay? If you have to cross this, right? Let's say you are standing here, right? And you want to go here. This is point A, this is point B. So to reach here, you have to cross this wall, right? You have to cross this wall. So what is going to happen by using the enzymes? What is going to happen if you use the enzymes? The enzymes will decrease this barrier. The enzymes will be like, chalo, I'll make it small, right? You jump very quickly and you reach here. That's how enzymes work. Okay, that's how enzymes work. Are you getting it? So same will be done here. What will be done here? The enzymes, they are going to break and make some bonds. They are going to break and make some bonds. But before that, there is an energy required to overcome the energy barrier. Like if I have to convert the substrate into the product, there is an energy barrier. There is an energy barrier and we have to overcome that. Right, the substrate has to overcome that. So energy required to overcome the energy barrier, right? Energy required to overcome the energy barrier of reactant and make it, right? And so energy barrier which is required, the energy which is required to overcome this energy barrier so that our substrate can form the product. So that bache Randhir, our substrate can form the product, right? This is actually that activation energy. This is actually that activation energy. Clear bache? Clear bache? Yes or no? Yes or no? So substrate needs to form the product. So there is an energy barrier. So that minimum amount of energy which is required to overcome that energy barrier so that substrate can become active, so that substrate can start the chemical reaction, that is the activation energy. So bache, if that activation energy, right, that is the activation energy. So if that activation energy will be low, okay, this is very important. If that activation energy will be low, so reactant or your substrate will immediately convert to the product. Immediately convert to the product. And this will not be done in one go. Let me tell you, this will not be done in one go. Okay, here in between there will be many transition states, right? These are the unstable states. There will be many transition states that are going to be unstable, right? Because here there is no proper making breaking. Some bonds are new bonds, some bonds are old bonds, so they are unstable. So this transition state will also always try to come to the stable state, right? This is our tendency. This is what we achieve. You are studying, you are learning. Why? Right? You want a good career, you want good money, you want to settle down, you want to go out, maybe you want to move to another country, right? So ultimately it is the stability that we all crave, right? So that's the point here. So substrate, we need to convert that substrate into the product. Right bache? And in between the transition states are there and that states are unstable. Okay, so activation energy need to be decreased. Clear bache? So right, so active sites are present in an enzyme. See this, So it is now understood that substrate has to bind the enzyme to its active site, which is present in that cleft or pocket, means that crevices, and I used to call it as cervices because I love that word. Actually it is crevices. So the substrate has to diffuse. The substrate has to diffuse towards the active site. It has to move towards that active site. So what will happen, bache? An enzyme substrate complex will be formed, and it is specific. It's not like any enzyme can convert any substrate. No, specificity should be there. That substrate should fit in that active site, right? We know about the lock and key hypothesis, right? If they have the compatibility, they will bind. And there is another hypothesis also. Jaise the conformational changes will be done, right? The substrate will change that active site position so that it can fit in. This is what we discussed. No, this is what we discussed. Okay, so that in detail is not required here, but this concept of activation energy, this graph is very important for your NEET paper. So first of all, you should focus that on x-axis progress of reaction is written, on y-axis potential energy is written. So this is the substrate, this is the product. First of all, what you need to observe, like when we are not using any enzyme, this activation energy is very high. So if that activation energy is very high, it will take time for the substrate to make the product. So when we use the enzyme, that enzyme is going to lower down the activation energy. And from the substrate, we will get product immediately. That's how the enzymes work. This is the role of activation energy, and this transition state, it is always unstable, right? It is always unstable. Clear, bache? So look at this. Potential energy of substrate is high, product is having less energy. Guys, substrate is having more energy, it is at this point. Product is having this energy, it is lower, means energy is released. So what type of reaction it is? It is exothermic. In exothermic reaction, the energy is released now. So what type of reaction it is? It is exothermic reaction. What type of reaction it is, people? What type of reaction it is? It is exothermic reaction. Are you getting my point? It is exothermic reaction. Right? Done, bache? Okay, done. So this complex formation is a transient phenomena. So during the state where substrate is bound to the enzyme site, a new structure of the substrate called transition state will form, right? And here you know that after the expected bond breaking and making is completed, product is released from the active site. So that's how the conversion will be there. So what is it? It is the exothermic reaction. So bache, in endothermic reaction, if you remember, in endothermic reaction, what will be the point? Right, substrate will have low energy, product will have more energy. So if that type of graph is there, then it means endothermic reaction, right? Because energy is absorbed here. Okay, the energy is absorbed here. Is that clear? So this is exothermic and this one is endothermic. Understood? So this is about the conversion. So finally the transition state will be converted back. So whatever is written here, I have already explained you this. Just for your quick revision, you can see y-axis represent potential energy, x-axis represent progression of the transformation, means it is representing the rate of the reaction. Okay, bache? Rate of the reaction. Clear? So that's what you need to figure out. So nature of enzyme actor, you know that enzyme plus substrate, enzyme product, and finally enzyme will be released. So bache, this is what we need to keep in our mind, that enzymes are not going to be utilized in that reaction. Once they will do their job, they will be released, and that enzyme will again be used for the new reaction. Right, Krutika? We can again use this enzyme for the new reaction. So enzyme plus substrate, they will form complex. Then this enzyme will convert the substrate into product. So once the product will form, enzyme will say tata bye bye to product. Enzyme will be like, I have to do another job. Okay? So enzymes are not directly involved into the reaction. They will do their job and they will leave. They will do their job and they will leave. That's how it works. Understood? That's how it works. Understood? Clear, bache? So first, the substrate bind to the active site, will fit into the active site. So when substrate will bind to the active site, it will induce the enzyme to alter its shape. Enzyme will alter its shape. Conformational changes will be there in the enzyme. What will be there in the enzyme? Conformational changes will be there in the enzyme. So fitting more tightly around the substrate. Okay, fitting more tightly around the substrate. Are you getting it? Are you getting it? So the active site of the enzyme now in close proximity of the substrate, it breaks the chemical bond, bache, right? New enzyme product complex will form. So enzyme releases product of reaction and free enzyme is ready to bind to another molecule. So that's how it works. That's all. So what are the factors that affect enzyme? I have already discussed, bache, that when it is the temperature, high temperature will denature enzyme, will break its structure. Low temperature will deactivate enzyme. So optimum temperature is required. Optimum temperature is required. Understood? Optimum temperature is required. Same for the pH. Optimum pH is required. Neither high pH nor low pH is beneficial for the enzyme activity. Okay? So how come the substrate concentration? But whenever we talk about the substrate concentration, we discuss Michaelis-Menten constant. What we discuss? We discuss Michaelis-Menten constant, right? But actually, see, just let's say, now I am teaching from last 7 hours, okay? I am teaching from last 7 hours. Even if I am teaching very good, right? Even if you have that, even in the starting you add that, na ki yaar, today I am going to complete this lecture. Still, even if I am teaching very good, you cannot listen to me for 6 to 7 hours. You'll be saturated. You'll be like, ma'am, bus. Can you take this lecture tomorrow? You'll be more productive. Am I right? Am I right or wrong? You will be more productive. Ki ma'am, can you take this lecture tomorrow? Hai na, we are tired. We cannot retain it. So up to some, right, up to a particular time you can retain whatever I am telling you. After that it will be like, no ma'am, not possible for us. Saturation. Just for an example, there is a reaction. Enzyme molecules are there and there is the substrate. Just initially when we increase the substrate concentration, okay, initially when we increase the substrate concentration, initially let's say we have started increasing it. Now let's say you have 20 substrate molecule, 5 enzyme molecules. This one enzyme, let's say one enzyme can bind to 4. One enzyme, just a minute, let's say you have 10 substrate molecule, you have 5 enzyme molecule. A layman example I am giving you, okay? A layman example I am giving you. You have 10 substrate molecule and you have 5 enzyme molecule. One enzyme can bind to 4 substrate at a time. So obviously one enzyme will bind to, one enzyme will bind to 4 substrate, 2 enzymes will bind to 8 substrate, 3 enzyme is still having 2 substrate. Means if I will increase the concentration, let's say I made it 12, so obviously rate will increase. Let's say I made it 14, obviously rate will increase. Let's say I've made it 16, rate will increase but not up to that extent. Now let's say I made it 20. What is going to happen? The enzyme, all its sites are occupied. One enzyme can bind to 4 substrate at a time. 5 enzyme molecules will bind to all the substrate molecules. They will start their job. They will break and make the bonds. But now after 20, if I will keep increasing, right, after 20, if I will keep increasing the substrate concentration, the rate will not increase. The rate will not increase because saturation is there, right? All the enzymes molecules are occupied. They cannot act on the new substrate molecule, so the rate will not increase. Okay? So substrate concentration, when initially it increases the rate and then saturation occurs. So see here, this is substrate concentration, this is the velocity of the reaction. So when bache, see, Km is what? Michaelis-Menten constant. In detail, there is no need to read that. But ultimately, what you have to keep in your mind, that Km is the concentration of substrate at which the rate of reaction reaches half of the maximal velocity, right? Km is the substrate concentration at which the rate reaches half of its maximum velocity. I am repeating it again. Km is the substrate concentration at which the rate of reaction reaches the half of its maximal velocity. Jैसे let's say maximal velocity is 100. So Km is the concentration at which the maximal velocity has reached 50. it has reached 50. That's what you need to remember. This is the Km. So that's how the substrate concentration affects, right? That's how the substrate concentration affect. Now look at this graph. So pH, if it is very high, sorry, if it is increasing, right, optimum pH is required. So optimum pH means maximum enzyme activity. So if you increase it, enzyme activity declines. If you decrease it from optimum value, enzyme activity declines. Same is the for the temperature. Optimum temperature means maximum rate. It will go down. Even if you will increase the temperature, enzyme will be denatured. If you will decrease the temperature, enzyme will not be able to act. So this is about the graph. So that is how the concentration affects. Okay? That is how the concentration effect. Done bache? Done bache? Clear? So now next is what? Concentration of substrate done. With the increase in substrate concentration, velocity will increases at first. So when it reaches the maximum, which is not exceeded by further, so enzyme molecules are fewer than the substrate. Always remember. So that's what I have explained. Okay. So now activity of all enzyme is also sensitive to bache some specific inhibitors, some specific protein. Jaise there are some specific protein that can stop enzyme activity, right? That is inhibition, and that proteins are inhibitors. That proteins are inhibitor. Sometimes these proteins, they compete with the substrate, right? They don't let that substrate to bind with the enzyme. Just we talk about the cholesterol synthesis. Okay? In the chapter microbes in human welfare, we talk about the statin. So statin will not let HMG-CoA, right? That enzyme is there, na? The enzyme for the cholesterol synthesis. So statin will compete with that enzyme. Statin will not let that enzyme to bind with its substrate. The cholesterol will not form further. So that is the example of competitive inhibition. Sometimes non-competitive inhibition is there. Sometimes that inhibitor, they change the active site, right? They don't let that substrate to bind there. Clear? So when the inhibitor closely resembles the substrate in its molecular structure and do not let that substrate to bind to the enzyme, this is competitive inhibition. So question can come from competitive inhibition. Okay? Okay? Thank you, Bache Malai. Right? That is competitive inhibition. So due to the similarities in between the substrate structure and the inhibitor, it will not allow it to join there. Right? So substrate cannot bind, and as a result, the enzyme action declines. So inhibition of succinic dehydrogenase, you know na, succinic dehydrogenase, we discuss it in Krebs cycle, right? So malonate will act like an inhibitor for it. Okay, okay bache. So this example is important. I gave you the example of statin also, that is also important. Okay, so examples are important from this part. Okay, so now bache, so it is OT Halil. Okay, so let's talk about the next part. The trick here is OT Lil. So what is it? Yes, it is OT. It's not OT Hill, it is OT Halil. It is not OT Hill, it is OT Halil. It is OT Halil, Lil, L-Y-I-L. That is the meaning here. Do you know what is it? OT Halil, simple bache. O means oxidoreductase. These are the 6 main classes of enzymes. What are they? These are the 6 main classes of enzymes. Yes, students, what is it? It is the 6 main classes of enzyme. Okay, so O, oxidoreductase. T, transferase. H, hydrolase. This lyase. Isomerase. Ligase. Okay, so oxidoreductase, obviously oxidation reduction will be done by this enzyme. Just your dehydrogenase, right people? That is your dehydrogenase. Transferase, that will transfer the functional group. Hydrolase, what that hydrolase will do? Yes people, what that hydrolase will do? Tell me what that hydrolases will do. So bache, it catalyzes the hydrolysis by addition of water. So from hydro, what, this is the hint that you have to pick. So it catalyzes the hydrolysis by adding water. It catalyzes the hydrolysis by adding water. So hydro word, from the hydro word you have to pick up the hint. Clear? You have to pick up the hint. Clear बच्चे? Then comes liases, right? So removal of group will be there from substrate and double bond will be formed. Liases में removal of group will be there from substrate and the double bond will be formed. So it is not hydrolytic. Group will be removed, but there is no addition of water. Group will be removed, but there is no addition of water. This is what you have to remember. Isomerase will convert one form into its another form. Isomer and ligase means joining, right? So understand the basic meanings here. So what's the trick? It is OT Halil. So oxidoreductase. So bache, that's your homework. In the comment section you will tell me about that names, that ma'am, this is that particular enzyme that is doing oxidation reduction, this is that particular enzyme that is doing isomerization. Okay, so enzyme which catalyze oxidoreduction between two substrate. So one is reduced, one is oxidized simultaneously. So see, S dash is here, it was oxidized, now it is reduced. Okay, transferase is transferring the group, so G is transferred to S dash. In hydrolysis, right, there will be the hydrolysis, but by the addition of water. Hydrolysis of ester ether peptide, but how bache? By the addition of water. This is extra that you have to note down, by addition of water. And in liases bache, double bond will be generated. What will be done? Double bond will be generated. Clear bache? Double bond will be generated ultimately. So water is not used for hydrolysis here. Okay, so next is isomerases. So all enzymes catalyzing interconversion of optical or positional isomer. One isomer will be converted to another. Simple it is. And then ligases, molecular glue it is. You know that it joins that sugar phosphate backbone, right? That carbon or oxygen bond, carbon sulfur, carbon nitrogen, PO, like this. Okay, so it is the molecular glue, that's all. So the last topic of the chapter, biomolecule, that is the cofactors. Okay, so bache, there are two categories when we talk about the enzymes. There are two categories when we talk about the enzyme. One is your simple enzyme and one is your compound enzyme. So in simple enzyme na, it is only protein. There is nothing like non-protein part, only protein, right? What will be there? There will be only protein. There is nothing like non-protein part. Clear bache? There is nothing like non-protein part, only protein will be there. This first point clear? Tell me, this first point clear? Yes bache. So it is your trypsin present in pancreatic juice. It is a proteolytic enzyme. Now when it is the compound enzyme, it is also known as complex enzyme. Fine, my screen is also not working. It is also known as complex enzyme. So bache, this enzyme is having a protein part. This enzyme is having a non-protein part. So here in this particular enzyme, if both the parts will join to each other, then only enzyme will be functional. So it is having protein part, it is having non-protein part. If both the parts will join together, then only the enzyme is functional, otherwise it is not. Okay, otherwise it is not. Protein part, non-protein part. So protein part is known as apoprotein. Very important, it is definitely one question will come from this part. And the non-protein part bache, it is the cofactor, right? It is the cofactor. So basically when it is a complex enzyme, so it is the hollow enzyme. That hollow enzyme is equals to apoprotein. That hollow enzyme is equals to apoprotein plus cofactor. It is apoprotein plus cofactor. So apoprotein is the proteinaceous part, cofactor is the non-protein part. And when both of them will join, the enzyme will become catalytic. Then you will call it as hollow enzyme. Understood? Then we will call it as hollow enzyme. Clear bache? Clear bache? So three types of cofactors are there. One is your prosthetic group. One is the prosthetic group, one is the coenzyme, and one is the metal ion. One is the metal ion. Bache, metal ions are inorganic. They are inorganic. Just say all the enzymes of glycolysis, they need magnesium, metal ion. Prosthetic group and this coenzyme, they are organic cofactor. जैसे prosthetic group. What's the main point here? It should be tightly attached to the apoenzyme, then only it will be functional. It has to be tightly attached to the apoenzyme. The coenzyme, it will be loosely attached. That's all. This is all about this cofactor part. Okay, this is all about this cofactor part, and you should know the examples. Okay, you should know the examples. Is that clear? Then you should know the example. So let me explain this part to you. So cofactor, so enzymes, they are composed of one or more several polypeptides. ठीक है बच्चे? So there are number of cases in which non-protein part is cofactor is attached to the catalytically to make the enzyme catalytically active. ठीक है बच्चे? So that flow chart that I made now, that will clear everything, Chetu. Right? That will clear everything. Clear बच्चे? So three kinds of cofactors are present: prosthetic, coenzyme, and metal ion. So prosthetic group is what? It is organic and tightly attached to apoenzyme. Okay, so जैसे peroxidase, examples are very important बच्चे. Peroxidase and catalase, they catalyze the breakdown of hydrogen peroxide to form water and oxygen, and heme is the prosthetic group. Okay, that's examples are important. Now when it is the coenzyme, it is also organic, so it will associate with apoenzyme in transient form, means it is not very tightly attached. Okay, so it will remain attached during the catalysis only, during that chemical reaction only. Okay, so here you can see this dinucleotide phosphate, right? They contain the vitamin niacin. So coenzyme nicotinamide adenine dinucleotide, right? And nicotinamide adenine dinucleotide phosphate, they contain vitamin niacin. Examples are important, Ikra. Okay, and then finally the metal ions. जैसे zinc is a cofactor for proteolytic enzyme carboxypeptidase. Very important MCQ. That's it. This is what you need to know. जैसे magnesium ions are very important for what? For the enzymes of glycolysis. So these are the few terms that you need to know. So this is all about the chapter. We'll share the PDF in the Telegram group, guys. Now after the class, I don't know why this lecture is again and again there. ठीक है बच्चे? So what you have to do? So what you have to do, so you have to like this video, you have to put a comment, right? After the class you are going to revise this chapter. I will keep the special class of this question practice session after few days, right? I will not keep it tomorrow. It will be there on 8th of February or on 9th of February. ठीक है बच्चे? And these videos are must watch. Pankhuri ma'am has also posted the important chapters. You should check it as well, right? And you should make sure that you have completed that. Okay, so see, and whatever changes are there in NCERT, in this video I have explained, even about the old NCERT, the new NCERT, so it is also a must watch. Okay, so here in the description of this video, even in the description of this video, there is a link for the crash course batch Endgame 2.0, and that is one of the best batches bache. The best team is there, the proper doubt solving, proper test, proper revision. We are starting from the scratch, right? So if you are skeptical, if you don't know that you know your basics are not good, you should go for the crash course or not, yes, this batch is your best choice. Okay, so the coupon code Ambika 10, this is the coupon code that you people need to use. Okay, so you will get this batch at a price of 4499, and trust me, you will get test series, proper revision, and the revision will be there from the scratch. So currently we are doing human physiology. Okay, so join it as soon as possible. Please do not waste your time. Do not waste your time. Okay, so we'll wait for your comments. I have covered everything. This lecture is more than enough for biomolecules. Don't be like, ma'am, at the end you finished it very quickly, but I have covered all the important points, and everything is covered from this chapter. Okay, I'll suggest you to read that metabolism part, the dynamic state part, and in the comment section do let me know which chapter you want me to start next. So I'll go as per your preference. Okay, so take care, bye bye, thank you. Do revise this chapter, people. Good night.